Um, I wanted to check something actually. Anyways, uh, uh, um, no, yeah, sorry, 55. Never mind. It's time. Alrighty then. Maybe we can start a little bit today off of watching the guy. I guess. Might as well. I'll be back. Let me get something to eat. Well, actually, no, I want to start it off with ab exercise. Hold on. Actually, I need to put some clothes in the washer. Be right back. Hold on, guys. I put now we have a bunch of cups on the table. Everybody know drug addicts. Let me sure it's not some good stuff right there. You already know. Alright, hold on. Hold the phone. Hold on the phone. Hold on the phone. Let me set a timer for the washer. Whoa. Oh, buddy. You know what? Maybe I should just grab this. It's like, maybe we can put this. Maybe just put this on that side, huh? It's nice today. It's very nice. Very nice. Hopefully charge a little bit. Hey, how's it going? Sorry, we'll get started here in a bit. Who's the baller, man?
What I was writing on. And 5 years later, here I am. And I'm teaching my own course in this exact lecture hall that I was in. So let's chat. So I'm going to start off with a little story about this lecture hall specifically. So obviously I went to Brooklyn College, and the first class I was ever in when I came to Brooklyn College was an intro bio course. And that intro bio course was taught by Professor Matt.
Sure. Whoa! What's happening here? Oh no, something broke. Oh well. We'll worry about it later. All right, buddy. That's what you got. Let me just go watch my answer book and just get some breakfast, and we'll watch this guy. Look at this. Perfection. Perfection. Ta-da. Can you guys see? Perfect. Chef Boyardee. Get her. Tell her. Tell her. Tell her. You tell him. All right, guys, I got my milk. Happy, happy, happy. Happy, happy, happy. So happy, happy, happy. You know? You know? All right. Yep. All right, patient. Well, every time I do, it makes me laugh. I'm like, yep. What do we got here today?
Here, which is a fantastic professor. I hope that you guys all have the opportunity to cross paths with her. But she taught in this lecture hall, and that was my first class ever. And I was seated right there in like the third to last row, and I can remember where I was sitting. I can remember what I was writing on. And 5 years later, here I am, and I'm teaching my own course in this exact lecture hall that I was in my first day of college. So that's crazy. But hopefully, I come with a newfound understanding of how to teach and how to learn and what it means to succeed. Right? Come on in, take a seat. My name's Yusuf. Nice to meet you guys. I'll get all your names in a bit, but before we do that, let's just talk a little bit about, what are we getting ourselves into? What do we necessarily mean when we say, like, let's study for the MCAT, let's prepare, let's get ready to take this big exam. So what is the MCAT? What does it mean? Why is it important? The MCAT is hard to you guys have seen that ad before, the first review ad, right? So yeah, the MCAT's important, but it's not impossible, and it never has been, So the MCAT is basically just this little holistic knowledge game about how much you know about pre-medical studies. And you'll hear a lot of people tell you that pre-med doesn't really matter, like it's not the most important thing in the world, it's not relevant to medical school. That's a load of bullshit. Pre-med is probably the most important arsenal of knowledge that you'll ever get.
Oh, man.
Well, what are these living things that we care about? We care about humans, we care about bacteria, and we care about viruses. Are viruses living? No, they're not. So they don't get included in living things, right? So in living things, we care about bacteria and we care about viruses. But so much of biology overlaps with non-living things that every now and again we talk about things like viruses and prions and things like that, right? Now, before we talk about living things as a whole and building our understanding up to how does a human physiologically function, what are living things made of? Cells, right? That's chapter one. Chapter one is the cell. Right, chapter one is the cell. That's where all of it begins. That's where we're all going to start. Hi, how are you guys? We're good. Thanks so much. Guys, this is just doing it. Thanks to him that I was able to come here. Thank you so much for having me. Thank you for inviting me, of course. All right. Okay. Okay. So chapter one, we're going to talk about the cell. What the cell is, what the cell does, blah, 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 blah. So this is a general question for the room, right? And this is what we mean by foundationalizing our understanding of science and scientific principles. Who can answer a very, very basic question? What is a cell? What is it? Has anyone ever asked you the question? Has the question ever crossed your mind? Maybe not, because sometimes professors who are much more knowledgeable than I am will overlook these very basic facts in order to get you to information that's actually going to allow you to succeed in their class and in future graduate studies if you choose to pursue them. But without this knowledge, you're not even able to appreciate those concepts, which is why throughout this course, you're going to hear me say, I am not a professor. I am a student, and I have a student-level knowledge of all of these concepts that may or may not help you go in the right direction, okay? I, at one point, knew as much as you did, or as little as you, or maybe you know more than you, but hopefully you can pick up on one or two things that I'm going to teach you. So what's a cell? A cell, in my definition, is a compartmentalized, organized, and quote-unquote well-oiled machine that can undergo various life processes. It's a machine, right? It's a little engine. It's a little engine that could. You know what I mean? So what do we mean by compartmentalized? What is this compartmentalization I'm talking about? Does that know? Yeah, so it's split up. It's split into what? Split into organelles. What's the purpose of having organelles? Well, before we talk about that, I'm going to give a little asterisk here. And who knows what right up the asterisk? What types of cells have organelles? Eukaryotic cells. Only. Do prokaryotic cells have well-defined and well-structured organelles? They do not. Prokaryotic cells are this weird soupy mishmash of genetic information and life-producing capacity, right? But there are certain things that every single cell is capable of, regardless of how they're defined, and we're going to talk about that in a second. By organized, I mean that each process occurs in a controlled manner. What do I mean by that? Well, when you get biochemistry, we're going to learn about this little molecule, this hormone called insulin, right? And insulin can react in cells in a very specific way in order to increase glucose uptake and increase glucose, breakdown and synthesis, blah, blah, blah. But it also, so positively affects a lot of glucose metabolism. Also, I know my handwriting is fantastic. If you need me to read something out, please let me know. It pro-affects glucose metabolism, but it also negatively affects fatty acid breakdown. Right? And that's really interesting thing that we're going to talk about called positive and negative feedback. And basically all of your life processes run on this sort of foundational understanding of positive and negative feedback mechanisms, right? Where something is affecting this in a positive way and pushing it forward, and something is affecting this in a negative way and pushing it back, right? We were just talking about this when we were studying cardiology, right? Where your heart, if you didn't know it, would like to beat at 140 to 160 beats per minute for your whole life. However, due to a constant level of regulation by your parasympathetic nervous system, it beats at roughly 60 to 100 beats per minute, right? That is called negative feedback or negative control, correct? So there's a sort of inhibitory process that occurs to organize those processes inside your body so that you don't blow up, right? So you don't die, right? Going back well, wait. Everything happens in a fluid manner. One thing leads into the next, leads into the next, leads into the next. So when we talk about compartmentalization, and we talk a little bit about how things are split up, blah, 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 you're going to hear me talk a lot about, well, this thing leads into that one, and that leads into that one, and that leads into that one, and then finally you get a result. Where DNA is in the nucleus, and then it gets into mRNA. And the mRNA exits the nucleus, and then it goes into the rough ER, which starts with the ribosomes. The ribosomes turn into a protein, and the protein goes to the Golgi apparatus, where it gets modified and packaged and turned into a vesicle. That vesicle travels through the cell, merges the membrane, and is exported out. You see how one thing led to the next, and led to the next, and led to the next. That is the well-oiled nature of a cell. Does that make sense? Very cool. All right, so I think I should have. I didn't bring anything to erase this one, so don't touch it, or even mask it. See how better that is? I had a professor back in the day. He's the greatest professor I've ever had in my entire life, and there's no competition. His name's Professor Lenny Levitsky. If you guys ever have the chance of coming across him, he's quite literally like the worst person in the world, but the best for your education. He didn't go to Florida. I just told him. I spoke to him the other day. He knows that I'm doing this. So he's kind of the person who inspired me to do this in the first place. So Professor Lenny Levitsky, he always looked like he was erasing the board with his hand, but really like the palm of his hand is this tiny, tiny piece of tissue paper, and that's what you use to erase all this stuff. Now, when I started teaching, and I put a whiteboard up in my room, and I had a little tripod because I was teaching, I tried using those erasers, and they just didn't do the trick. So I was like, this man must have been up to something. So I took a little piece of toilet paper, and I put it inside my hand, and I erased it just completely effaced the board. And that's why I do that now. Shout out to Lenny Levitsky. So, you know how many sections? How many sections? Like, how long? Like the actual course? I don't know. I really don't know. I wish I did. I'm like writing up a syllabus now as we speak. So I'm gonna have an interview by like next week. Tuesday, Thursday. I'm here on Tuesday, Thursday from 6 to whatever we get done, but never past 9. That makes sense? So today probably are like first 2 chapters of biology, right? My laptop's charging over there, so I need that to teach. And my charger gave out, which is one of the unfortunate things that happened today. But I'm using a different charger that I have. It's just charging really slowly. So we're gonna teach a little bit of the stuff that I remember before we look at my laptop and the rest of the information. So we talked a little bit about like what cells are in my definition, right? But now let's dive into the textbook and see what the textbook has to say about cells. And the first thing that the textbook says about cells is this blanket statement called cell theory. Let's talk about cell theory. Is it cell like this character from Dragon Ball Z or something? Yeah. Let's talk about not that guy, but let's talk about cell theory. So cell theory has 4 components. Number 1 being like one of the first things that we actually said when we came in. So number 1 being all living things are composed of, say with me, cells. All living things are composed of cells. So anything that's living is named cells, right? That's quite basic knowledge, right? Let's look at number 2. So cells are the functional unit of life. Now, I use a lot of shorthand when I write, because it just makes things faster. So whenever I write functional, I go like this. So that means function, and that's AF, function, right? A cell is a functional unit of life. What do we mean by that? What's the difference between statement 1 and statement 2? Well, you can use flour to make bread, right? But it didn't all come from the flour. Something was turned into the flour, right? So all the things that compose the cells is like bread is made of flour. But cells are the functional unit of life. That's saying that flour is the functional unit of bread. But there are other things that compose those cells, meaning there's other things that make the flour, right? So it's not that the flour was just there and then made the bread. So the cells are not just there and made the life. That's what we're saying. Right? What we're saying is there was something in between that came together into the cell, and then the cell became the functional unit of life, and then the cells are now what makes up all life. Right? Cool. So number 3. I don't remember. So, cool. Cells form from pre-existing cells. Right? Cells form from pre-existing cells. Well, what the hell are you laughing at? Number 4. Cells carry genetic info that they pass down onto other cells. What are we talking about here, and what are we talking about here? So asexual and sexual reproduction both, right? So what we mean by saying reproduction, this is kind of confusing. And the reason you really want to get a grasp on this is because the MCAT does kind of ask about it every now and again, where they're like, oh, well, if this would happen, what rule of cell theory is being violated here? Or like, how do viruses not apply to cell theory, which is something we're going to talk about in a second, right? So cells form from pre-existing cells. Well, we have a cell, and the cell buds off of it and becomes whatever it is, right? But if that cell doesn't have genetic info, it can't do the same thing, right? Or it can't code for a protein that it needs, or it can't code for some sort of hormone, or some sort of blah, blah, whatever it is, right? So we need that genetic info to be passed on when the cell arises from a pre-existing cell. Because this statement doesn't mean anything to us if the viability factor, which is the genetic information, isn't passed on as well. Does that make sense? So what we're talking about is we have a cell, and it's made up of a bunch of stuff, and somewhere in that cell inside of the nucleus, we have genetic info that gets passed on to a daughter cell in one way or another. And if there's equal division, it's called mitosis, and if there's an unequal division, it's called meiosis, right? Guys, by the way, if you ever need to leave, if you need to walk out, if you want to eat here, if you want to do whatever the hell you want. At the end of the day, there's no one here, so you need talking to a camera, right? So don't feel bad about leaving or coming in late. Okay, cool. So this gives us mitosis / meiosis, and this gives us organelle theory, right? And those are the two main things that we're going to talk about for the rest of the chapter. So one thing that a lot of people understand is like, oh, well, they bring up cell theory, and they just never talk about it again. They do. They're just not telling you that they are. Because the rest of the chapter is elaborating on cell theory. So while you guys write that down, I'm going to check to see my laptop. It is. That's good. Come get here, and you're just going to walk away. And I'm walking away, I'm not ignoring you, I promise. Right? So let's talk about, and now you know this, right? Do you guys have a grasp on that? Yes? If I'm going too slow or too fast, please let me know. If I'm saying something that you already know or something that's obviously like, you know, you already know, you went in like 8th grade, please tell me to shut the fuck up and like move on, right? Okay. Right, so let's talk about our first question. I just love this part. Let's talk about our first question. What makes viruses not alive? So we talk about eukaryotes, we talk about prokaryotes, we talk about viruses, right? Okay, that's our hand up there. A little bit of projection. Yeah. So viruses, number one, they can't reproduce. And I like you said, like cells can. Because clearly, viruses have made it this far, right? And they can reproduce. And we saw a really good example of that 3 years ago, right? So clearly, viruses are capable of reproduction. But what do they need in order to reproduce? They need a host. They need a cell. They need something living to do the work for them. Because viruses don't have ribosomes, and viruses don't have nuclei, and viruses don't have Golgi apparatuses, and viruses don't have peroxisomes, and they don't have like cytoplasm, things like that. They're just creepy, crawly bundles of genetic information. That's it. And it kind of poses an ethical dilemma. Like, if that's not alive, but it can proliferate, like, why is it not alive? Why is this our definition of what alive is? But it is, right? So, right, what else? What makes viruses not alive? So viruses don't form pre-existing cells. And the second thing is, they don't pass on to daughter viruses, right? They don't pass on to other viruses. What do viruses do in order to reproduce? We're talking about this in the chapter, you guys don't know. But what do they do to reproduce? They take their genetic information and inject it into a host, a cell, right? And that cell, because it's programmed to do so, takes the genetic information, runs it through the machine, and reproduces these proteins. And miraculously, because of beautiful chemical interactions, the proteins reassemble, and it either lays dormant inside the cell, or it blows out of the cell and transports itself to the body. Any questions about that? Those two things that I said before, either lies dormant or blows up the cell of these, those are the two things known as what? They're two types of cycles. The lytic and the lysogenic cycle. We're going to talk about that a little bit when we get into viruses. But the existence of the lytic and lysogenic or lysogenic cycle is more indication that viruses are not living things. Does that make sense? Yes? Good. Let's have a short discussion, a very short discussion, about eukaryotes and prokaryotes. Okay? What's the first thing we put up there? Sorry? Exactly, the way that they're working. Feel free to come down. How are you? That's it. So this one has membrane-bound organelles. This one's basically just one big organelle, right? So whereas eukaryotes use the mitochondria and the inner membrane of the mitochondria, and like the mitochondrial matrix, the intramembrane, whatever, I forgot what it's called, like I'm blanking on the word, the intramembrane states, right? So whereas, you know, eukaryotes use that in order to power the electron transport chain, the same gas cycle, blah, blah, blah. What do prokaryotes do? They just use their membrane. They just use the membrane in order to produce energy, produce ATP, and undergo all those processes that we do. This brings up a really interesting theory that we're going to talk about in a little bit, and gold star to anyone who knows the name of the theory. What is the theory of this sort of merging of prokaryotic, eukaryotic sort of cells way back in time, hundreds of years ago? Endosymbiotic theory. Endosymbiotic theory is a really, really important thing inside of the sort of foundation of cellular and molecular biology. Can you tell me what endosymbiotic theory is? You're absolutely right, right? You're absolutely right in the fact that a cell ate another cell, and that kind of became an organelle. That organelle was the mitochondria, right? So there's a cell that's already producing its own energy, and another cell came along and engulfed that cell and created a environment within which that cell could undergo all these energy-making processes in a controlled manner and for the benefit of the other cell. It's like a parasite, right? But this parasite is giving you energy, and you're feeding it nutrients, right? So, interesting thing is that all these people in this room, right? And all the mitochondria that we have in all of our bodies, how many? Trillions. What? Who knows, right? Where do they all come from? One person. One single, the first female human ever produced every single mitochondria in this room. Because mitochondrial inheritance is from the mother. And I would write that down if I were you, because it's a very, very hot MCAT fact. Whenever you see the words mitochondrial disease on the MCAT, you can bet your sweet ass they're going to ask you where you got it from. Every single mitochondrial disease is inherited from the mother. That's not to say that it's X-linked. Those are different things. You inherit a mitochondrial disease from your mother. Because you get your mitochondria from your mother. Why? Why can't you get your mitochondria from your father? Yeah, the sperm doesn't have one. It doesn't have a mitochondria. It's not capable of producing that level of energy by itself. The egg cell is so much larger than the sperm that it carries almost all of the thing, the sort of material that becomes the embryo, right? So maybe 1% of whatever's left comes from sperm. But what does come is half of all of the genetic information, right? It's containing that 1% of information, right? All right.
There's some, but it's just negligible because there's just so much in the egg. But yeah.
So, what else about prokaryotes? Well, they're single cell, right? The bacteria.
Hold up. What's he got? Can't do this. Yeah, it's pretty much what I want to do. It's like,
H. pylori, E. coli, right? Clostridium difficile. These are all single-celled organisms, right? And they've evolved in different ways to have different effects on the human body, and that's how they survive, right? So let's talk about mycobacterial tuberculosis, which causes what do you mean causes? Tuberculosis, right? So it infects the human body, and it causes you to cough and sneeze, blah, 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 and you pass on to another human being, and they replicate their body and cough, and you give someone else, blah, blah, blah. And that's how they survive. It all happens in one cell. And since they're single cell, it's a lot easier for them to reproduce. What do humans need to do to reproduce? Rhetorical question, right? We'll get there later. The next chapter is actually reproduction, and I don't know how far we're going to get into like that chapter, because in reproduction, if you guys are like reading the textbook before you come here, which you don't have to do. All my lectures are made so you don't need to study all before you come to them. Which is why we're going really slow. But I'd rather go like this than overstep and not cover something that someone has read before, right? So when you get chapter 2, you're going to get smacked across the face with this very long page about the menstrual cycle. And it's really damn difficult to understand because there's like 5 different hormones at play and different levels and different things going on. Blah, blah, blah. You say tropic and non-tropic and downstream effects and positive and negative feedback mechanisms. The hypothalamus, you're like, what the hell is the hypothalamus? They never explain it to you. I'll teach you. That's what I'm here for. Okay? So that you don't cry at your desk like I did when I read that page, right? Okay, cool. So single cell, right? And another thing here is what? No membrane bound organelles. No membrane bound organelles, right? So, and eukaryotes, very specific wording here by the textbook, not by me, can be multicellular. Can be multicellular. What's an example of a unicellular eukaryote? Huh? First of all, I think that toes the line between both of them, right? What else? Right. An egg cell? I don't know if that ball has an organ. It's like a cell, right? But yeah, that's eukaryotic cell for sure. That's membrane. Yeah, I would say that. But I think yeast. I think yeast have organelles, if I'm not mistaken, right? Those are single cells, correct? Okay. And honestly, that is really all you need to know about prokaryotes and eukaryotes. That's like it. That's the basic level of knowledge you need to perform well the MCAT, right? Now, jumping back on what I'm doing here and like why I'm spending my time doing this, right? Like I said before, I'm here to teach you how to do well in the MCAT, right? But I don't really care if you don't do well in the MCAT. Because I have friends in medical school who didn't do well in the MCAT, who personally did pretty poorly on the MCAT, like got below an 80th percentile and still study with me in the same rooms and the same lecture halls and take the same tests and do just as well as I do. Because the one thing I want to say from the start is that this exam will never define your success as a medical student or a physician, ever. It will never determine who you are. The only thing that I'm here to do is give you a foundational understanding and appreciation and passion for the information that goes on this board. Because if you genuinely care, you'll do well. Let me tell you a little bit about my story. So I started studying for the MCAT in 2021. It was a really bad time in the world. I was locked up at home, not coming back for my next semester, blah, blah, whatever it is, and I was pretty lonely, to be honest. I wasn't meeting out with friends. I didn't see my family all the time. They were at work or coming back, blah, blah. Some people my family got sick, and then I had my wisdom teeth removed, and I had to take like 2 weeks off studying, so I was on a blah, blah, and I was just getting along, right? But the MCAT gave me something to look forward to. It gave me some form of solace from the noise that was going around me. And I became so passionate and so headfirst driven into everything that I was studying that I genuinely gave a shit. I actually cared. And if you didn't care about anything you were learning before, which is fine, I beg you to try to care now. And the way that I developed a care in the world was by studying every single day. They say that once you do something enough, you learn to appreciate it, regardless of where you first started. So once I hit the 100, 150, 200, 300, 400, 500 hour of studying for one exam, I had a deep respect for the information I'm giving you now. And that's why it was related in a way that matters. Because I have a love for the things that I've learned. And it developed because I gave a shit. And that's all you need. That's all you need to do well. The people who didn't do well in the MCAT, why are they in medical school? Because they gave a shit. They cared. They're some of the most passionate people I've ever met in my entire life. What score did I get? I got a 98 percentile score. There are people at my school who got the 78, 80th percentile who I will always say to the day I die that are smarter and more passionate than I am. Because they care more than I do. And they have more passion and they have more drive than I do. So when you study for this test, care. Genuinely put your head down and care like you never have before. Because you can. And you have the time to do so. And don't tell me that you don't. Because I'm a second year medical student sitting there taking 6 hours out of my week to tell you to study for an exam that you're doing. So don't point fingers at me and say you don't have time. Because if I do, then you do too. So let's keep going.
Woo! Motivation right there.
What is cytosol? And how is it different than cytoplasm?
Again, what you
But I believe that cytoplasm is like the whole thing, I think, right? And cytosol is fluid, right? Whatever fluid, whatever, blah, 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 like albumin, different proteins and vitamins and little ions and whatever shit is floating around inside your cell, that's holding up all your cells, suspending them inside the cell. That is what we're talking about right now. What's in there? What floats inside of there? What is sort of this mishmash of stuff that holds ourselves together, right? But the biggest thing, and it's probably the most important thing that's holding, is what's something known as the nucleus. And this is cell HQ, right? This is cell HQ, right?
Yes, sir.
So in the text, they say control center of the cell, right? And it's surrounded by null membrane. And whenever something's surrounded by null membrane, you bet your ass it's important, right? What exists inside of that membrane? Right? So let's say we draw a little thing right here.
A couple things, a lot of things. Well, a couple things. Not a lot of things, just a couple things. All right, hold on, guys. Let me just put this away real quick. I'll be right back. don't think he has. Does he have? I don't think he has. Does he have it? Maybe in homeostasis, potentially. I hope so.
Right? And we have all of this DNA. And whether I draw a bunch of stuff like that, well, that's how it's compacted and stuff like that. We'll talk about why it's compacted that way, how it's compacted that way, blah, blah, blah. Right? By the way, heterochromatin or euchromatin? People who take
the. Ooh. It's probably going to be hetero.
Exam. Hetero? Yeah? Are you sure about that one? Yeah. So we'll talk about heterochromatin.
Ooh, he tried to get us. That was a good one, though. I got a couple more vitamins. That's like $40 total, which is kind of a lot. But anyways, I just want to try them out. These are the ones not produced by the body. We got vitamin E, vitamin B1. Folic acid, we should. The code's done.
And we'll talk about euchromatin. We'll talk about why DNA is wrapped that way, right? And we'll talk about histones, right? And I'm being a little less over in a little bit. But we got this double envelope right here. What exists in the envelope in order to allow the smallest bit of communication with the outside world? Pores. There exist nuclear pores. And this is how things get in and outside of the nucleus. My friend who wanted to lay down a bit of sinus infection, I'm sorry if I'm congested. There's the nuclear pores in order for things to get in and out of the nucleus, right? And the pores, cool, makes sense, right? The DNA is sort of, compressed into this sort of tightly bound structure around what? So we have obviously linear DNA, and of course double-stranded, right? I'm not going to take my time to draw for double-stranded. That's always not right. With double-stranded, it comes linear, and we wrap it around something. What do we wrap it around? This gets wrapped around histone proteins. Have you guys ever heard the phrase that DNA is negative? Yeah? Yeah? Like anyone? DNA is negative? What's negative about DNA? Is it just like sad all the time? No. Where? I can't see it. It doesn't exist. Yeah. I'm just pointing like, all right. So DNA, and we'll get more into this when we get to biochem. I can draw it out for you. But DNA has a bunch of these bases. Right? Out of those bases, on the edge, we have these phosphate groups. And phosphate group I'll always draw as PO4 2 minus. Right? And that doesn't seem like it makes sense. Because normally, when you think of phosphates inside general chemistry, you're thinking PO4 3 minus. Right? Because that's just the normal charge that phosphate has. When we talk about phosphates, we're talking about this. In general chemistry. So if you're talking about like sodium phosphate, it's 3 sodiums connected to that. Why is it 2 minus in the body and 3 minus in chemistry? Well, the body's held at a certain pH, right? And at that pH, one of these sites is protein. So it's HPO4 2 minus. But there's no hydrogen attached to it. The carbon backbone of DNA is attached to the phosphate. So it goes like this. And that R group is the DNA. Does that make sense? So when that R group attaches to the phosphate, you can see that the terminal end of the terminal of the molecule, right, is negative. There are negative charges that are, very scary word, resonated here, right? There's a resonance that occurs. If you guys don't know what resonance is, we'll get into it when we talk about orgo, right? But there is a sort of sharing of this negative charge that occurs over the terminal of the molecule. And that creates a negative terminal. How do you get a negative thing to attach to something? Bring a positive thing here, right? So if I take a negative terminal of a magnet and a positive terminal of a magnet, and I throw them in the air, they're going to snap together and come back down. And it's really quite hard to blow them apart into strong magnets. Think of histone proteins, like the strongest fucking magnet you've ever seen in your entire goddamn life. It's a little ball, right? And they're made up of little subunits that you don't need to know, even though the book says you have to. You'll never need to know them. I don't even know them in medical school, right? But it's called histone. Why is it called histone? Elena? Why is it called histone? Anyone know why it's called histone? The way you're going to remember this, I actually don't remember the specific amino acid, right? I think it's histidine, but it could also be lysine. That the histone protein is made up of amino acids that have what type of R groups? Positively charged R groups. Positively charged R groups. If this is all a bunch of gibberish to you, write it down and we'll talk about it when we get to biotech. That histone proteins are constructed of amino acids, AAs. We're not using capital A's, we're not alcoholics, right? Not alcoholics anonymous, that have positively charged R groups, right? So if they have positively charged R groups and the histones are made of those amino acids, the histone proteins are positive. And if the histone proteins are positive, and they're these little balls like this, and the DNA is linear, sections of the DNA can use their backbone to coil around and attach to the histone proteins. And that's how we wrap them around those histone proteins and get it completely tightly packed. Does that make sense? Is anyone not following? Yeah. Yeah, so what happens is that the histone proteins have positive amino acids, right? Either histone or lysine, I'm not 100% sure which one. And you'll hear me say that all the time, because I don't know everything, right? You guys can definitely look it up for me, that'd be great, right? Because you have to, I know, right? So the histones have amino acids, which R groups are positive. So let's just talk about amino acids for a quick second. That's the backbone of the amino acid, and this is the R group. That R group we're saying is positive. Positive. And if that positive R group.
Okay, let me put the clothes on dry real quick. I'm excited. This guy's dropping some knowledge. Drop the knowledge, teach. Drop the knowledge, teach. Yep.
Put near the phosphate terminal of the DNA, it can wrap around the histone protein. The whole histone protein becomes positive, right? Okay? So when you break a histone protein, you bring a little ball, and I always draw histones like this because it shows that it's made up of parts, but what else is at the positive side? So when a histone ball is brought near the DNA, which has these negative groups on it, the DNA wraps around the histone protein and gets magnetically and electromagnetically attracted to it. Okay? All right. We're going to fast forward a little bit. I do this all the time. I call it overexposure. Or I don't call it that. Professor Lindsay calls it that, right? But I'm going to do exactly what I'm going to overexpose you to something not covered in the chapter. So that's how we make what? Heterochromatin. What does hetero mean? Hetero means two, right? Two different things. Heterochromatin is when DNA and histone proteins come together and produce compact DNA. Heterochromatin is the compact form of DNA. Does that make sense? Thanks for writing that down. Heterochromatin is the compact form of DNA. Don't you dare judge me. Euchromatin would then be the loose form of DNA, right? So write that down. Euchromatin is the loose or relaxed form of DNA.
Yeah.
So now we know how to go from regular linear DNA to heterochromatin. How do we go from heterochromatin to euchromatin? How do we undo that attraction? Well, there has to be a way for what? There has to be a way for us to separate those charges, right? Because if we take the positive charge and make it neutral, or even better, make it negative, then they'll repel, correct? That attraction will be broken. Let me introduce you to a type of reaction. Can I erase this? Let me introduce you to a type of reaction known as an acetylation. By the way, can someone who has a phone or a laptop look up for me what amino acid makes up histone proteins?
It's arginine and lysine.
Arginine and lysine. Cool. Those are both positively charged, although it's not histidine, which would be really helpful. This is like histone protein. Okay, so arginine and lysine, right? Okay, so now let's talk about a reaction called histone acetylation. What's an acetyl group? That's an acetyl group. That thing right there. That is an acetyl group. For people who haven't taken organic chemistry, you can also write it down like this. That's an acetyl group, right? So when we talk about histone acetylation, what do you think we're doing? We're taking this group, and the R is the histone, right? We're taking the histone, we're attaching a acetyl group to it, right? Good? Yeah. So what we're doing is we're taking those positive amino acids, right, of the histone, going positive with this, and instead of taking the positive, which normally is done like this, right? So let's say this is the rest of the histone, this is lysine, because lysine stretched out like this. That's the end of lysine, that's the positive part of lysine. Now instead of NH3, what we do is we take off one of those terminals and we attach this acetyl group to it. And what have we effectively done? We've neutralized the charge. We have destroyed the potential of this to attract a negative molecule. But what was this a part of? It was part of the histone protein. So when we take the histone and we acetylate it over and over again at all of the different arginine and lysine terminals, we are effectively making the histone neutral. We have zeroed out the charge. And since it's no longer positive, it can no longer attract DNA. Because DNA is negative, and neutral things don't attract negative things. They attract nothing. Neutral things are just neutral, right? And we're going to talk about all of these intermolecular forces when we get to organic chemistry and biochem. So this is a little overexposure, but the foundational understanding comes from understanding the fact that, and if you understand this, then good. If you don't, it's fine. The fact that London dispersion forces and dipole-induced dipole forces are weaker than ionic attractions, right? Dipole-induced dipole is what's going on right here, the negative versus the acetyl group, or the dipole-dipole, right? Because we can talk about the induced dipole on the CH3 terminal, or we can talk about the polar moment of the carbonyl group right here, right? Cool? So that's like dipole-induced dipole, whatever it is. But those are always going to be weaker than ionic attractions, which basically just snap two things together. Does that make sense? So now that we've weakened that bond, the DNA is allowed to go free. And this is exactly what we do in order to transcribe DNA. We loosen up a piece of the DNA that we need, it gets red, and it snaps back on the histones after the histones are deacylated. Deacylated, when the acetylation is undone. Does that make sense? Does everyone understand that? So now you know, because the textbook's not going to tell you, but I am, you know how DNA gets unwound for replication. Because in the back of your mind, you're just like, oh, well, DNA replicates before mitosis, which we're going to talk about later in the chapter. How? How does it happen? Now you know, because I just told you, right? Cool. So what were we talking about before? Does anyone remember? Because I almost know, but I do. I'm going to do it. Do this, it has pores, and all that DNA, hetero or euchromatin? Hetero, right? Hetero. Cool. Right? Now, the nucleolus is this like little dark spot on the nucleus with RNAs inside the wall. That's just a fascinating proof textbook. Every time it comes up, we'll look at it, going through what is this? What happens here? All over that. All right, so we talked about the nucleus. And we talked about how DNA gets unwound, we talked about how DNA gets bound, we talked about like acetylation, deceleration reactions, we talked about histone proteins, really proteins, or DNA winding, blah, blah, blah. A lot of information, right? We're done. Done with that. Okay. What's next? What's the next most important organelle? The mitochondria. The mitochondria is the powerhouse of the cell. Oh my god, it's so cool. How are fucking so bad? Right? Mitochondria. The mitochondria is where almost all of the effective energy in the cell is produced. Here's a quick fact for you. Your body produces tons of ATP every day, right? You have to. Right? It's why you eat, it's why you drink, why you do this in order to survive, right? Let's say I ran a marathon. You would guess that the majority of the ATP produced in my body at that time was used for muscle contraction, correct? Untrue. Where's the majority of ATP in the body used? That's 10% of glucose is the brain. I know where you're going with that one, but this fact blew my mind when I thought about it. Have you guys heard of sodium-potassium pumps? Greater than 50% of your ATP that your body will ever produce is used by those pumps in order to maintain membrane potentials. That's how important cellular potentials are, to communicate from cell to cell, and neuronal pathways, and exocytosis and endocytosis, and regulating the amount of fluid and ions that are inside the cell. That's how important it is. That more than 50% of the ATP you'll ever produce is used for those. So that's a cool fact, right? Your body produces tons of ATP every day, right? You have to, right? It's why we eat, it's why we drink, like we do this in order to survive, right? Let's say I ran a marathon. You would guess that the majority of the ATP produced in my body at that time was used for muscle contraction, correct? Untrue. Where's the majority of ATP in the body used? That's 10% of glucose is the brain. I know where you're going with that one, but this fact blew my mind when I thought about it. Have you guys heard of sodium-potassium pumps? Greater than 50% of your ATP that your body will ever produce is used by those pumps in order to maintain membrane potentials. That's how important cellular potentials are, to communicate from cell to cell, and neuronal pathways, and exocytosis and endocytosis, and regulating the amount of fluid and ions that are inside the cell. That's how important it is. That more than 50% of the ATP you'll ever produce is used for those. So that's a cool fact, right? Not relevant to the MCAT, but a cool fact, nonetheless, right? This is a mitochondria. And it has an outer membrane, and then it also has a inner membrane. No, another outer membrane. No, inner membrane. What are these folds? What are they called? Cristae. You guys see how I color coded that? Isn't that so cool? I'm actually colorblind, so you're welcome. Oh, where did I get colorblindness from, by the way? Who did I get it from? No, my mom. Why? It's X-linked. No one else could have given it to me because I only have one X chromosome. Thanks, Mom. Where'd she get it from? My mom's not colorblind. Where'd she get the gene from? Well, it depends. She got it from her parents. She's a woman, right? She got it from her dad. Right? So my grandfather's colorblind, and I'm colorblind. Right? Cristae. What did Cristae do? Cristae increase the surface area for energy production. Have any of you guys ever heard the coastline paradox? So the coastline paradox is basically like this idea that if you have a country, right? Pretty fucked up looking country, right? But if you measured the coastline using like 10,000 kilometer meter sticks, then you get a very, very short length. But if you shorten it to like 5,000, you'd be able to get into the grooves, and then it'd get longer. And then if you shorten it to like a centimeter, it'd get even longer. So like all coastlines are technically infinitely long, right? Because you can just like fit more shit the smaller and smaller you go, right? So since there are these little grooves over here, I want you guys to think that in every single nook and cranny of those, we're fitting more proteins for the production of energy. Right? When you guys did orgo labs, what did you do to all of your reagents that were solid? You crushed them up. You crushed them into a fine powder, and then you add them into the flask. Why? Surface area. There's more points of contact in order for the effective reaction to happen. What's the effective reaction of the mitochondria? Cellular respiration. To produce energy, correct? So since you have more folds, you have more ability to perform that reaction, right? What's the inside called? The matrix. Mr. Anderson, right? What's the outside called? This part. The intermembrane space, the word I couldn't remember before for some reason, even though it's just the space between the membranes. The intermembrane space, right? What happens on the inside that we're so worried about, the inside membrane? It's the house of the electron transport chain. The ETC happens along the inner membrane. So the Cristae function to increase the efficacy and the velocity and the overall happening of the ETC, the electron transport chain, which is where we derive the majority of our energy. What time is it, by the way? 7:01. It's been an hour. Okay, cool. So we'll probably just do like one chapter today, and then we'll take a look at chapter 2, save the rest for next time. You guys can feel free to leave whenever you like, by the way. Okay? So that increases the efficacy and the amount of like stuff that you have in order to undergo the electron transport chain, right? So the intermembrane space in the matrix allow for a separation of several components of the electron transport chain that will undoubtedly help us down the line, right? You guys know about like proton motive forces and the old protein model. That's why we have the separation, right? Okay. There's one more really small fact that I always forget to mention about the mitochondria. Mitochondria, and the MCAT wants you to know this, which is why I'm writing it down. It has the ability to kickstart. Kickstart what? Not you. Kickstart what? I say it. Apoptosis. What is apoptosis? Not you. Program and control cell death. Control and program cell death. This isn't the cell exploding. It's the cell taking cyanide and drinking it, right? It's basically just like, guys, something's wrong here. I'm gonna go kill myself before you die, right? That's basically what the cell is saying. What's necrosis? Well, necrosis can happen to a single cell too, right? So it's like, it's just uncontrolled cell death, right? So necrosis is apoptosis, right? And if you guys like hop on the whole Anki wave when you're studying for the MCAT, which I will teach you how to do eventually, right? Whenever we get a bit more efficacious in our sort of lecturing styles, I'll probably put it up on the screen. You're gonna see a bunch of cards about like necrosis versus apoptosis, necrosis versus apoptosis. Is it super important? Not really. To know the differences between the two. That's more of a medical school thing. Because the reason it's important to medicine is because when you look at a slide of cell under a microscope, you want to be able to tell, is that necrosis or is that apoptosis? And that can tell you a little bit about the disease that's happening inside the person that you got the cell culture from. Does that make sense? Or the tissue sample, right? That's how we got to know about the mitochondria. All right. So one more thing. Remember how I said that your mitochondria are inherited from your mother, right? That is called extranuclear inheritance. That's where inheritance does not
Hold up. All right, play.
happen from the nucleus, right? Extranuclear inheritance. Because all of the other DNA information comes from the nucleus, right? That's where it all starts from. Okay. There's a couple other like small organelles they want you to know about. So lysosomes, right? Let's like break that down a little bit. So we're talking about like the smaller organelles because honestly, the majority of the questions about organelles on the MCAT are going to be about either the mitochondria or the nucleus. But every now and again, you might get like a little nugget of information about another organelle. So let's talk about the first one, the lysosome. Why is that such an easy one to figure out? Why is that so easy to figure out? Because it has the word lysis in it, or at least a derivative of it. So that tells you that this organelle has a bunch of hydrolytic enzymes that can break down material. For some reason, whenever I was studying, I confuse the lysosomes and vesicles a lot. What are vesicles? They're like little cardboard boxes, basically. That the cell's just like, I'm gonna put this in here and pass it off to someone, and you're gonna walk with it and drop it off somewhere else. That's like what a vesicle is. Right? What's a lysosome? What does lysis mean again? Lysis means to break. Right? To break. All right. So let's talk quickly about the smooth and rough endoplasmic reticulum. And the way I wrote it in my notes is probably the way it was in the textbook, right? Let's talk about the RER, which is the rough endoplasmic reticulum, and the SER, which is the smooth endoplasmic reticulum, right? What's the first fact about the rough ER? What's so rough about it? You get shot and you go there? No. No, what is it? You said it. Ribosome. It is quote unquote studded. Bro, it's dripping with ribosomes. So that means it can do what? Protein synthesis. Another one of my shorthands. This double arrow means leads to. This single arrow is an elaboration, meaning that I'm explaining what was said before. Okay? Double arrow means lead to. So studded ribosomes leads to protein synthesis, right? What's so special about protein synthesis? Huh? You need proteins to survive. Proteins are everywhere. They do everything. Enzymes, they're proteins. Transport, they're proteins. Channels, they're proteins. Pores and membranes, they consist of like fats and proteins, right? Proteins are everywhere in your muscles, in like the space between neurons and everything. Proteins everywhere, even inside your blood. Gold star is the person who can tell me the protein in your blood that helps to carry other molecules around.
Hemoglobin?
No, that's oxygen, but good guess. Albumin. Okay, I do this thing with my students over at downstate because I teach a lecture to first year students as well every Friday. So you guys do Tuesday, Thursday, I'm a demo Friday, right? So what I do, and I forgot to bring that, which sucks. That's the other tragic thing that happened today. I forgot my bag of Johnny Ranchers. Every single time someone gets
Does it carry? Oh, okay.
The question, right, that I say is difficult, I throw them a valid answer. I just like whale it at them, right? So next time, I owe you, and who answered the other question? You, right? Someone answered another difficult question before, not you. And I owe you a valid answer. Right. So, center rhyme zones and talk about algorithm, right? So algorithm is most likely referred to. So like when you talk about where these formulas are probably synthesized, it's probably the argument that's where all the rhyme zones are, right? Okay. What about smoothie are? Two things. That's the only really I know about smoothie. But smoothie are two things. Number one is that it lacks ribosomes, which is why it's smooth. There's like under number one is three things: synthesis of lipids and detox drugs. I don't know why. They don't tell you this. But the MCAT also wants you to know one thing. The way we detox drugs is by making them more hydrophilic. That's called first pass metabolism. When you eat something, right? Have you ever heard that whenever you eat something, it goes to the liver first? That's called first pass metabolism. And when things get metabolized inside the liver, like on first pass or on second pass or whatever, whenever you eat hydrophilic. The way we detox drugs is by making them more hydrophilic. That's called first pass metabolism. When you eat something, right? Have you ever heard that whenever you eat something, it goes to the liver first? That's called first pass metabolism. And when things get metabolized inside the liver, like on first pass or on second pass or whatever, what the liver's doing is it's modifying that molecule to make it more hydrophilic. Like that's how we detox things, right? So it's adding on molecules to make it hydrophilic. Why are we doing that? Think critically. Think very, very critically about why we're taking something that we want to leave the body, big hint, and making it more hydrophilic.
Because we wanted to leave. Making them more hydrophilic. We wanted to go into the cells, into other tissues.
Sorry? Dissolved in what? In water, and what does water make up a lot of? The blood, right? And the blood turns into urine, and the urine gets excreted from the body. So when we make something more hydrophilic, we allow it to be dissolved in the blood.
Oh, hydrophilic. I thought he meant hydrophobic. What? Doesn't make sense if it's hydrophobic. It makes sense if it's hydrophilic.
Blood travels
gotcha.
Dissolved in what?
Wow.
In water, and what does water make up a lot of? The blood, right? And the blood turns into urine. Whenever you eat something, it goes to the liver first. That's called first pass metabolism. And when things get metabolized inside the liver, like on first pass or on second pass or whatever, what the liver's doing is it's modifying that molecule to make it more hydrophilic. Like that's how we detox things, right? So it's adding on molecules to make it hydrophilic. Why are we doing that? Think critically. Think very, very critically about why we're taking something that we want to leave the body, big hint, and making it more hydrophilic. Sorry? Dissolved in what? In water, and what does water make up a lot of? The blood, right? And the blood turns into urine, and the urine gets excreted from the body. So when we make something more hydrophilic, we allow it to be dissolved in the blood. The blood travels to the kidneys filter it out, put it in the bladder, and you allow it to be dissolved in the blood. The blood travels to the kidneys filter it out, put it in the bladder, and you piss it out. That's how we get rid of toxins, right? So I'm gonna ask you a question. There's a dead person in front of you, right? On this table, there's a cadaver. And I go into the cadaver, and I slice off a really thin slice of muscle tissue, and I go to the liver, and I slice off a little bit of the liver, and I put the two things into a microscope. I want you to tell me, between the muscle sample and the liver sample, which one of these is higher in each one?
The muscle and the liver. That's a good question.
So between the rough ER and the smooth ER, what's higher in each one? Because of exactly what we talked about, right? That the detoxification processes that happen within the liver happen within the smooth endoplasmic reticulum. So when you see an electron micrograph of a hepatocyte, right? And it,
okay. That makes sense.
So between the rough ER and the smooth ER, what's higher in each one? Because of exactly what we talked about, right? That the detoxification processes that happen within the liver happen within the smooth endoplasmic reticulum. So when you see an electron micrograph of a hepatocyte, right? And if those are weird words, hepatocyte just means liver cell, right? An electron micrograph is a very, very, very small microscope that shows you an image of a single cell, usually, right? So when you take an electron micrograph of a hepatocyte, you're able to see a lot of smooth ER. But in the muscle, where you need to produce proteins, and a lot of them, you're gonna see a lot of rough ER. Does that make sense? The second thing is the smooth ER also helps to transport proteins from where to where? From the rough ER to the, what's next? The Golgi. From the rough ER to the, what's next? The Golgi. The Golgi. Also, shorthand leads to explanation. Single arrow is explanation, double arrow is lead to. That's why I erased that arrow over it. Okay. So, obviously, we just talked about the nucleus, talked about the mitochondria, talked about lysosomes for some reason, just like thrown in there. They talked about the rough and smooth ER. What are we going to talk about next? The Golgi, right? That makes total sense. And that's exactly where we go next, right? Okay. So, do you see how the textbook is painting a picture for you, right? We have the nucleus, which allows all this to happen. And we have the mitochondria, which work together in order to give you the energy and the instructions on how to produce life. After we transcribe into the nucleus, the mRNA from transcription, right? Or transcription, yes, goes into the rough ER in order to produce proteins. Those proteins go into the smooth ER where they're transported over to the Golgi apparatus. It's creating a timeline for you, and they don't even say it. And that's why I'm going to tell you. Because if they don't tell you, you'll never know. And who told me? No one. I had to figure it out my damn self. Actually, Professor Lindsay. And he's the greatest professor I've ever had in my entire life. You can't use it. Not at all. I paid him a lot. All right, Golgi apparatus. Let's just like read off my notes here because I'm sure you guys know what it kind of looks like. That weird blobby thing, like a bunch of like, it kind of looks like this, it's like that, and it like passes through each thing, and all of those are like little membranes, and when it passes through them, everything gets put into like a little vesicle. Those vesicles transport through the cell, and that's how you get these from one place to another. Okay, I didn't see you talking, but Elena was kind enough to bring you guys snacks, so you guys pass me around, and I really don't care if you eat my class. I don't even care if policy, but pass me around, I don't want them. Right, and you guys figure that out. And we have napkins. Okay, so I'm just gonna keep talking, right? So. All right, do you guys remember when you're like creating proteins, you have a primary structure, and a secondary structure, and a tertiary structure, and a quaternary structure, right? So the primary structure is what? The chain of amino acids. And if you don't know what an amino acid is, it's a basic building block of a protein, right? It's coded for inside your DNA, and amino acids come together in bond, these things called amide bonds, right? And then it's chain together. The primary structure is just a straight chain. Inside of those amino acids, there are these groups that interact with one another through things called hydrogen bonds. And the interaction of those hydrogen bonds creates the secondary structure, where we fold into different components, which we're going to talk about when we get to biochem, right? They fold into alpha helices and beta helice sheets. You guys have heard this before, right? Just give me a nod. Yes? Alpha helices? No? Never heard alpha helices and beta helice sheets? No, we'll talk about it when we get to biochem. Basically, it's the conformation that proteins like to be in. So they either twist around one another, which is known as alpha helix, or they fold into these little sheets of diagonal amino acids called beta helix sheets. And all that does is it reduces the actual, like, repulsive forces between the molecules inside the protein. Does that make sense? All right, cool. You guys need to move this, right? Something like that, right? Okay, so then we have tertiary structure. This is where the protein starts to like fold in on itself, and it gets modified, right? We're talking about modification. Well, it might get phosphorylated, or it might get hydroxylated, or it might be a component put onto it, or whatever. That modification happens in the Golgi. Protein modding. Anyone play video games here? Like download hacks on the protein. Like you're making it more efficient at what it does, right? You're just hacking it. That's it, right? And quaternary structure is very, very special, and it's only like talked about the select number of proteins that you would need to know about, and it's where multiple tertiary structures come together and they form one big protein complex. Anyone know an example of when that happens? What protein in your body is a quaternary structure? Hemoglobin. Yeah, hemoglobin, right? It's how many structures? It's 4, right? It's like 4 different terminals, 2 of each type. And I'm not gonna get into like the different things, but like some hemoglobin is alpha 2 beta 2, some hemoglobin is alpha 2 gamma 2, and then some people have like a disorder where they're not able to make the proper type of like globin. That's what's called like a globin, like an alpha globin or beta globin. They can't make the proper one, so their blood just doesn't work, which is unfortunate. Okay. Okay. So you guys can see, like, you can't see my notes, but you guys can see what's so important, right? This is basically what I'm talking about. So we have the nucleus, no membrane. The fuck up with nucleus, right? We have the nucleus, no membrane, all DNA right here. And you guys are trying to get sent out. One gets sent out, we have mRNA coming out. And then over here we have the rough endoplasmic reticulum, which is continuous with the smooth endoplasmic reticulum, which is continuous with the Golgi, which is continuous with the outside of the cell. Right? So when we produce neurotransmitters and hormones and albumin and proteins that need to either go outside the cell or into the membrane or into the cell, this is where they go down. Does that make sense? Nucleus to rough to smooth. What's so rough about that? Okay, ribosomes. Nucleus to rough to smooth to Golgi. And then the Golgi is like that membranous thing. And when you plug off those membranes, they turn into vesicles, and those vesicles carry the material that you just synthesized and either bring it back inside the cell or put it on the membrane or bring it out. Right? I make this joke all the time with my friends, and they're just like, wow, we were just like so depressed. Okay, that's how I'm gonna tell you guys this joke, right? So like 3 ways out of medical school. What's number 1? What's the number 1 way out of medical school? You graduate. No, you don't know that. That's number 1. That is not number 1. Number 1 is you graduate, you become a doctor. What's number 2? You drop out, right? So either you graduate or you quit, but there's a third one. You could die, right? So there's 3 ways out of medical school. You drop out, you quit, or you die, because they're not kicking you out. Because every year they can keep you is another $70,000 you're giving them. They're not kicking you out, right? So there's only 3 ways for a protein to go: into the cell, onto the cell, or out of the cell. Which seems straightforward, right? Into the cell, onto the cell, out of the cell. So 3 ways. And for some reason, right, if you guys believe in if you guys believe in God, or if you believe in the universe, or if you believe in like crystals and energy and all that stuff, for some reason, everything in like the body can really easily be like turned into 3s, right? But I think that's more so because of the human psychological tendency to think of things in 3s than it is any sort of coincidence. Because honestly, there's probably 1,000 different ways to do things in the body. But when we talk about enzymatic destruction of neurotransmitters and hormones, and we talk about synapses in the nervous system, there's all the 3 ways out of synapse. You get taken back up, you get destroyed, or you leave, right? And it makes it here. You either get taken back up, you get pushed out, and then later destroyed, or you leave, and you just enter the memory, right? Break things down to really simple terms. This is something to all of my students, right? I'm talking at a level that a high schooler could come in and understand, right? Right, I'm going very slow, but raise your hand if I'm taught you something that you didn't know before you came in this room. That's how we do it. You don't just start up here and just keep fucking pushing. You don't like train for absolutely no time at all and run a marathon. You crawl and you whine and you complain and you trudge your feet and you slowly limp your way up until you can walk and you can hobble and you can run and you can jump and then you go. We are fucking crawling with both of our legs cut off right now. And that's just how it's gonna be. This first lecture, I've taught the MCAT curriculum how many times? Maybe 5 or 6 different people, right? This is usually how long the first lecture takes. It's the longest one, and every single lecture after this gets faster. I can teach 2 chapters of biochemistry in 45 minutes. If you just stick along for cells, it takes 2 hours, right? Because when we get to biochemistry, what are we gonna talk about? Histones and protein folding and hydrophobic interactions and Golgi apparatus and protein synthesis and phosphate backbones, all of which we just talked about, right? So when you encounter it a second time, and a third time, and a 4th time, you'll be begging me to stop, right? Because you're gonna know each other, hopefully, okay? Okay, this is a cool little other, what's the word now? How many of you have had infection before? I'm talking to the women. When you guys get your ears infected, after you get a piercing, or the men to go ear piercing, right? The women work on getting ear piercing, and how many of you guys have an infected ear? Yes, yes, yes. My sister, every piercing she gets, infected completely. Just, I don't know what the hell's wrong with it, right? Not saline spray to give you, but spray on your ear whenever it's infected. And sometimes they're like, if it turns like a certain color, put hydrogen peroxide on it, right? Hydrogen peroxide. Okay, so what's the next organelle we'll talk about? A peroxisome. Someone in the textbook. A peroxisome is filled with hydrogen peroxide in order to break down material. In medical school, we go wild over peroxide. We go crazy because like all of your immune cells, they use hydrogen peroxide in order to just like fuck the shit out of bacteria. Also, I have the filthiest mouth known to man, right? But it tends to help people remember things. So excuse my language, but hopefully you'll thank me for it in the future, right? So what is hydrogen peroxide? It kind of looks like this, right? And what happened? Yeah.
Why did it go?
Right there. Got it. All right. It's like very difficult. This is huge to me. It's probably very small to you. Okay. Go right here. So this is hydrogen peroxide, right? And what happens? It's filled with hydrogen peroxide, H2O2, in order to break down material. So this is similar to a lysosome, but it doesn't use enzymes. It uses hydrogen peroxide. We'll talk about how that happens. When this hydrogen peroxide is primed to do something, what happens is you get a dissociative reaction. This is called a heterolytic bond dissociation, right? So it goes 2 ways, heterolytic, right? And what you get is 2 of these hydroxide radicals. And those, the most information you need to know about peroxisome, those can just rip things apart. Because that radical, that little electron, it's really, really hungry for a partner. So what it does is just starts nabbing electrons off of a bunch of other things. And then those radicals that work paired together, and then neutralizes the toxic capacity of whatever compound you're written down. Does that make sense? So in peroxisome, we use hydrogen peroxide to produce radicals, which are known as something that you're going to read about in a lot of articles when you start answering questions about that. They're known as reactive oxygen species, ROS. Reactive oxygen species. If you guys know Professor Alexander Greer at Brooklyn College, most of his research is about reactive oxygen species. Very interesting stuff. Does that make sense? We're almost done with organelles. We are going to talk about the cytoskeleton in transit. Because what's going to happen is like, you're going to get asked so many questions about it later down the line. You're just going to learn from that. Because me lecturing to you, and I've noticed that like me lecturing to you guys, and you're trying to learn it for myself, just reading it and writing it, it doesn't work. Because it's just the same thing over and over again. So I'd rather you get down the line, get to the questions and understand them, but definitely read the textbook and like, try to understand what's in it. Okay, but one thing we're talking about is the fact that microfilaments, this is the biggest thing. Microfilaments, which are a component of the cytoskeleton. These are made of actin. Actin, that's the protein that makes them up, right? Okay? And this plays a role in cytokinesis. Cytokinesis being separation of the cells when we do mitosis or meiosis, all right? And this next one is microtubules. These are easy to remember because they're made of tubulin. Tubules made of tubulin, right? Microtubules made of tubulin, and microfilament is made of actin, right? Okay. One thing about microtubules is that they're involved in the synthesis of cilia and flagella. Where can we find cilia in the body, and where can we find flagella in the body?
Cilia in the lungs, flagella in the sperm.
Cilia where? Yeah, sperm cells, right? Sperm cells have flagella, that's how they move around. And actually, there's a diagram inside of the reproduction chapter, which you guys should open up. I'm talking about the Kaplan books, by the way, right? Those are the books I use. Those are the books everyone should use, in my opinion. If you look at the reproduction chapter of the Kaplan books, there's a diagram of the actual motor function of the sperm, and there's this very cool motor looking thing inside. It literally looks like an engine. It's super cool. At the end of that is flagella, which allows them to move around. Cilia where? Respiratory tract, right? And they help to kick up dust and other pathogens. They're these tiny, tiny little proteins that kick things up, and then you either expire them out, or you sneeze it out, or you cough it out, or whatever. Or something that's really disgusting that we learned in medical school is that the cilia move this stuff up, and you just swallow it, and then it gets destroyed by your stomach acid. So you're swallowing dust and dirt and all that stuff all the time. Okay, so that's a little bit about cytoskeleton, right? Centrioles, right? Those are involved in mitotic division. So those are these are all the lesser sort of known and talked about organelles. Centrioles are involved in mitotic division, right? And the microtubules kind of connect to this centrioles, and then it pulls things apart, right? And for the centrioles. And then there's just other things that you don't really need to know about all too much. Intermediate filaments, you're just going to hear the names of them all the time. Those keratin, dinesin, vitamin, all that stuff. And then we get into a sort of part of the chapter that I didn't think was super relevant. It's tissue formation, right? Tissue formation. So what are tissues? So we talk about cells, right? And we just explained a whole ton of a lot of how cells work. What are tissues? They're just organized sheets of cells, right? They're tissues. Organized sheets of cells. Right? Now there's 4 tissue types. There's epithelial tissues, connective tissues, muscle tissue, and neurons, nervous, or nervous tissue, right? More specifically. Another thing they don't tell you, these are somewhat organized in restorative or regenerative capacity. Except muscle tissues, in what I've learned, have a little bit more capacity than connective. So, another thing they don't tell you, these are.
Hi, Anjali.
Are somewhat organized in restorative or regenerative capacity. Except muscle tissues, in what I've learned, have a little bit more capacity.
I'm not sure what was said.
Muscle tissue and neurons, nervous, or nervous tissue, right? More specifically. Another thing they don't tell you, these are somewhat organized in restorative or regenerative capacity. Except muscle tissues, in what I've learned, have a little bit more capacity than connective. So epithelial tissues are pretty good at regenerating, then muscle, connective, and neurons. So their neurons are quite bad at regenerating. Connective tissue is quite slow. Muscles regenerate slowly, but faster than others, and epithelial tissues regenerate quite fast. Okay. What are epithelial tissues? You hear this word going around, the epithelium, right? In the textbook, they say it covers the body and lines its organs and does this and forms the parenchyma and blah blah. They're functional. They're the functional tissues of the body. They perform function, right? So it says epithelial cells usually make up the parenchyma, the functional unit of the organ, right? So when we talk about liver epithelium or lung epithelium or ovary epithelium, right? It's the stuff that does the work, right? Does that make sense? Cool. Have you guys heard of the layer thing where it's simple, stratified, pseudo-stratified, blah, blah, blah? Yes? No? Any no's? Kind of, right? So stratified, or sorry, simple is just one layer. So it's just this. That's one layer. Stratified is multiple layers. And pseudo-stratified is the little cup who's sitting there like this. So it looks like it's multiple layers, but it's really not. They overlap over one another. That's pseudo-stratified. So when they say stratified epithelium or simple epithelium, all they're saying is how are the cells arranged, right? So simple is one layer, stratified is more than one layer, pseudo-stratified looks like it's more than one layer, but it actually just wants you to do bad in your exam. And you got shapes: cuboidal, columnar, squamous, blah blah blah, right? The chapter goes into a little explanation of how to classify prokaryotes. I don't find this section of the chapter very important. I don't find it super important to discern between like archaea and bacteria. The only thing there is that like they both exist under under prokaryotes, right? And then archaea live in like really, really harsh environments. That's pretty much the only thing, right? But the one thing they do is they talk about these shapes of bacteria, which come up all goddamn time, right? So we have cocci, bacilli, spirilla. Who can take a wild guess what this one is? Spirals. They go like this. They're the fun ones. Anyone ever read Uzumaki by Junji Ito? You should. It's a fantastic book. Well, it's a manga, but it's about spirals. Cocci. What are they? They're dots, right? They're little circles. Spheres. Because apparently the world is 3D. What about bacilli? They're rods. You guys know that meme of like the metal rod like dropping on the floor and makes a loud noise? Or am I just like stupid? I don't know. All right. Aerobes and anaerobes. All right. What does obligate mean? Is something obligatory on you? Like I'm Muslim, and my prayers they're obligatory, which means I must do them. Right? I have to. Right? So if something is obligate to you, you have to do it. Right? So when we're classifying living things, and we say that something is an obligate aerobe, what do we mean? It needs oxygen to survive.
Yeah.
Is that us? I sure hope so. Because if it's not, get the hell out of my room. What's next? Obligate anaerobes. Obligate anaerobes. They cannot survive in the presence of oxygen.
Nah.
They would die. Why would they die? We talked about it earlier. Who remembers what we talked about? What does ROS stand for? Reactive oxygen species. Something about these organisms, when they come near oxygen, they turn it into reactive oxygen species and they just die. Right? What's next? Facultative anaerobes. Right? What does facultative anaerobe mean? They switch. Oops, can't spell. They switch to anaerobic processes when beneficial. So like our cells are technically facultative anaerobic. Because like if we don't have a lot of oxygen around, they can switch to anaerobic processes. Right? We can like switch to only using glycolysis, kind of turning off the circadian cycle. But we still need oxygen, right? We as humans, as a system, we need oxygen. And it's not that our cells aren't using the oxygen, they're just doing a little more of the anaerobic respiration, right? So like a little bit of this. But there are sort of organisms that can switch to anaerobic, and switch back, and switch back, right? There's no more. This is the easiest one. Aerotolerant anaerobes. So aerotolerant anaerobes. I know it's quite small, but this is AERO, tolerant anaerobes, right? What's an aerotolerant anaerobe? It's anaerobe, right? So what's it doing? It's not using, but it tolerates oxygen. So if my sister's annoying me, right? It's not like I'm gonna kill her if she's in my room, or I would die if she was in my room. I would tolerate her being in my room, right? I don't want her there, and I have no use for her, but I tolerate her being there. So it's an aerotolerant anaerobe. It's not gonna use the oxygen, but it's not gonna die either, right? All right. One important thing about prokaryotes, something we didn't put up on the board before. I don't know why it's not like listed in the what's it called, the textbook related. We have extra napkins out of it. Yeah, I'm gonna use one to erase the board. So, what did we talk about before? Like the differences between prokaryotic and eukaryotic cells. What did we not touch on? Some prokaryotes have a what? A cell wall. What's a cell wall? What's a cell wall? It's a wall. It's a wall that surrounds the cell, which you believe in, right? So prokaryotic cell wall. Two types. Gram positive, gram negative. Definitely need to know this when you have that. Like 100% need to know this when you have that. Gram positive has something known as a thick peptidoglycan layer. Peptidoglycan is just this special, like, modified protein that exists on the cell walls of prokaryotes, right? That's peptidoglycan. And in a cell wall of a gram-positive bacteria, has a very thick peptidoglycan layer, which means gram-negative would have a very thin peptidoglycan layer. a very thin peptide white membrane, right? And one more thing that doesn't really come up with the MCAT, but it might, since they mentioned it, you might read it somewhere. These contain something, gram-negative bacteria, they contain something known as lipopolysaccharide. Lipopolysaccharide, or LPS, right? What is lipopolysaccharide? What's up? Yeah, it's an endotoxin. Have you seen immunology or microbiology before? Yeah, there you go. So basically, lipopolysaccharide was evolved by bacteria to make them more pathogenic. What is pathogenic? Causes disease, right? So pathogenicity is the ability for bacteria to produce disease, correct? So this lipopolysaccharide makes them a lot more dangerous to our bodies. They activate the immune response. And that's why like E. coli is so bad, because it's gram-negative. The lipopolysaccharide, it causes widespread, massive disease inside your body. Does that make sense? Cool. What color is something that is gram-positive? Purple. And then gram-negative is pink. Cool. How are you going to remember that? What's up? Yeah, right? You see it in the lab, you memorize it, you figure it out, right? Oh, here's that little diagram. I guess it's in here. You're talking about flagella. Yes? See that diagram over there? Like if you look at chapter 1 of the flagella thing, that is the tail of the sperm. It's so cool. I'm going to show the camera. So that is your little like motor that exists inside of the sperm cell, and it allows for motion, right? So that's kind of what's going on. And they talk about flagella a little bit. You would never believe that the flagella is composed of a protein called flagellin, right? Scientists are so creative, right? Cool. All right, so the reason we talk about flagellas is because a lot of prokaryotes have flagella, right? Cool, that's how they get around and things like that, right? One more thing. They don't have mitochondria, right? We talked about that before. But one more thing that comes up every now and again on the MCAT is the size of the prokaryotic ribosome. You guys, in a couple weeks, are going to meet my roommate. His name's Steve. And Steve is a great guy because he taught me a little trick about the ribosomes. You go 30, 40, 50, 60, 70, 80. And you go S, 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 S. And this is pro, and this is euc. And you add these together, and you know that the prokaryotic ribosome has a 30S and a 50S subunit that somehow becomes a 70S ribosome. And you know the eukaryotes have a 40S and a 60S and an 80S ribosome.
What the heck?
What do those mean? Those are called Svedberg units, right? Svedberg units. And it's a unit of sedimentation time. So they took these little like subunits in the whole ribosome, and they see how quickly they settle down to the bottom of some sort of mixture, right? And then they took the guy who just tacked his name off the unit, right? Those are Svedberg units, right? Sedimentation time. For some reason, 30 and 50 becomes 70, and the 40 and 60 becomes 80, but that's what it is. So the prokaryotic ribosome is a 30S and a 50S subunit. The eukaryotic is a 40S and a 60S subunit. You will get a question on the MCAT about this. It will happen. 100%. You gotta know that. Sure. And the sad part is I didn't eat Steve before I took the MCAT. I had no way of memorizing that before, and it was the fact I didn't know. I probably got that question wrong. Okay. So we're going to talk about prokaryotic cells, binary fission, right? Binary fission. Basically, the cell decides it wants to become two cells, it replicates the DNA, and just dissociates. That's really it, right? But there needs to be a way for bacteria to kind of diversify the genome, or else it would have died, right? Or else they kind of would have just not made it, right? So we have a lot of ways of genetically recombining our DNA inside of bacteria. And those four things, or three things, I guess, are transformation, conjugation, and transduction. Right? So number one is transformation. Number two is conjugation. And what was number three? Transduction. Right? All right. So transformation. What happens? So basically, a bacteria dies, right? And when it dies, bacteria dies, which leads to release of genetic material, which leads to reuptake by other bacteria. And if that dead bacteria had genetic information that that bacteria didn't have, it now has it, right? So let's say that like there was one gene, one extra gene inside of the bacteria that died, and it gets released out of the environment, and this one bacteria that really needed that gene takes it, now it has it. Done. Dead, uptake, done. Genetic diversity, right? It's like, any of you guys ever watched Naruto? Right? Where like the Uchiha clan member dies, just fucking take his eye and put it in yours, and now you got the Sharingan, right? So that bacteria just got the fucking Sharingan. That's what happened, right? So, dead bacteria, take its DNA. Easy. So I go up to a dead guy and I steal his wallet. Now I got all his money. Very unethical, but now I got his money, right? What about conjugation? It's bacteria sex, right? It's bacteria sex. What's a plasmid? It's a little circle of DNA. So this is plasmid transfer. So a plasmid is a circle of non-nucleoid DNA. Why did I say nucleoid? Because like bacteria don't have nuclei, but they have DNA inside this region called the nucleoid region. Am I right, bacteria guy? Yeah. So that's non-nucleoid DNA. It's aside from that. So this is a little circle of DNA that the bacteria just has somewhere lying around, correct? Now, if the bacteria is F plus, it can initiate conjugation. What does F plus mean? So basically, if the bacteria has a gene that allows it to have the ability to create a plasmid, or sorry, to create a conjugation bridge, it will. So let me explain what I mean by that. Okay. Can I erase that on top? Yeah? Okay. So let's say that we have, let's say that we have two bacteria. What's the shape of those bacteria, by the way? They're bacilli, right? This one's F.
Wait, bacilli. I thought it was carbac. Okay. Oh, okay, sure. Well, whatever.
Once, because it has a plasmid that allows it to form a conjugation bridge, right? And this one is F minus. I think that is fertility, right? So that was fertile and that was not, right? So what happens is that this is the nucleoid region, and let's say that this is the F plus plasmid. That's the plasmid that allows it to make a conjugation bridge, right? It's gonna copy that DNA into another plasmid. And now there's two of them. Do you need two of them? No, right? So that plasmid codes for proteins that allow it to make a bridge. And a bridge is called a pilus, right? So it's gonna grow out of here, and it's gonna grow a little more, and it's gonna grow a little more, and it's gonna grow a little more until it attaches over here, right? And that is called a sex pilus. That's why it's bacteria sex. So it's sex pilus, right? And what happens is that plasmid slips through the sex pilus and into this F minus bacteria. And that leads to the formation of two F plus bacteria. What's the point? What's the point if that's the only thing we're like conferring? Because eventually, at some point, it may happen that a bacteria dies, right? And through the process of transduction, you pick up a gene, you pick up a plasmid that you want to give to someone else. And now that you're F plus, you can replicate that plasmid, throw a conjugation bridge, or throw a conjugation bridge or a sex pilus, and transfer both those genes onto another bacteria. And this is how bacteria confer resistance. You guys ever heard of MRSA and MRSA? Methicillin-resistant Staphylococcus aureus, or vancomycin-resistant Staphylococcus aureus, right? They confer resistance through DNA changes that have occurred evolutionarily, and they give it to other bacteria in their population, and now you have a problem. That you can't treat that staph infection with vancomycin or methicillin, right? That's how that happens. Transduction is the one that everyone likes. Why? Why is transduction the cool one? You're using something called a bacteriophage. What's a phage? A phage? It's a virus, right? Bacteriophage. A phage, a virus for bacteria. Bacteriophage to inject DNA into a specimen. They look like this. Sorry, orgo people, for scaring you with the hexagon. They're like that, and they got all this DNA in here, and they come up to a bacteria, and they go, boop. That's it. And that DNA, what does it do? It integrates into what? Into the native bacterial DNA. The DNA opens, accepts the new DNA, and now what can it do? It can replicate that viral DNA for whatever it wants. Either the synthesis of new viruses or the use for sort of, what's it called? Resistance or blah, 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 whatever it is. And it got that from a virus. Does that make sense? Virus is alive or not? It's moving packets of genetic information. All right? Very good. Okay. Let's talk a little bit about prokaryotic growth. All you want to know is that the graph looks like this. What happened here? They exhausted their resources. What happened here? They're adjusting to the new environment. What happened? What happened here? Looks like this.
For what?
Very good. Okay. Let's talk a little bit about prokaryotic growth. All you want to know
Prokaryotic growth.
is that the graph looks like this. What happened here? They exhausted their resources. What happened here? They're adjusting to the new environment. What happened here? They're replicating out the back door until they reach a phase where the amount of resources they have is equal to their potential, meaning that they're dying at the same rate that they're reproducing. That's called the exponential phase, the lag phase, and the death phase. Oh, sorry, no, that's the lag phase. That's the lag phase. The resources they back here, they are replicating out the back door until they reach a phase where the amount of resources they have is equal to their potential, meaning that they're dying at the same rate that they're reproducing. That's called the exponential phase, the lag phase, and the death phase. Oh, sorry, no, that's the lag phase. That's the lag phase. I think that's called the plateau phase, stationary phase. Lag, exponential, stationary, death. Make sense? Cool. Anyone want to take a break? You guys all right? If you're all right, maybe done. Yeah? All right.
Keep going, buddy.
Okay. We're going to talk about viruses and subviral particles. So like I said before, viruses are not alive, meaning that they're dying at the same rate that they're reproducing. The gene that allows it to have the ability to create a plasmid, to create a conjugation rate, it will. So let me explain what I mean by that. Is breakdown, we're talking about viruses not alive. They're not made of cells, they don't really have organelles, and blah, 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 right? So they have genetic material, a protein coat, and sometimes lipids, right? But basically, what happens is that most viruses just have a little shell, either like this or like this, and then just have genetic material in there. And that genetic material is held inside of what? What is this thing called? Capsid. And it's made of proteins. And since it's made of proteins, it can denature. And denaturation is breakdown of protein material. So it's sensitive to pH, sensitive to temperature, blah, blah, blah. So when you get a fever, when you have a viral infection, what's your body doing? You try to kill the virus, right? Try to kill whatever infected you, right? And honestly, the rest of the information about viruses, we kind of talked about it. So here it says, viruses can't reproduce by themselves, they're obligate intracellular, right? We talk about bacteriophages, talk about viral genomes, so basically single-stranded or double-stranded RNA or DNA. I don't know why they say that, because that's literally all it can ever be, either single-stranded DNA or single-stranded RNA, or double-stranded DNA or double-stranded RNA, right? Okay. The one thing that we haven't talked about yet, right? Retroviruses. What's the most famous example of a retrovirus?
HIV.
Most famous example of a retrovirus. HIV, what does it stand for? Human immunodeficiency virus. Because it takes your immune system and just goes, done. And now you die of common cold. Sucks, right? Okay, so retroviruses basically have this enzyme called reverse transcriptase, where they take RNA and turn it into DNA. And like bacteria, right, where the virus injects its genetic material into the bacteria and it integrates into your DNA, if the virus makes DNA and it enters your body, it can integrate into your DNA. And now you're doing the work of the virus. And sometimes that virus can code for proteins which destroy your immune system. And you live a very, very long and painful, arduous life and pass away from a common infection. And that's what happens when people get HIV and AIDS. Except we have drugs called antiretrovirals now. You're very going to get HIV because they inhibit reverse transcriptase. Right? Reverse transcriptase inhibitors are antiretrovirals. Right? Okay. Lytic versus lysogenic cycles, we talked about that already, right? So basically, two things can happen. Number one, that DNA integrates into, or DNA, RNA, whatever it is, it gets transcribed and integrates into your body, right? Now either the DNA can integrate, and it'll just keep copying until it's copied into thousands of cells, right, or millions of cells, and it just lays dormant. But that viral DNA is just there, and sometimes it'll code for proteins that are pathogenic, blah, blah, you get a low-level infection. But things can enter, that's the lysogenic cycle, right, where it integrates and just goes and goes and goes and goes, right? Viruses in the lysogenic cycle can enter the lytic cycle. And I don't exactly know what the signal is for that, because I'm not an immunologist, right? but you have great economics over downstate Mid-Alaskan, right? So they enter the lysogenic cycle and enter the lytic cycle, where they form all the proteins
It's
that
kind
are
of hard to understand what he's saying because the audio is not the greatest. But what can I say? I don't know. What can I say?
required for viral synthesis and produce hundreds of thousands of viruses that blow your cell to pieces and they distribute all over your body and they inject their DNA in other cells and repeat the process over again. You can see how that could really hijack your body, right? Because let's say where they form all the proteins that are required for viral synthesis and produce hundreds of thousands of viruses that blow your cell to pieces and they distribute all over your body and they inject their DNA in other cells and repeat the process over again. You can see how that could really hijack your body, right? Because let's say that one viral particle did that, and then replicated to 100,000 cells, and those 100,000 cells each made 10,000 viruses, and now those 1 million, 10 million viral particles go and infect 1 million or 10 million cells. Then those 1 million, 10 million cells become a trillion cells, all infected, and all of them blow up with 10,000 viral particles. You've got a problem now.
You're dead.
That's why they're so good at survival. Cool. What is superinfection? Superinfection is when you're infected by more than one virus at a time. It can be on a cell or an organism. Last fact out of this chapter, and it's one of my favorites. If you guys don't mind, we're almost done. Like we're gonna be done in like 10, 15 minutes, but I need to explain alpha helices and beta pleated sheets before we go on to this next one. Is that okay? Yeah? All right, I promise it's pretty time. And thank you guys so much. It's really hot in here, and you guys have been super patient and very receptive. I really appreciate it. Because listen, if I'm doing this, it means I have the time to do it. Well, whether or not I have the time, this is more of a distraction to me from my actual work than anything else, right? This is like escapism. And I also enjoy it. I love teaching. But the fact that you guys are here, but you clearly have better things to do. I really appreciate it. And I hope that you guys stick around. And tell your friends. If you guys got something out of this, tell all your friends. I would love to see a packed room, right? Okay. So we previously talked about amino acids, right? What are amino acids? The building blocks of proteins. Biochem and orgo people, I am so sorry to bring all this down. If you don't understand what's on the board, it's fine. This is an amino acid. And that R group means that it can be anything, right? It can be like a methyl group, well, not anything, but it can be like any multitude of a bunch of different things, right? That's an amino acid, right? And in your DNA, there are these codons, and those codons get transcribed into mRNA, and those mRNA codons get read by ribosomes, and those ribosomes signal for the addition of an amino acid into a chain. And that's how protein synthesis occurs, in very basic terms. We're going to need to know it in a lot deeper terms soon. It is so fucking hot up here. Okay, so that's an amino acid, and it's going to add to another amino acid. In what type of a reaction? This is called a dehydration synthesis.
I get against that all the time. Dang it.
Where H2O gets kicked out, right? And it's going to look something like this. I'm going to put R prime right here so you know which one went where. Okay? So, right? These two hydrogens from here and this oxygen right here got napped away. They form H2O, and now we have a connection between this carbonyl oxygen and this nitrogen. Who knows the name of this functional group? This functional group right here. So amide, right? Or an amide. An amide or an amide. Professor Litsky was notorious for saying amide, and he swears that's the correct way to say it, right?
Oh, it is. Oh, nice. Dehydration, also known as a condensation reaction. It's like, oh no, what have I learned? What have I learned? But we're okay.
That is functional group right here. So amide, right? Or an amide. An amide or an amide. Professor Litsky was notorious for saying amide, and he swears that's the correct way to say it, right? That is a ridiculously stable bond. And we're going to talk so much about this bond between this carbon and this nitrogen when we get to orgo. Because if that bond didn't exist, we wouldn't exist. And if that bond didn't exist exactly the way it exists, we would never exist. Our entire body, all its processes, all of our DNA, all of our proteins are contingent upon the existence of that bond exactly how it is. It's a beautiful thing. And we're going to talk for 15, 20 minutes about it. All right? So the amide bond is very strong. So now this is something we call a dipeptide. Di meaning two and peptide meaning protein, right? So peptide is protein. A dipeptide is a 2 amino acid protein. This isn't really a protein yet, it's just 2 amino acids, right? But this is not called a monopeptide. This is just called amino acid. Amino acid, dipeptide, that turns into a tripeptide, and then turns into a polypeptide. Many amino acids, right? But when these get linked together, right? And the way you know you're doing this properly is that when they get linked together, can you guys imagine that another amino acid comes to this terminal right here, and there's another NH3, there's another NH3 group, R double prime, and those are going to add together? Can you imagine that, right? The same way these did in your notes, you can look and see how they added, and that's going to do the same thing. So the way you know you do this right, Professor Litsky always said, is if your carbonyl groups are facing in opposite directions, you did it right. R triple prime, R double prime, R prime, R. Down, up. Right? If you go in opposite directions, you did it right. H, and that's it. You guys see how there's a lot of oxygens, a lot of nitrogens, a lot of hydrogens. Who remembers the rule about hydrogen bonding? Chem is FON, F-O-N, right? So hydrogen bonding, what is hydrogen bonding? That's it. Bond didn't exist, right? These are the addition of an amino acid into a chain. And that's how protein synthesis occurs, in very basic terms. We're gonna need to know it in a lot deeper terms soon. It is so fucking hot up here. Okay, so that's an amino acid, and it's gonna add to another amino acid. In what type of reaction? This is called a dehydration synthesis, where H2O gets kicked out, right? And it's gonna look something like this. I'm gonna put R prime right here so you know which one went where. Okay? So, right, these two hydrogens from here and this oxygen right here got napped away, they form H2O, and now we have a connection between this carbonyl oxygen and this nitrogen. Who knows the name of this functional group? This functional group right here. So amide, right? Or an amide. An amide or an amide. Professor Litsky was notorious for saying amide, and he swears that's the correct way to say it. Right? That is a ridiculously stable bond. And we're going to talk so much about this bond between this carbon and this nitrogen when we get to orgo. Because if that bond didn't exist, we wouldn't exist. And if that bond didn't exist exactly the way it exists, we would never exist. Our entire body and all its processes and all of our DNA and all of our proteins are contingent upon the existence of that bond exactly how it is. It's a beautiful thing. And we're going to talk for 15, 20 minutes about it. All right? So the amide bond is very strong. So now this is something we call a dipeptide. Di meaning two and peptide meaning protein, right? So peptide is protein. And dipeptide is a 2 amino acid protein. This isn't really a protein, it's just 2 amino acids, right? But this is not called a monopeptide. This is called amino acid. Amino acid, dipeptide, that turns into a tripeptide, and then turns into a polypeptide. Many amino acids, right? But when these get linked together, right? And the way you know your groups properly is that when they get linked together, can you guys imagine that another amino acid comes to this terminal right here, and there's another NH3, oops. There's another NH3 group. And those are going to add together. Can you imagine that, right? The same way these did in your notes, you can look and see how they added, and that's going to do the same thing. So the way you know you do this right, Professor Litsky always said, is if your carbonyl groups are facing in opposite directions, you did it right.
Carbonyl groups are facing opposite.
Oh, okay. R triple prime, R double prime, R prime, R. Down, up. Right? You go in opposite directions, you did it right. H, and that's it. You guys see how there's a lot of oxygens, a lot of nitrogens, a lot of hydrogens. Who remembers the rule about hydrogen bonding? Chem is FON, F-O-N, right? So hydrogen bonding, what is hydrogen bonding? It's not really a bond. It's a very special, strong interaction between specific hydrogens and acceptor electrons. So in hydrogen bonds, we have donors and acceptors. Right? And it's a special type of intermolecular force, right? Inter meaning between two, and molecular meaning molecules. So it's a special force between two molecules. And we have donors and acceptors.
Neutrophils electrophile.
So whenever there is a hydrogen connected to an oxygen, or a hydrogen connected to a fluorine, or a hydrogen connected to a nitrogen, that's a donor. And it's only going to make sense in 1 second if it doesn't make sense now. And what's the acceptor? It would be the lone pairs on an oxygen, or the lone pairs on a nitrogen, or a lone pair on a fluorine. So these electrons are the acceptors. What do you mean by donor and acceptor? Well, if we have water, which looks like this. And then we have another water molecule like this. They're going to orient themselves in a way. You guys see like there's a line going through the hydrogen. That line is going to link up with this electron pair, and that's going to be a hydrogen bond. Like that. And that's why water configures itself so nicely into ice. It has like that crystalline structure because it lines itself up into hydrogen bonds. Does that make sense? So that's an H bond. Not an H bomb, Oppenheimer, but an H bond. Right? Okay. So proteins are capable. Take your photo. Everyone, take a photo if you want. Everyone. If you have to, look at that every single day. It's really important. So, can I erase? Did everyone take this down? You need a moment. All right. So, those interactions, they're really strong. And since they're really strong, they give us back a ton of energy when they form. I'm not going to talk about what we mean by giving back energy. That's thermodynamics. We're going to talk about it in general chemistry, and it's honestly probably the most important chapter in the entire book. Thermodynamics is very difficult to understand. We're going to have a whole lecture on it by itself. But you guys see here NH, and then over here we have a bunch of oxygens with lone pairs around them, right? So what's going to happen is that the NH and the oxygens are going to link up in a way that lines up those electrons to that donor. And that's going to be an H bond. Does that make sense? So the protein is going to get super long. Like if I could, oxygens are going to link up in a way that lines up those electrons to that donor. And that's going to be an H bond. Does that make sense? So the protein is going to get super long. Like if I could, I would write them in like this big of a font, and it would probably span the entire board over here and more, and keep going till fucking South Korea, right? So what's going to happen is that that very long protein is going to twist and contort in ways that match up the donors to the acceptors. And not just the oxygen acceptors, the nitrogen acceptors as well. Because look on your sheet, the acceptors are lone pairs of oxygens, nitrogens, and fluorines. Fluorines don't normally happen inside of our body. So oxygens and nitrogens mostly, right? And when they configure like that, there's two ways they normally configure. Number one is in an alpha helix. And number two is in a beta pleated sheet. An alpha helix and a beta pleated sheet. What stage of protein folding did I say this was? We have primary, which is the line going to South Korea, right? This is secondary. When the protein contorts and contracts itself to line up the hydrogen bond donors to the acceptors, that's secondary protein folding. So this, if you want to note, secondary structure of peptides. Begin using the scientific terms. Familiarize yourself with them. Everyone following? Yeah? Give me a head nod. Good. An alpha helix is what it sounds like. It's what it sounds like. It's a helix. It twists in on itself, right? And on the inside, what do you think we need? Polar or non-polar things? On the inside. Away from the polar environment, the non-polar things come, right? So you, a rule of thumb for the body. Polar things outside, non-polar things inside. Because the body is mainly just like water circulating around. Water is super polar. So as to not disrupt water, or not disrupt whatever molecule we're making, all the non-polar things kind of cave in on the inside, right? So the inside of the helix is usually non-polar stuff. The polar stuff lays on the outside. And it twists around, and it forms a little column, right? Cool. And we're going to talk more about this when we get biochem, right? Because there's one amino acid that doesn't follow the rule when it comes to alpha helices. What is it?
Proline, right?
It like kind of fucks up the alpha helix a little bit.
Proline.
Proline. Proline is the amino acid that fucks up the alpha helix. What happens is your helix, and a proline comes around and you go helix. And this is called kink. Don't kink shame. Proline's just doing its job. See, I have so many one-liners, man. And this guy's always laughing. All right. So, we got alpha helices. What about beta pleated sheets? Do you guys know Issei Miyake? Rest in peace. He's a really nice designer, right, from Japan. And he has this collection called Onbise, which means the pleated men, right? So what happens is the men's collection of a bunch of clothes that have pleats in them, folds. They're little folds like this. And all of the pants and shirts, they look like this from the top down. So they have these little folds. And like when you want pleats, so what they do is they take your pants and fold them over themselves and stitch it like that. Those are pleats. So when these beta pleated sheets are made, I don't know why the fuck they're called beta, because the first thing is called alpha. Why don't you just call it helices and sheets? And the helices and pleated sheets. Why alpha and beta? Because alpha and beta mean 20 different things, you go to 20 different continents, right? But anyways, beta pleated sheets, what happens is that the protein lines up like this. And then another part of the protein down the chain, somewhere closer to South Korea, lines up like this. And the hydrogen bond interactions happen between those two parts of the sheet, right? We're going to talk about the scariest thing in medicine. These kill you. You ever heard of mad cow disease? Mad cow disease is something. Can I erase this? something known as a prion disease. Prion disease. Prions are aberrant pathogenic proteins. What does aberrant mean? Unregulated. They don't really follow rules. And these proteins normally are what? What do you think they are? Why did I give this 20-minute rundown? They're usually beta sheets or beta pleated sheets. And what happens is that these sheets of proteins come into your body because you ate the brain of a raw cow, like the raw brain of a dead cow, right? And it has these proteins inside of it. And what it does is it collides with one of your well-formed proteins and unfolds the whole thing. Whoa! Because this is such a strong, like, attractive force that your protein just goes ahead and joins the party. And then that 4-folded pleat goes on and hits another protein, does 5. Then it becomes 6, and 7, and 8. And after a while, you have this stack of 100 different proteins just knocking into everything you have and unfolding it. And then you die. Because your proteins no longer work.
Wow.
That's prion disease. So they put it at the end of the virus section of this chapter because they're not really viruses, because they're not contained in anything, but they're just pathogenic proteins. So one of the notes I wrote, they are infectious, wrongly folded proteins, or infectious, misshapen proteins. Make sense?
Yes, sir.
Cool. Go home. No, I'm kidding. All right, guys, thank you so much for coming. Just a couple words before I leave. Number one, please tell your friends to come, because I do not want people to miss out on the information I'm giving. But of course, I recorded the whole thing. I really do hope this is still recording. Oh, thank God. So, let's chat. This is chapter 2, general biology. This chapter is all about reproduction. And when we talk about reproduction, we're not necessarily only talking about reproduction in a macro sense. That's fine.
Hold on, buddy. What do we got? I don't know. It's like some basic stuff. We can go over it, I guess. I kind of want to pre-live this stuff, but I guess.
That's fine. We're not necessarily talking about reproduction in a macro sense. We're talking about cellular reproduction, how cells get from one generation to the next, etc., etc.
Oh, I see.
That,
Cellular reproduction. I thought he was talking cellular. Okay, I think probably here, right? I'm not sure.
Small errors in lasting vector that I want to correct. So number one doesn't actually correct the vector itself. That was holding up pixel proteins. So the pixel proteins have lysine and arginine, which two amino acids that are positively charged. That's making positive, right? I said instead of cysteine, it is lysine and arginine. The second thing is that when I was talking about hydrogen peroxide in terms of peroxisomes, right? I said that they undergo a sort of bond breaking that looks like this. And the reason those arrows only have one end is because those are fishhook arrows. Those are 1 electron arrows. These are 2 electron arrows. The number of tips is the number of electrons, right? So that type of bond breaking, I called it a homolytic. Right? I said instead of cysteine, it is lysine and arginine. The second thing is that when I was talking about hydrogen peroxide in terms of peroxisomes, right? I said that they undergo a sort of bond breaking that looks like this. And the reason those arrows only have one end is because those are fishhook arrows. Those are 1 electron arrows. These are 2 electron arrows. The number of tips is the number of electrons, right? Okay. So that type of bond breaking, I called it a homolytic bond association. It's not homolytic, it is heterolytic. This is called a heterolytic bond association. Hetero meaning 2 and lytic meaning breaking. The bond breaks into 2 things, right? On the 1 and on the 2. It's called a heterolytic bond association, not homolytic. Homolytic would be like this. Bond breaks onto 1 thing. That is homolytic, this is heterolytic, right? Okay, cool. So now that my mistakes from last week are out of the way, we can move on to this week. And this week, not this week, it's the same week, it's just the next day. We're going to talk about reproduction, like I said before, right? And when we talk about reproduction as a whole, and reproduction on a species and individual level, we have to talk about reproduction in the cell. So the first thing we're going to talk about is the cell cycle and mitosis, right? So what is mitosis? Well, your biology, what's mitosis? Yeah, it's just cell division, right? So autosomal cells are said to be diploid, and they contain 2 copies of each chromosome, and then germ cells are haploid. And basically, mitosis, well, your biology, what's mitosis? Yeah, it's just cell division, right? So autosomal cells are said to be diploid, and they contain 2 copies of each chromosome, and then germ cells are haploid. And basically, mitosis allows us to take a diploid cell and make 2 diploid cells out of that, right? So in humans, when we have a cell, right, and it has 2 N chromosomes, meaning diploid, right, meaning that if I were to look at chromosome 1.
Wait, hold on, guys. We'll continue, but I have to pee. I'll be back. right, we're back. Yes, sir. You. Let's get it.
Well, obviously, first I have a pair, right? And that would be diploid, right? And in chromosome 2, I'd have a pair. In chromosome 3, I'd have a pair, right? Now, it's going to get quite difficult when we start to talk about diploid versus single and double-stranded, right? So normally...
My bad. Oh, relax. I just took a little break. Kanye, chill. Okay, I'm back. I'm back, guys. Oh my goodness. I'm here. Don't worry. We'll get through all this today, and I'll be that. Hopefully, it's not too late so I can hit some Candy County videos. Don't worry, I'm here.
When we go throughout the life of the cell, the chromosomes, these are single-stranded, right? And then when DNA synthesis and replication occurs in order to prepare for mitosis, these become double-stranded, right? And instead of 2N, for a time, they become 4N, right? And we're going to be talking about that in a bit. Okay, so, and then why are germ cells haploid, right? Why are they haploid? Well, if we have, let's say, some cell of some organism only has 3 chromosomes, right? So they have 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, right? So they have 6 chromosomes total. Haploid,
Hold on, buddy.
right? Why are they haploid? Well, if we have, let's say, some cell of some organism only has 3 chromosomes, right? So they have 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, right? So they have 6 chromosomes total.
Okay.
Well, if we have germ cells, this is a somatic cell. And we have a germ cell, chromosome 1, chromosome 2, chromosome 3. If we had 2 copies of each, and there's a germ cell that's going to combine with another germ cell to produce this organism, or this cell that will become the organism, right? Now when they combine, there's going to be 12 chromosomes, not 6.
Right.
So in order for the germ cells to combine and create a 2N organism, we need N number of chromosomes. And what's N? N is just however many you have. And that means we have 3 chromosomes. So you guys see how this is N and this is 2N. So the germ cells are N, and
Yes.
the somatic cells are 2N.
Yes.
So in humans, what does that mean?
Got
In
33.
humans, that means that our germ cells have
23.
how many chromosomes?
23.
23.
Professor.
And the somatic cells have 46.
46.
So if I were to take a cell
Professor.
out of my cheek, right? A cheek cell. And I were to put it into a machine and it doesn't karyotype, right? I would see 2 copies of each of my 23 chromosomes, right? And unless they were getting ready for mitosis, they'd be single-stranded, right? Cool. If I were to take a sperm cell out of my body and look at that and karyotype that, I would see a single strand of each of the 23 chromosomes, but not a pair. Right? So that raises some questions.
Yes.
Number one, and the one that is most pressing to the idea of evolution and how people sort of become different is, if we carry 1 copy of each of those 23 chromosomes, how do we get any genetic variation? We're gonna have to talk about that when we talk about meiosis, right? And the second thing being, how do germ cells form in the first place? Like, how do we differentiate between mitosis and meiosis? What are the signals that occur in order for mitosis/meiosis to happen, right? Because how does a certain part of the body know to act a certain way? And like, how do sperm cells know to become haploid, whereas everything else in the body knows that they have to become diploid when they reproduce, right? So we're gonna talk about that.
Yeah.
But before we talk about that,
Yep.
we're gonna talk about the cell cycle. Okay. The cell cycle is basically just a rotation of things that will happen throughout the life of the cell. It's split up into G1,
G1,
S,
G2,
G2,
and M.
and M. And these are phases. G1 phase, S phase, G2 phase, N phase. Right?
Right. Yes, sir.
Okay. So, all of these are referred to by a certain term. Anyone know that term?
No. I'm not sure, buddy. Interface? I think, no, I don't think so. Cell cycle?
interphase.
Oh, I got it. Let's go.
Interphase, right? G1S and G2 are known as interphase. And in my notes here, it says during interphase, individual chromosomes will not be visible because they exist in a less dense
Hold on. Play it.
And in my notes here, it says during interphase, individual chromosomes will not be visible because they exist in a less dense or condensed form called chromatin, right? And DNA must be available for replication.
Right.
The nucleus kind of unwinds, and you get DNA replication or DNA transcription involved or whatever's going on over there, right? The G1 stage is known as the pre-synthetic gap, right? The pre-synthetic gap. Okay? All right. So this is where the cell makes organelles and increases in size.
Okay.
And there's a restriction point after that. I'm going to do that in a different color. There's a restriction point here, right? What's a restriction point? Well, like if you try to go to a concert, right? You need a ticket, right? So this is your restriction point.
Yeah.
And this restriction point is just known as the G1S restriction point. And basically, what's going to happen here is that the cell is going to check to see whether or not it has everything that it needs to move into the S phase, correct? To move into the S phase. And if it doesn't have everything it needs to move on to the S phase, it won't, right? It'll continue in G1 until it's ready to move on to the S phase, or until something else happens. Until a problem is detected in the cell. Maybe the cell has some aberrant gene that can cause cancer, and it needs to be killed. The cell will get killed, right? But if anything's wrong, and the cell determines that it can't move into the S phase, it won't. Does that make sense?
Yeah.
All right. S phase is also known as the synthesis phase. What do you think happens here? You get synthesis of DNA for later mitosis. What did I write? Synthesis of DNA in preparation for mitosis, right? So this is the point right here in S phase where we go from this to that, single-stranded to double-stranded. And that's important because each of those little swingy lines you see is called a chromatin, right? This is very specific language. Each of those is called a chromatin. So chromatins, right? But not just chromatins, they're called sister chromatins.
Yeah.
Sister chromatins, right? Because they exist as part of the same chromosome. So two sister chromatins make up the chromosome at the S phase of the cell cycle. And right in the middle, we have this dot. And it's visible when you look at it. What is that dot? It is the centromere, right? That dot is the centromere. And the centromere is going to be important later, right? Cool. That's pretty much all you need to know about the S phase. Any questions about that? Right? You guys might be wondering, how does DNA replication happen, blah, 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 how does synthesis occur, everything like that. We will get into that when we talk into biochem. We get very in-depth into DNA replication, right? Right.
Okay.
If G1 was the pre-synthetic gap, what is G2?
Post.
It's the post-synthetic gap.
Most likely.
The post-synthetic gap.
That's what I need.
So, this is basically just quality control. And right after G2, what do you think we have? What is there another one of? Right? After G2, we have another red line, another bouncer before the club, right? We got a restriction point. Because clearly, at some point, we wanted to make sure that the synthesis of DNA took place in the proper manner, right? And if we only checked back here, we would have no way of making sure that the replication happened properly, right?
Yeah.
Okay. So like I wrote my notes right there, cells make sure that there are no errors in DNA replication at and after the G2 phase, between the G2 and M. If that was the G1S checkpoint, what is this? G2N, right? Who's a great fan? I know G4 pilots on a first name basis. Yeah, G1, G2. All right. So after G1, S, and G2, this is when the party can really start happening. This is mitosis. This is mitosis. Oh man, look at like people. Hey. Oh, how's Orgo? Good? Lab or lecture? Lecture. Awesome. Some people I recognize, some people I don't. Welcome. I owe you something. Last time, right? Cash.
Cash? What the heck?
Don't raise it around, right? You too, right? No, you gotta earn it. All right, for everyone who's new here, hi, I'm Yusuf Hassan, second year medical student at Downstate. I'm running this lecture for the MCAT. Also, before you guys came in, I had a little QR code up on my laptop to sign up for an email list associated with this program. This man right here, he has a photo of his butt. If you guys are going to stay the whole time, I will have a PDF of it up on my laptop. Also, very informal lecture. If you guys need to leave early and you don't want to bother the guy or notes, come up to me. I'll show you the QR code. You can scan it.
How does good, buddy? Come on.
We were just having a little talk.
We got time.
Actually, you know what? My laptop might die.
Let's go. Keep going. Keep talking.
So, we talked about G1, SG2, okay, whatever. All those together are collectively called interphase, right? Now, the reason they're called interphase is because mitosis is broken up into phases as well. So it makes sense for scientists to be like, everything that happens before mitosis is a certain kind of phase, everything that happens during mitosis and after mitosis is also a phase, right? So we go interphase, mitosis, interphase, mitosis, interphase, mitosis, interphase, mitosis, until we die, right? And that's basically just what happens to cells, right? Unless you're a germ cell, in which case you undergo meiosis. And like we talked about before, you become haploid, is that thing, correct? All right, cool. So are we ready to talk about mitosis, or do you guys want to copy this down like a little more? If you guys want to take a photo, please go ahead. Go, you guys. We're gonna keep moving. The first thing will go slow, like they'll take a bit more time. If you guys weren't here last week, it took us 2 hours and 15 minutes to get to the first chapter of biology, which is normally how long it takes. But as we go on, the lectures will take less and less time. Especially when we get the lectures that I genuinely enjoy. Not really I don't really enjoy this stuff too much, I just know it. But when we get to cardiology, we get to homeostasis, we get endocrinology. We'll do it with endocrinology when we talk about the menstrual cycle. But it's fun to do that. Everyone ready? Let's keep moving. That was exciting, 10 people poured into the room. All right. So, who thinks they know about mitosis? It's not a trick question. You can raise your hands. Who's learned about mitosis before? Yeah, if you think you know about mitosis, you do, because it's almost as straightforward as everyone has always taught it to you. And the MCAT doesn't really discriminate either too much anyways. So, when we talk about mitosis, what we're gonna do, I'm gonna write it up here, right? And it's split up into phases. So here, I'm gonna draw a line, and we're gonna do. I always forget that. Prophase. We do metaphase, anaphase, and telophase, or telophase, whatever you want to say. Okay. So,
That's like a 6.
before they talk about mitosis, they talk a little bit about control of the cell cycle. Like we said before, we have 2 restricted points. What were they again? It was the G1S and then the G2N, correct? So that's where the cell kind of checks to make sure that things are going properly, right? So right here in my notes I have is G1S phase, right? The cell determines the DNA is good enough for synthesis, right? There's no problems in the DNA. If there are problems in the DNA, what happens? Usually cancer, right? And G2, or you just get cell doesn't work, right? And G2M, it checks your atom size and existence of organelles for splitting, right? Because when you give a daughter cell, everything.
Freaking volume.
The cell determines the DNA is good enough for synthesis, right? There are no problems in the DNA. If there are problems in the DNA, what happens? Usually cancer, right? And G2, or you just get cell doesn't work, right? And G2M, it checks your atom size and
Checks for adequate size.
existence of organelles for splitting, right? Because when you give a daughter cell everything that it needs, we don't give it damn mitochondria, it's gonna die, right? Okay, cool.
Right.
What did we say was important about mitochondria last time? What was so special about mitochondria? Huh? Well, yes, but unique about mitochondria. Other than you have no DNA and what else? You get it from your mother. Every single mitochondria in this room can be traced back to the first female human being to ever exist, right? Quite cool. And the MCAT really wants you to know that fact. If they ever talk about mitochondrial DNA and any sort of diseases that have to do with mitochondrial DNA, you got that disease in your mom. There's no other way to happen. Why? Because sperm don't have mitochondria. Sperm cells are not built with mitochondria. Seminal fluid has fructose in it, which the sperm use in order to do the rest of glycolysis to power their motility. But they do not produce mitochondria. And they don't have mitochondria, and they can't do electron transport and acidic acid cycle, blah, blah, blah. It just wouldn't be beneficial. Right? All right. Now, here's the thing. The MCAT, the books at least, right? Like these are the books I'm using. I humbly request that you get them. The Kaplan and MCAT books are quite good. If you get them used, you get them new. Because when they good. Yes. Does not matter what year. As long as you don't go back to 2008, I think you're good. Mine are 2021, 22, because I was fortunate enough to be able to afford the ones that were for the year that I was taking them. But I know that's not, everyone's desire or what everyone can do. So if you guys want to get them from someone who took the MCAT 3 years ago, like I did, right? Because my books are now 2 or 3 years old. Feel free. And it'll probably be the exact same information. And it's not the exact same information. It'll still be on this board, right? Yeah, cool. So the book makes a really, really small reference to cyclins and cyclin-dependent kinases. Basically, cyclins and cyclin-dependent kinases you don't necessarily have to know for the MCAT, but they are these molecules that kind of come and go throughout the cell cycle in order to kind of signal to the cell that this is where we're at and this is what we need to do next. Do you need to know about them? Not really, right? But first of all, before we do anything else there, very specific thing. What's a kinase? You never know what a kinase is. We're going to talk about this over and over and over again. A kinase is an enzyme that...
Oh, kinase. How am I blanking out right now? Phosphorylates, adds a phosphate group, but with the breakdown of ATP.
Phosphorylates.
Yeah, am I spacing out?
Something using ATP. It uses ATP to phosphorylate something. And the reason that's an important distinction to make is because there are other enzymes that don't use ATP to phosphorylate things. They use phosphate groups, inorganic phosphates, right? So, if this enzyme is using ATP in order to phosphorylate, it is known as a kinase, right? So here we're talking about cyclins and cyclin-dependent kinases. A cyclin-dependent kinase is a kinase or an enzyme that phosphorylates something using ATP that is dependent on a cyclin. And a cyclin is a type of protein. So, cyclins rise and fall throughout the cell cycle. Oh, Sasha. Rise and fall throughout the cell cycle, right? And as they rise and fall, they activate and deactivate certain cyclin-dependent kinases that will go and phosphorylate other proteins and things throughout the cell in order to kick off different parts of the cell cycle. That's all you need to know. If you get a passage about them and it explains a little more power to you. But I don't know much else about them anyways. All right. And I have no problem admitting that. I don't want to know about it. All right. Then we make like a little sort of segue into cancer, right? Because cyclin and cyclin-dependent kinases are usually like how people start talking about cancer and how people begin to sort of, begin their discussion about cancer. So when we talk about regulation, we talk about checking, stuff like that. What's the biggest check when it comes to cancer? Everyone knows the buzzword. It's a gene. P53, right? Well, also, this isn't under like the mitosis metaphase. I'm just using it because I put this up too early. But P53 is a gene that goes for a protein that checks the rest of your DNA and checks to make sure it's okay. All right? Also, you guys are right in front of the camera, right? If that camera ever stops recording, you tell me. Okay? Cool. You're in charge. So I don't have eyes that like stretch over there. Okay, so P53 is a gene that goes for a protein that is able to check to see whether or not the DNA is okay. If P53 is mutated,
Annie, are you okay? Are you okay? Are you okay, Annie?
you are simply fucked. You are simply shit out of luck, right? Not always, right? The cell can still recognize that there's a P53 mutation and then destroy that cell, right? And what is it called when we get controlled and organized cell death?
Controlled and organized cell death. Apoptosis.
Apoptosis. What is the other type of cell death?
Necrosis, but it's not controlled.
Necrosis, right? Yeah. Necrosis. When you get to medical school, you're going to learn that there's like 70 different kinds of necrosis. It's like coagulative necrosis and liquefactive necrosis and fat necrosis, blah, blah, and it's all in your pathology lecture. And it's just the worst thing ever. You have to like look at a picture of like, it's like a photo on a screen, right? And it's just purple. And they're like, what type of necrosis? I'm like, I don't know, do purple, right? Stupid. Where's my red one? Okay, so now that we've talked about CPKs, cyclin-dependent kinases, blah, 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 let's finally talk about mitosis, right?
Good, buddy. Well, am I normal?
Okay. In condensation, chromatids, number 1. Number 2, right? You get a disappearing of the nuclear membrane. Nuclear membrane kind of dissolves, right? A lot of DNA is held inside a nucleus, right? And now it needs to be able to move around the cell. This is what you're gonna do is you're gonna rip these DNA pairs apart. You're gonna pull them away from one another, right? So you're pulling opposites apart of the cell, and they're contained within that membrane. They can't do that. So you gotta pull the DNA apart and assemble them into a new membrane that exists in the daughter cells, right? That's what we're gonna do. One more thing they say is that centriole pairs separate. I, for one, let me give you guys a little bit of background, okay? So I studied for the MCAT for 6 months. And in those 6 months, I studied for 500 plus hours, right? 550 something hours like that, right? I took 6 practice exams, each of which I think were like, I think the correct number is like 230, but let's say 200 questions, right? That's 1,200 questions right there. And then I did all of UWorld, which is 2,500 questions. So what's that? 2,500, 1,200 is 3,700. And I did all the AAMC material. Let's say I bumped you up to 5,000 questions. And then I tutored 6 different people on the entire MCAT curriculum. Let's say I did 5,500 questions. In 5,500 questions, I've been asked about centriole pairs once. It's not going on the board, okay? It's not going on the board. What's going on the board is the stuff that I've been asked 10, 20, 30 times about, all right? Make sense? Cool. So when people are like, oh, dude, but like I read this thing in the textbook and it seemed really important, but you didn't talk about it. I didn't talk about it because it's not important, okay? You're talking to the guy who's done like 6,000 MCAT questions. And then how many people done when they come to me? I'm not putting it down or anything, 0, right? I've done 6,000, they've done 0. They're like, oh, it seems so important. It's not. Trust me. I don't mean to say that to put you down. I mean to say that to put your heart at ease. And not every single thing in this book is important.
Thank you.
And like I said before, you maybe need to know 60 to 70% of the information well. You don't even need to master it. You need to know 60 to 70% of the information well to do really damn good on this test. All right? Cool. So, like I said, centriole pairs is not going on the board. All right? What's metaphase? Right? Everyone knows metaphase. What does meta mean? For my language people, meta means the middle, right? So, chromosomes meet in the middle of the cell. What's the middle of the cell called when it's metaphase?
Metaphase plate.
What do you eat your dinner off of? A plate? The metaphase plate. The metaphase plate. Right?
Yes, sir.
Okay, cool. So, you remember those centrioles I talked about before? They're not going on the board, but because you never really asked about them. But what we are going to put on the board is the fact that in metaphase, what happens is that those little centrioles, those things that exist, they move to the opposite end of the cell. So now they're on the board. So the metaphase plate exists, that's where the chromosomes are. And this is when you can start worrying about the centrioles. Because here it's like, oh, the centriole pairs form and then they split out and then they start forming this minute. No. This is where we start worrying about them. Centrioles at opposite ends of cell. What do they use? They use spindle fibers.
I really have bad organization on the paper.
To attach. To what?
What does that say? I mean, I can't even read my own writing. I'm trying to figure out this.
What do they use? Start worrying about the centrioles. Because here it's like, oh, the centriole pairs form and then they split out and then they start forming this minute. No. This is where we start worrying about them. Centrioles at opposite ends of cell.
But I got like one space, a little bit of space.
What do they use? They use spindle fibers to attach. To what?
Centimeters.
Attach to?
Centimeters.
What is it called? Yeah, centromere. Attach to centromeres, right? The little things in the middle that you talk about, the centromeres, right? Cool. Why are they attaching there? Right? Because in anaphase, what's going to happen?
You're gonna pull apart.
In anaphase, they get pulled apart. What gets pulled apart? This is really damn important.
What gets pulled apart? The sister chromatids get pulled apart.
What gets pulled apart in mitosis? Because the things that get pulled apart in mitosis are not the same things that get pulled apart in meiosis. The sister chromatids get pulled apart.
Because the meiosis are called the chromosomes. I think.
Pulled apart where?
I'll opposite ends of the poles.
Opposite ends of the cell.
Yeah.
One goes this way, one goes that way, and here are the cell borders.
Sure.
Right? Those middle fibers form, and these condense, right? And these meet in the middle. The middle fibers go gotcha bitch and just pull, right? Opposite ends of the cell. What happens in telophase? What happens in telophase? Some people call telophase cytokinesis, right? Those are two different things. Cytokinesis is a part of telophase. Number one, cytokinesis. Cytokinesis, what is it? Split the cell in half, right?
Yeah.
Right? What else? You got a new nuclear membrane?
New nuclear membrane. And oh wait, I just said that. Oh, and then you should get you should get the chromosomes like getting loose. Becoming loose. I think, right? Or no? I don't think so.
Brand new, right off a lot. Chromosomes uncoil, right?
There.
And the spindle apparatus dissociates. And that's in my notes.
What he said?
That's a little, and that's in my notes. Chromosomes uncoil, right? And the spindle apparatus dissociates.
Spindle apparatus dissociates. Spindle apparatus. I've in no space. Yep.
And that's in my notes.
Yep.
Right? So that's a little, what's it called? Exercise. At opposite ends of the cell. Yeah.
Right.
So central and opposite ends of the cell, use spindle fibers to attach
Yeah,
to.
I can read that either.
Right. So that's a little exercise, or a recognition test, visual recognition, right? I'm gonna draw a cell right here, and this is the cell of the organism that only has one chromosome, right? From prophase, right? Single or double-stranded prophase. Single or double?
Double-stranded prophase. Double?
Right, double. Right, and then those little centrioles appear, but they don't matter until we get to metaphase, right? Prophase, metaphase. And this diagram is not original from me. It is quite literally just in the MCAT book. Anaphase.
Yes, sir. Shoot, I messed this one up.
Telophase. Oops. See that? I know it's quite small. I apologize for that. I hope you guys can kind of see what we're doing. Right. Okay. So that's mitosis. Right?
Yes, sir.
What are we going to talk about after mitosis? Meiosis, right? Makes most sense, doesn't it?
Yep.
Everything makes sense. That's the thing. If anything ever doesn't make sense, you're the one in the wrong. That's what science will teach you. If something doesn't make sense, you probably don't know it. And I've been taught that many times by being medical school. Okay. You guys have all this down? Or do you want a minute? You guys want a minute? Please feel free to tell me. Good? All right. So meiosis is a lot like mitosis in some ways, just slightly different. It's like meiosis went to college and got a degree and knows how to do more stuff, right? And it's special. But as you guys can imagine, when you take on a lot of responsibility and you begin to do more things with your life, there's also more of an opportunity for things to go wrong, right? Like the other day, I wasn't feeling so great. I had a sinus infection that I'm finally better from, right? And like I forgot my charger at home, and then I forgot my charger here, and I lost my laptop, charger, blah, blah, whatever, and then my necklace broke, and I missed the bus, and then I forgot my wallet at home. Like everything was just going wrong. And it was just like I was sick, and I was trying to go home again the weekend, and my car broke down last week, and there's a lot of things going on. So a lot of things could have went wrong, right? In meiosis, there's a little more going on in mitosis. So the MCAT also likes to focus on what could go wrong, right? So try to keep in mind when we talk about meiosis, what could go wrong, and what would happen if something did go wrong, Because interestingly enough, things do go wrong. And when things do go wrong, you are born with horrible congenital defects. What does congenital mean? You're born with congenital. Right? Congenital. Congenital. A congenital disease is something that you're born with. Okay. So we're going to talk about meiosis. Little bastard. Been talking about this guy since 9th grade. How many years has it been? 2 minutes. 10? Right. Meiosis is split into 2 categories, and scientists are so creative. First one is called meiosis 1, and the second one is called meiosis B. No, I'm kidding. Meiosis 2, right? All right, so over here, so instead of the big line, like the big 4 lines, we're going to go like this. And on this side, we're going to have meiosis 1.
You messed it up, right?
And here we're going to have meiosis 2. Why is it spelled like this?
I have to do your little drawing.
Because myo means muscle. That's why it's not spelled like that. All right. Pay very close attention to what goes in the board now.
Yes, sir.
This is separation of blank, and this is separation of blank. Who can fill in the blanks? My man. You match. Close enough. All right. You're absolutely correct. Say that one more time, louder. Homologous
I can't. Okay.
chromosomes. All right? Sister chromatids. One of these terms we saw before, the sister chromatids thing, right? And one of these terms we haven't seen before. What are homologous chromosomes? They're chromosomes of the same type, right? So in my body, in my cells, I have a chromosome 1 for my dad, and I have a chromosome 1 for my mom. And they float together right next to one another, or wherever the hell, right? And they are homologous, right? Homologous chromosomes, both copies of chromosome 1, right? And even though in my body, right, because I'm a genetic male, correct? In my body, I have an X chromosome and a Y chromosome. Those are still homologous, because they're of the same pair, right? I have 23 pairs of chromosomes, 22 somatic, and 1 sex chromosome pair, correct? In women, they're the same, and in men, they're different, right? Okay, so separation of homologous chromosomes happens in meiosis 1, and separation of sister chromatids happens in meiosis 2. There are a lot of new faces. If you guys missed the last lecture, it is recorded and it's up on my YouTube channel. There's a little QR code going around. A couple people took a photo of it. If you need to leave early, get it from one of them. I believe it was you, and it was you, right? So, but if you're staying the whole time, I'm going to pull it up on my laptop later, or we'll discuss after that, right? Okay, so meiosis is clearly different than mitosis, right? So meiosis 2 is basically just mitosis, right? But meiosis 1 is a little different, right? Okay, so the same things need to happen in order for somebody to go into meiosis. We got to get the replication, the DNA synthesis, the organelles, and everything, right? But before that, we need to separate homologous chromosomes before anything else. And meiosis 1 is still split up into phases, like mitosis, right? So we have prophase. What comes after prophase? Metaphase. What comes after metaphase? Anaphase. What comes after anaphase? Telophase, right? All right, telophase. Cool. So this is basically the same thing. Like I keep saying, it's the same thing with slight differences. Okay. Prophase 1, prophase. Oh, also, I forgot to say, each of those gets a little number next to it. Now that we're special, and it's just a 1. It's prophase 1, metaphase 1, anaphase 1, telophase 1, right? Okay. So prophase, like before, the chromatin condenses. Homologous chromosomes, they form little blah blah whatever pairs, right? They form this thing called a tetrad, right? So they kind of line up and they line up next to one another. That's a little tetrad, right? Okay. Now, one special thing happens in prophase. So this is basically same as mitosis, except that tetrad formation where they stand next to one another side by side. A very special thing happens. What's that very special thing that happens? Crossing over. Why does crossing over exist? Let's talk for a second. So I am a combination of my mom and my dad, right? Hopefully no one else. Okay. So this is the chromosome that my mom gave me, and this is the chromosome that my dad gave me, right? And let's go back into my mom and dad's body when they were getting ready for meiosis, mitosis, whatever, right? Now my mom got a chromosome from her mom and her dad, right? So this is the chromosome that my mom's mom gave her, and my mom's dad, and whatever, right? And then there, right? So now we're talking about my parents. That's my mom's mom, my mom's dad, my dad's mom, and my dad's dad, right? Okay. Now, if crossing over didn't occur, right? And we just had, I need to double strand these. Oops. We're just gonna look at those for now. It's not really need two of them. So when they line up in the tetrad like this, they get super close together, right? And what happens is that these homologous chromosomes, let's say this is my dad's chromosome 1, right? And both of those are the same copy of the one chromosome he got from his mom. And those are both the same copy of the chromosome that he got from his dad, correct? What happens is that little sections of the chromosomes, they swap with one another, right? They swap with one another. So what happens is that a little bit of the green goes on to the rest of the red. And then the red and the rest of the green, right? And then the same thing happens over here, where the little bit of the red went on to the rest of the green. And the rest of the red went on to the rest of the green. And now you can see that these chromosomes, when they eventually part ways, they are solid mixes in some way or another of the chromosome that his mom gave him and the chromosome that his dad gave him. Does that make sense?
Yeah.
Do you guys understand that? All right. So that's the importance of crossing over. It implies a level of genetic variability, right? Because if you go back and you look at the red chromosome and you trace it back to my dad's mom, that chromosome must have been a mix of her dad and her mom. And you trace that back to her dad, it would be his dad. And we get these crossing over events that happen at each and every single generation of donation of genetic material that instills a level of diversity that wasn't seen before. And that's why I don't look exactly like my mom, or I don't look exactly like my dad, although my dad would differ, right? He would say that I do, right? But that's necessarily what happens when crossing over occurs.
Yeah.
Right? So you guys understand that? You guys understand how this increases your genetic diversity? That's a very important part of the first step of meiosis 1. Understood? All right. Cool. What's next? Metaphase? Basically the same. Those homologous chromosomal pairs line up in the middle. Metaphase plate, same thing, right? Anaphase. What's different about anaphase? Those homologous chromosomes can get pulled aside instead of the sister chromosomes. So that little thing pulled to opposite sides of the cell, right? Homologous chromosomes pulled to the opposite side of the cell, right? Okay. Okay?
Yeah.
Cool. Telophase is basically the same, except the chromosomes don't really unwind after that because the job's not done, right? So at the end of meiosis 1, we basically have two cells. Well, at the beginning, right, we had this. And now you have this. Whereas if this were mitosis, you would start with this and end up with this. Do you see how that's different? Because these cells are 2N. 2N, single-stranded. This is 2N, double-stranded. But here we go from 2N double-stranded to N double-stranded. So these... These are haploid cells. These are already haploid cells. Does that make sense? Ploidy is really difficult to understand. It can get tough. Ploidy is quite difficult. So that's why I keep trying to map out exactly what's going on in mitosis and meiosis, right? That's understood. I know my writing's quite small. I'm trying to write larger. I'm just not used to it yet. I know. I've never lectured a class like this. This is my first time ever doing so. All right. So we talked a little bit about meiosis 1, and now obviously meiosis 2. So prophase 2. Anything special about prophase 2? Well, they may be ground with this period, central, blah, 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 right? So nothing special. Metaphase 2. Nothing special. Anaphase 2. So what happens in anaphase 2? The homologous chromosomes are pulled apart here, and now the sister chromosomes are pulled apart here, right? That is known as what? Separation, well, whatever. Also known as disjunction. Okay. Telophase 2, nothing special. So nothing special, sister chromatin fold apart, nothing special, right? This is basically mitosis, right? In mitosis, we already have like that little double thing right there. So see, you take this, you funnel into meiosis 2, and that becomes this. So we went from this to N double-stranded to N double-stranded, and then we get to N single-stranded. And that is our germ cell. And for women, it's eggs or ova singular. And for men, it's sperm singular, right? Yeah. Oh, yeah. So you have the centromere right there. We have little centrioles, and what's going to happen? So that's metaphase, basically. And what's going to happen is that those centrioles are going to pull the sister chromatids apart into opposite ends of the cell, and you're going to get like that line down the middle that they're going to split off eventually, and that's how they're going to separate. Does that make sense? Oh, that one is going to look like. That's a good question, because that double is going to get pulled by the centriole, right? And then this double is going to get pulled by the centriole right here. Okay.
Yeah.
Term earlier called non-disjunction, right? So right here, this is called disjunction, where the homologous chromosomes are pulled apart, right? Disjunction. Later, we're going to talk about a problem. Remember I said there's a lot more going on. So there's more of a chance there's a problem, right? We're talking about something called a non-disjunction error, right? Where anaphase 1 fails a non-disjunction error, right? Where anaphase 1 fails to pull apart a homologous pair of chromosomes. And what happens is that instead of getting this, you somehow still end up with this and an empty cell. A cell that has 2 of the copies and a cell that gets none. And in humans, sometimes it's not a problem. Well, it's always a problem, but it's not a problem for the formation of the cell. But if that cell gets fertilized down the line, now you've got a problem. Who knows what the diseases that can happen due to a non-disjunction error?
Probably. I mean, I think this is we have too many chromosomes in the pair, right?
Turner syndrome is one of them that happens. Yes. Can you catch? Can you catch? Nice. Down syndrome is another one.
Yeah.
Trisomy 21 is Down syndrome. So there's Turner's, there's Klinefelter's, there's Down syndrome.
Where one X chromosome is missing. Yeah.
And there's XYY or Superman syndrome, right? All right, cool. All good here? Let's move on.
I guess so.
So why is XYY syndrome called Superman syndrome? Well, it's because you have 2 Y chromosomes. Does it actually give you superpowers? No, I think it makes you infertile. Sorry? Yeah, aggressive and infertile. Great combination.
Oh, it's messed up.
No, it can happen. Yeah. I mean, considering the Y chromosome goes for like testosterone production, it can increase your testosterone. But then again, like normal testosterone in males varies over a very, very large margin. So like the lower bound of normal testosterone is like 200 something, and the upper bound is like 970 nanograms per deciliter. So it's possible that when people have overproduction, it just doesn't push them above the lower bound. So that means that, right? Yeah, next time. Let's stop by. All right, now we talk about meiosis and the formation of germ cells. We're going to talk about the reproductive system. So they talk about X and Y chromosomes, right? So XY is male, XX is female, right? And that's determined by the 23rd pair of chromosomes. You have 1 through 22, and then you have your sex chromosomes. And these sex chromosomes can either be XX or XY, right? You always get a Y chromosome from your dad. If you have a Y chromosome, you got it from your dad. An X chromosome can come from your mom or your dad, right? Why am I bringing that up? It's important because when we start talking about X-linked diseases, we're going to be able to track them. So before we do that, now that we're talking about chromosomes and everything, before we talk about the reproductive system, let's talk a little bit about X-linked inheritance, right? Let's talk a little bit about that. So, I, myself, this is a true story. I am colorblind. I am red-green colorblind, right? And colorblindness is a trait that exists on the X chromosome, right? So if you were to karyotype me and write out my genome, you would see a defect and my X chromosome that confers colorblindness. Who did I get that from? Mom. I had to. Why did I have to get it from my mom? Because my dad gave me that. And my dad didn't give me that, I'd be a woman.
Right.
Right? And my dad gave me an X chromosome, I'd be a woman. I'd be one of my sisters, right? Now, let's take a look at my sisters. Who can figure out, given the information I just told you, that my mom gave me colorblindness, or let's talk about my sister Sarah, right? I have 2 older sisters, not you. Does her karyotype look like this? Or does it look like this? Or does it look like this? Can you figure it out given the information I gave you?
It looks like B. Right? What do you guys think? What do you guys think? The middle one? Is anybody even here? I don't know if anyone's even following along. Anyways, I think it's the middle one. I'm studying Kanye. Chill, buddy. Kanye, don't worry, buddy. We're not gonna make it.
No, you need more information. What are the 2 questions you need to
Okay, never mind. I thought it was the middle one.
ask? Is my mom colorblind? Is my dad colorblind? And is my mom a carrier? Are the questions you need to ask, right? Because just because my dad gave me the Y chromosome doesn't mean he's not colorblind. What if he's just like me and he got that from his mom, right? So you wouldn't know. So you don't know the genotype of my sister, right?
That's a good one.
Okay,
Good question.
let's talk about how I got that. So this is my mom, right? And I'll tell you that my mom got that from her dad, because her dad is colorblind, right? And my mom's mom is not colorblind nor a carrier, so my mom is that. So like she got that from her dad and that from her mom. And my dad is not colorblind. He's not, right? So we got the Y chromosome from his dad, we have the X chromosome from his mom, right? Do we know if his mom's a carrier or not by that? No. Because if she was, there'd still be a 50-50 shot of her passing on that defective or good X chromosome, right? So those are my parents. Who remembers Punnett squares? That's my mom, that's my dad. And there's me. So when you see a male with an X-linked disease like colorblindness or hemophilia, I think hemophilia is X-linked, right? When you see a male with an X-linked disease, right, it had to have come from mom. And when you see a female with an X-linked disease, that means something special. When you see a female with an X-linked disease, right, especially X-linked recessive disease, this colorblindness is what? It's X-linked recessive. Meaning that you need 2 copies, or if you're a woman, you need two copies, and if you're a man, you need just 1 copy because you only have 1 chromosome, right? But since it's recessive, in women, it needs to display like this. So if a female is colorblind, you know one thing off the rip. Her dad's colorblind. Has to be. Because the dad has to pass on the X chromosome. So her dad is definitely like this. And where did that one come from? Comes from the mom. But mom could be this or this. Here it's a 50-50 shot, you pass that on, and here it's definitive. Does that make sense?
Yeah.
You guys understand X-linked inheritance? Okay? Cool. The book does a lot of talking about the reproductive system. I think it is best done through diagrams. So if you guys would like, I do urge you, once again, I'm saying to get these books, right? The Kaplan and MCAT books. Look through the diagrams, all that stuff, you have it? Good. All right. Look through the diagrams and everything like that. That's where I get all my notes. And that's how I sort of, learn all this stuff. I will do very, very crude drawings of things that are in the book, but you should definitely take a look at them. One more thing. The Y chromosome. It's special. It contains a specific gene that the X chromosome doesn't have. What is that gene called? It's called the SRY gene. It is called the sex-determining region Y. Sex-determining region of the Y chromosome, right? And this activates at, I believe, 6 to 8 weeks of gestation. I'm in Unit 5 in medical school right now. Unit 4, before summer break, was reproduction. I hate this term now, because I had to learn every single thing that happened inside of a fetus when this activated. It was the worst, all right? But all you need to know is that this codes for male gonad formation. So basically, your balls run for the first time, right? And that's basically what happens, because before that, the fetus, embryologically, it looks like a female. It has ovaries, or like what will become ovaries, and whatever. And if this gene never activated, right, or if the transcription factors that code for this activation, or the different proteins and stuff like that that code for male gonad formation, if those never occurred, it will continue to be a female. So by this gene, we confer male sex. Yes or Y? 6 to 8 weeks of gestation. So that's what it happens. What's gestation? Like when the baby is developed inside of the mother. Right.
All right, buddy, let's go. Come on. What's all this pausing about?
All right, cool. Let's move on. Male reproductive anatomy and physiology. So first, we have the testes, or the testicles, which is a functional unit of male reproductive anatomy. That's where testosterone is produced, most of it. That's where sperm is produced, etc., etc. Why do I say most of testosterone produced there? One of the places in the body that happens. You don't even know it, but fun fact. That's where testosterone is produced, most of it. That's where sperm is produced, etc., etc. Why do I say most of testosterone produced there? One of the places in the body that happens. You don't even know it, but fun fact, it happens in one of the places in the body. I'll let you guys look that one up for there. Oh no, it's not a question, but I'm just saying that testosterone can be produced in one other place in the body. You don't need to know it. I'll have you guys look it up for later. Not that. Good guess though. All right. In the testes, we have these things called seminiferous tubules. Seminiferous tubules. These produce sperm. This little arrow down from an underlined object means that this is a part of that, or I'm elaborating on something, right? So the seminiferous tubules are a part of the testes. The testes also have interstitial cells of Leydig, probably the dude who found them, right? And these produce testosterone, which is eventually going to make my hair fall out. Special question: what does testosterone turn into that makes men's hair fall out? Sorry? Well, if that's going to make your hair fall out, you wouldn't have it. It's called DHT, dihydrotestosterone. And some men in later life go on a medication, commonly known as minoxidil, right? And minoxidil inhibits the conversion of testosterone into DHT to minimize hair loss. Cool. Interstitial cells of Leydig, Leydig, Leydig. Interstitial cells of Leydig. They produce testosterone. And the seminiferous tubules produce sperm. Right? From the seminiferous tubules, the sperm enter the epididymis. The epididymis. Right?
From the what?
From the seminiferous tubules, the sperm enter the epididymis. The epididymis. Right? And this is where sperm are stored until ejaculation. I always hate teaching this chapter because what happens is I begin teaching a student the MCAT for like the first time, right? And we talk about cells, we talk about mitosis and meiosis, and literally the next time I meet with them, I start teaching about cock and balls. It's just the worst. It's like not a good one. But this is the way the textbook runs, so I'm gonna have to teach it, so please bear with me. I promise it's not all like this. So, the epididymis, right? When you guys are in medical school, and you do anatomy lab, and you get to hold the testes, right? You like hold them, dissect them, cut them open, look into them. The epididymis is going to sit on top of the testicle. And it's connected by a tube to the rest of the reproductive system. Because the testicles, they hang, and then the rest of the system's inside the body. Why do the testicles hang? The ovaries don't. Why do they hang? Good job, you catch? The bodily temperature is too high for sperm to survive, right? If the testicles are inside of the human body,
Hold on, guys. I'll be back. Hold on.
They would die, and they would be infertile, right? And no more human race, bye-bye, right? Which is why there are muscles that are not under our control, like men's control, that drop and elevate the scrotum, which the testicles sit inside of, depending on the temperature. When the body is very hot, like when you're sick, the scrotum drops and the testicles move away from the body. And when the body's cold or cold outside, they move closer to the body, right? In order to control and thermoregulate the ideal temperature of sperm formation. It's a really beautiful process. It's very interesting, right? Cool. So, we talk about ejaculation, right? So in ejaculation, sperm travel to the vas deferens. The vas deferens is the tube that the epididymis connects up to, right? And then from the vas deferens, they go into the ejaculatory duct. And this is bilateral, right? So the left and right testicle go up the left and right vas deferens into.
All right, hold on. Definitely won't be able to be a urologist or a gynecologist, maybe, but I can't be a urologist.
Left and right ejaculatory ducts, and then those two ducts fuse, right? Those two ducts fuse into the urethra. Into the urethra. Which is actually, a lot of people think the urethra is just like the piss hole, right? It starts inside the body. The urethra continues out towards the penis, right? So it's literally the hole that goes through, that's the urethra, but it starts inside the body. And then the urethra goes into the penis, and the sperm will exit with the. Seminal fluid and blah, blah, whatever it is, right? One thing they didn't mention here, because nothing really travels through it, it just like inserts things in the Cowper's gland, right? The Cowper's gland, or the bulbourethral gland, is going to add some of the seminal fluid, which we're talking about in a second. So right here, it notes, sperm passing through the reproductive system, basic seminal fluid produced by seminal vesicles, the prostate, and the bulbourethral gland, or the Cowper's gland, right? And produces clear viscous fluid that clears out the urethral. Okay, that's important. So the one thing about the Cowper's gland, which is one of the three things that produces seminal fluid, but you don't really need to like keep that in mind. The one thing you do need to know about the Cowper's gland, also known as the bulbourethral gland. The Cowper's gland, right? It produces fluid that quote unquote cleans out the urethra before ejaculation. Sorry? This is urethral gland. The Cowper's gland, right? It produces fluid that quote unquote cleans out the urethra before ejaculation. Sorry?
even somebody in the video is laughing. What the heck? Oh my goodness. Oh wow. Okay.
Yes, pre-con. That's what this is. Cleans out the urethra prior to ejaculation. Okay? So, besides that, why? Why? Why does that need to happen? Why do we need to clean out the urethra before ejaculation?
Probably to make sure that the sperm is released healthily. I don't know if that's a word, but I think so. But it's probably to make sure that the I mean, I would think so. Maybe it's also so it's not carried over. It's not carried over into the vagina.
Yeah, and acidity.
Yeah.
pH. slightly acidic. And if sperm mixes with that acidic environment, it's possible that some of the sperm die, right? They die. And that means lower chance of fertilization, right? So we send out fluid in order to A, lubricate the urethra, and B, make sure that the sperm don't die, blah, blah, right? Okay. The book goes on and on about spermatogenesis, like over and over again. I have 2 lines of notes about it. I've also maybe seen 2 or 3 questions about it ever. I'm not going over it. If you'd like to study it yourself, you're more than free. Are we all good on male reproductive anatomy? Okay? So just as a diagram of like how all this works, like one-sided, this is the testicle, this is the epididymis, this is the tube leading up to the vas deferens, right? And then it connects to all the garbage over here and then out the urethra, right? But that's the Cowper's gland, the prostate, and all, and everything, the ejaculatory duct, and all that, right? And this is like the best drawing I can possibly do, I promise you. I'm a much better speaker than I am an artist. I hope you agree. Good? All right, go.
Sure.
Also, if you're wondering why I'm not using the entire board, it's because of that book. Let's talk about female reproductive anatomy and physiology. By the way, I use shorthand for male and female as well. You guys have probably seen them before. This is male, and this is female. It usually goes up. Yes, it usually goes like this, as to not confuse one with the other. Yeah. I believe it's up and to the right.
Because my biological told me it's like Roman soldier. Okay, and the thing is like
That's funny. All right, cool. This is, for some reason, by the textbook a lot more simplified than male anatomy, although it is probably 10 times more complicated in real life. The only thing they want you to know about is the ovaries, which consist of thousands of follicles, right? You have the ovaries. And one of those follicles each month will develop into a egg cell. It's very cool. When you guys get to medical school, you look at histology of the ovary. So basically, you take a slicer and you look it under a microscope. You can see like 7, 8, 9, 10 follicles at different stages of development, right? And then what happens is that when one of them becomes the mature egg, it releases chemicals that make all the other ones stop developing. It's very cool. Very good. And then restart, right? We will be going pretty in depth about the menstrual cycle, and it's ridiculously complicated. I just need you to stick around with it. I don't know why they put it right at the beginning of the book, because there's a whole chapter on endocrinology, and it would have been way better to put it there. But they put it here instead. But I guess it's a good introduction to how hormones work. Okay?
Sure.
All right. You have the ovaries, right? And then that's kind of it. You also talk about the fallopian tube and the uterus, right? So the fallopian tube, the egg cell travels to the uterus via this canal. And what's special about the fallopian tube?
Fertilization occurs.
This is normally the site of fertilization. Can you catch?
Oh.
Cool. Right? And what about the uterus? That's like the big balloon. That's like HQ, right? It is a muscly organ that connects into the vaginal canal, right? So if we had the uterus over here, very, very muscly, surrounded by smooth muscle like this, right? It would thin out over here, and this entrance here would be what?
Cervix.
A very, very small pinhole, the cervix, and this would be the vagina, the vaginal canal. Because the vagina is the outside, and this is the vaginal canal. And this is the uterus. It has a little bulb top right here. It's also incredibly small. Like, when I did dissections in an anatomy lab, the uterus is maybe that big, which is wild when you think about how large it gets when a woman gets pregnant. It's ridiculous. It's not even the size of my fist. And on either side, it connects via fallopian tubes into the ovaries. Ovaries, fallopian tube, uterus, cervix, vaginal canal, vagina. That's what you need to know in terms of female reproductive anatomy. And this all floats right here. Just almost at the level of the belly button is the top of the uterus, and then it goes down, right? The ovaries hang out right over there. Okay? Cool? This one. Yeah, so the uterus, the muscly organ that connects into the vaginal canal, right?
Yep.
The fallopian tubes, they are lined with something. They are completely filled with something. What are they filled with? Little hair-like structures. Not fibulae, there's very specific things in there. Cilia.
Oh, really?
So what happens is that when the egg cell exits, it floats into the fallopian tube and it's gestured along. It's just slightly rocked towards the uterus. And it's just gently glided along the way by the cilia, right? Where else in our body do we see cilia? You said last week, the tube. And it's just that when the egg cell exits, it floats into the fallopian tube and it's gestured along. It's just slightly rocked towards the uterus. And it's just gently glided along the way by the cilia, right? Where else in our body do we see cilia? You said last week, where? The respiratory tract, right? And that's how we don't just like gently glide things along. We fucking kick pathogens out of our respiratory tract and cough them back out, right? Towards the uterus. And it's just gently glided along the way by the cilia, right? Where else in our body do we see cilia? You said last week, where? The respiratory tract, right? And that's how we don't just like gently glide things along. We fucking kick pathogens out of our respiratory tract and cough them back out, right? Cilia. What are cilia made of? You said last week? Well, what structure? What structure makes up cilia? Someone look it up and tell me. What are cilia made of? You got a question? What are cilia made of? Microtubules.
Oh, okay.
Microtubules make up cilia, right? So, in medical school, you will learn about a specific disease that confers a problem with forming microtubules. And these people are infertile, whether you're a man or a woman. Why? Because you need flagella for sperm, and you need cilia for women, and they get repeat upper respiratory infections. Because the cilia in their respiratory tract, it doesn't work. It doesn't get the viruses and pathogens out. So it makes sense. That is what we call pathophysiology. So that's called Cartagena syndrome, right?
Cartagena syndrome, interesting. Where is... okay. Yeah.
Cartagena syndrome. And in Cartagena syndrome, you can have the destruction of cilia, and that will necessarily not allow you to have you lose microtubules, and you won't have the function of cilia, and blah, blah, blah. Okay? Cool. They go on and on about oogenesis, which is the formation of sperm. All right. And now, unfortunately, I have to torture you. Before I torture you and we start talking about hormones, I'm going to stop the recording, and we're just going to have a chat. That's not important video. All right, we're live. This is a re-recording of a lecture that I gave way back when, and the audio actually cut out during my recording. It was really sad. And that's also unfortunate because this is a really, really high-yield concept on the MCAT. This is female sexual development. Specifically, we're going to be talking about the menstrual cycle. Okay.
Why do you cut out the video?
It's female sexual development. This is part of general biology, chapter 2. Right. So, earlier in the chapter, we talked about ovaries, right? Ovaries are the functional sexual organs of females, right? And they are derived of embryonic structures, the same embryonic structures as the testes, because we already spoke about earlier in the chapter, male sexual development. And they're also under the control of FSH and LH.
I think we're good. I need to pee. I'll be back. I'm with you. We got 20 minutes on it. Yeah, let me just be real quick. Excuse me. All right. Let's finish it up.
As a reminder, since this is
It's
real
like he went from no beard to beard. Time jump for sure, huh?
recording, FSH is follicle stimulating hormone.
Run the bench.
And LH is luteinizing hormone.
Right.
LH. Both of these are collectively referred to as the gonadotropins because they are tropic to the gonads, right? Meaning that they are the middlemen of the messengers, that they go to the gonads and the gonads release something else and something happens, right? So, are collectively referred to as the gonadotropins because they are tropic to the gonads.
Tropic to the gonads.
Right? Meaning that they are the middlemen of the messengers, that they go to the gonads and the gonads release something else and something happens, right?
Okay.
So, in men, right? In men, what does FSH do?
And then FSH would stimulate the release of spermatogenesis. So spermatogenesis.
Does anyone remember? It's the other way around. Right. So, men don't have follicles, right? But the way I think of it, men don't have follicles, but if they did, they make sperm, right? FSH, spermatogenesis. LH is for testosterone. And you know that because it's opposite for men and women. In women, FSH makes estrogen.
FSH makes what? Wait, hold on. FSH. I thought FSH was the shoot. I'm gonna have to take a look at my notes. FSH. I thought that was the maturation of the follicles, or no? Yes. And LH was ovulation. But he's saying so FSH makes testosterone.
Estrogen. And LH.
I'm sorry. He's saying FSH makes estrogen. Okay, I should write that down.
Opposite for men and women. In women, FSH makes estrogen. And LH makes progesterone. Later down the line. Because remember, progesterone isn't there in the beginning, right? It has to be induced. So these are the gonadotropins, right? We're talking about men, then we're talking about women. But one of those in both people, in both types of people, is tropic. Meaning that they release something else to do their effects, right? These are called the gonadotropins. And since they're the gonadotropins, their release is regulated by GnRH, gonadotropin releasing hormone. Gonadotropin releasing hormone. Okay. So let's draw out a little chart.
What are we at? Oh no.
GnRH from the hypothalamus fires onto the anterior pituitary.
I can't see.
And this releases
Oh no.
LH and FSH. And since we're talking about female sexual development,
That's
FSH is going to hit the hypothalamus.
I can't see.
Fires onto the anterior pituitary from the hypothalamus. Fires onto the anterior pituitary. And this releases LH and FSH. And since we're talking about female sexual development, FSH is going to hit the ovaries.
FSH will hit the ovaries. Yeah, that makes sense.
And that's going to release estrogen.
Okay.
And then LH is going to hit the... Let's talk about it.
Talk about it what?
Let's talk about it. H is going to hit the Let's talk about it.
LH connected to what?
Let's talk about the menstrual cycle. Because the menstrual cycle is basically the
What? Finish it. What the heck? What's up with this guy? What the heck is going on? Freaking jerk. Would it be progesterone? Is that what he doesn't want to talk about it? Or what? Would it be progesterone? Why doesn't he want to talk about it? Hold on. We're waiting. I really got. We'll be on 413 fertilization. Definitely see what he has to say about embryology. The rest, I don't know. I think I'm okay, to be honest. But I still haven't hit yet. Might as well just finish it all right.
up and down action of these hormones over time and how that kind of influences the way that the female body works. So just an overview. The female body, you've got the uterus, right? And that uterus connected to two fallopian tubes, which connect to the ovaries, right? And here is the cervix, the entrance to the uterus.
Okay, so LH would surge mid-cycle LH in the ovulation, triggered by rising estrogen levels, causes ovulation, and corpus luteum. Prepares the endometrium for implantation. Graphene follicle to rupture. Secondary oocyte into the fluid. Okay.
Here is the vagina, or the birth canal.
Estrogen. So ovulation to trigger plus progesterone via corpus luteum. Why did you just say that? So LH is ovulation trigger. Plus progesterone via corpus luteum. Via the corpus luteum.
Right? And inside of the uterus, implantation of the fetus normally is going to occur like here. So we need to thicken and thin and control the environment in which the embryo is going to implant, right? Inside the uterus. That's the goal of the menstrual cycle. Right. So the menstrual cycle is a cycle of multiple days, it's like 28, 30 days, something like that, right? It begins when menstrual flow starts. Day 1 is the first day of active menstrual bleeding, right? And when that happens, it happens because, well, it's called shedding of the uterine lining, right? It happens because the levels of all these hormones drop off from the end of the last cycle, right? And when those hormones drop off, you know that LH and FSH go back and turn off GnRH back to the hypothalamus. So when these drop off, GnRH gets turned back on, and then your FSH and LH levels are once again allowed to rise. So phase 1, increase GnRH, yields.
I need more space. Just on any page. Increased GnRH.
Increased FSH.
Increased FSH.
And LH. And that's
LH.
going to increase estrogen.
Increased estrogen.
And this estrogen,
okay,
right, leads to thickening of endometrium.
Okay. Sure.
Thickening of the endometrium. It's going to make the blood vessels, it's going to vascularize, it's going to grow, it's going to become more mucousy, it's going to become a warm, welcoming environment for
Sperm.
the embryo.
Wait. Oh, okay.
And the estrogen's also going to
Yeah, that makes sense.
go back and shut down GnRH secretion. Right? And shut down GnRH. It's going to make the blood vessels, it's going to vascularize, it's going to grow, it's going to become more mucousy, it's going to become a warm, welcoming environment for the embryo. And the estrogen's also going to go back and shut down GnRH secretion. Right? Right. But you can think of estrogen kind of growing in a delayed form, right? So even though as it rises, it turns off the GnRH secretion, there's still FSH floating around that's increasing the estrogen, right? So the second step is when estrogen levels peak. And this leads to a flip of the feedback, which means it now positively impacts GnRH secretion, which leads to massive spikes in GnRH. Why? I've asked the question so many times, I still have not an answer. That massive spike now positive, and this leads to a flip of the feedback. Which means it now positively impacts GnRH secretion, which leads to massive spikes in GnRH. Why? I've asked the question so many times, I still have not an answer. That massive spike in GnRH leads to a massive spike in LH, or sorry, a massive spike in FSH, but more importantly, a massive spike in LH. And that LH spike is colloquially known as the LH spike.
Okay.
Who knows what the LH spike does? Not yet. Ovulation. LH spike leads to ovulation.
Yeah.
So, all of that stuff in the first step, right? What was that doing? It was stimulating the development of one of the ova, right? And it's carried inside of a follicle. And that follicle makes estrogen and blah, blah, whatever else, right? And when the LH spikes, the egg, it breaks out of the follicle, the ruptured follicle, right? So when the LH spikes, that egg cell breaks out, and now it's allowed to get fertilized. That ovulation, there's only one woman in the room right now, but at around day 14, after your last menstrual cycle began, you may realize that you have abdominal pain out of nowhere, right in the middle of your cycle, between two cycles. You're like day 14, day 15, there's abdominal pain. That abdominal pain is called Mittelschmerz, right? It's called Mittelschmerz. It's a German word, I think, right? And it describes the pain that is associated with ovulation, right? That pain.
Well, where does it? So, I'm confused. I think that would really help out. Growing follicle, release of the what? Hold on. Ovary releases a mature egg ovum.
Pain is called Mittelschmerz. There are many women who that's actually incredibly painful. So they go to the emergency room and they always ask the question, "When was your last menses? When were you last on your when were you last on your period?" I was like, "Oh, about 2 weeks ago." Classic Mittelschmerz. All right. So this first part is called the follicular phase.
Yeah.
The second part is called ovulation. Right. And when that FSH goes up and the LH goes up, right, the FSH is just going to lead to more estrogen. And estrogen is going to further deepen the endometrium. Right. Next is the luteal phase. So LH. Now we're going to talk about LH. Next is the luteal phase. So LH. Now we're going to talk about what LH does. So the LH stimulates the ruptured follicle to become the corpus luteum. And this corpus luteum, under the response of LH, under the luteum, stimulates the ruptured follicle to become the corpus luteum. And this corpus luteum, under the response of LH, under the control of LH, continuously secretes progesterone. And what does progesterone do? Estrogen builds the lining, progesterone maintains the lining. Which is why when women, we'll talk about that. So the LH, what does it do? Corpus luteum, progesterone. Estrogen builds the lining, progesterone maintains it. Which one's more important?
Estrogen.
Maintenance. Because if you don't maintain it, sheds.
Oh, that's
Right.
true. I guess so.
Estrogen builds the lining, progesterone maintains it. Which one's more important?
But if you don't have it there, how can you
maintain it, sheds.
maintain? They're both
Right.
important.
LH stimulates ruptured follicle to become the corpus luteum. When LH goes down, the shedding will happen. And that progesterone is going to go back and negative feedback on GnRH. So, corpus luteum releases progesterone. Now, the fun part. 2 pathways to go down now. Can I erase this? Yeah, everyone? I just asked if I could erase it. All right, there's 2 paths to go down after the luteal phase. So let's draw like 2 little things like this. No implantation. Implantation. If there's no implantation, all the men in the room, since we don't know anything, what happens? Menses. Right? The reason I said that is because a bunch of people left after the last lecture because we're continuing, and all the men stayed in the room because we know nothing about the menstrual cycle. There's one female. Thank you. Thank you, Laura. She's our fact checker. When no implantation occurs, if you don't become pregnant, right? Basically what happens is that the corpus luteum becomes insensitive to LH, which leads to a drop in progesterone, which leads to shedding of the uterine lining. And that drop in progesterone is going to turn off the negative inhibition of GnRH, right? And then that's going to restart the menstrual cycle. The GnRH is going to come back up and blah, 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 whatever. Right? Oh, shedding of the uterine lining. What a complex process. If fertilization does occur, an implantation does occur, the embryo secretes a specific compound known as beta human chorionic gonadotropin, beta HCG. That's what you're testing for in a pregnancy test. Beta HCG, beta chorionic gonadotropin, human chorionic gonadotropin, beta HCG. That's what you're testing for in a pregnancy test. Beta HCG. And beta HCG is an analog of LH, but it's an analog of LH that the corpus luteum cannot become insensitive to. So it maintains the progesterone concentration until implantation and placenta forms, and then the placenta is able to maintain pregnancy. Good. One more thing, menopause. As a woman ages, her ovaries become less and less sensitive to FSH and LH, and they atrophy, right? Which leads to decreases in estrogen, decrease in progesterone, and menstruation stops. And you have increased FSH and LH with minimal response. That is menopause. Right. So, before we go, let's just go over the whole thing one more time through dictation. Right?
Yeah.
So, shedding of the uterine lining occurs from the previous cycle because of drops in progesterone. The drop in progesterone takes off the negative feedback of GnRH. GnRH goes up, which releases FSH and LH. FSH stimulates the follicle to allow the ovary to allow the ovum to develop, and the follicle begins to, and the ovary begins to produce estrogen. Estrogen leads to thickening of the endometrium, and specifically they use a term sometimes, glandularization of the decidua. It's just like blood vessels and mucus production inside of the endometrium, right? We move on. Next, the LH doesn't really do much yet, but the estrogen is going to have negative feedback on the GnRH. The estrogen is going to continue to climb because there's still FSH floating around the body, right? Now, when the estrogen reaches a peak point, that negative feedback is going to turn into positive feedback. And you're going to spike in GnRH, a spike in FSH, and more importantly, a spike in LH. That spike in LH causes ovulation. So the ovum ruptures out of the follicle, and that ruptured follicle, under the control of LH, turns into the corpus luteum. Now we enter the luteal phase, where the corpus luteum is being acted upon by LH in order to produce progesterone. That progesterone is going to have negative feedback on GnRH and decrease all of that, but estrogen is still working, right? Still building the lining. And now the corpus luteum, being controlled by LH, is going to continuously secrete progesterone. And progesterone is going to maintain the uterine lining, right? Very important. If you lose progesterone, you lose the uterine lining, right? Now, one of two things can happen. Either the woman is, never mind, the fertilization occurs, right? Which normally occurs inside of the fallopian tube, and you get the formation of an embryo. That embryo can come in and implant, and we can begin to secrete beta human chorionic gonadotropin, which is an analog of LH that the corpus luteum cannot become resistant to, right? Which means progesterone will continue to be produced until the placenta is formed and it takes over in the production of estrogen and progesterone. But if fertilization and implantation do not occur, the corpus luteum is going to become insensitive to the LH produced by the body, and it's going to stop producing progesterone. And that stopping of the producing of progesterone leads to shedding of the uterine lining, and once again, the GnRH increases, restarting the cycle. So that was the menstrual cycle and female sexual development. Hope you enjoyed. I'll see you guys next time. Have a good night. Now, okay. So welcome back. This is day 3, I believe, right? And day 1 and 2 went pretty well. Unfortunately, the recording from day 2 cut out. I was telling you guys before that the audio just didn't work perfectly for day 2, so I have part 1 uploaded on the YouTube channel, and you'll be able to find that on the little drive link that I shared. So for everyone watching back home, what I'm going to do is underneath each YouTube video, I'm going to have a link to the spreadsheet. If you guys saw, there's a spreadsheet of all the lectures. And inside of that spreadsheet, I'm going to put any of the mistakes that I made during the lecture. I'm going to re-elaborate on them and fix them in that spreadsheet, right? And the spreadsheet will also act as a preemptive syllabus so that if you guys look, you can see on send out, right? In the schedule, the Excel spreadsheet, because my study decided to take the exam. Most people decide to take it around April, May-ish, right? Some people opt for June, some people opt for March, right? It really depends. So take a look at that study schedule, look at how I organize things, take a look at like what we're going to be getting at later, right? And it'll just basically be the way that I formulate the schedule around this. Because this pace that we're going at, like although this is a lot of information, this is quite slow, right? So we'll spend about 2.5 weeks getting through biology, which is the first book, and it's very dense. That is 100% true. But we should not be spending that much time getting through like general chemistry, or like specifically more so orgo. Orgo should take like 2-3 sessions, we should be done. And that's just reflective of how much of the information is actually on the exam for organic chemistry. Biochem we can spend a bit of time, right? Because that's a large portion of the exam. It's quite difficult. All right. So what did we talk about last time? Would someone like to remind me? Sorry. We talked about the menstrual cycle at the end. That's the part of the lecture I have to re-record, actually, right? So that's going to be annoying. But we talked in general about cellular and human reproduction, the beginning of reproduction, right? So chapter 3 is embryogenesis, reproduction, and development. Right? Embryogenesis, reproduction, and development. What does genesis mean? creation, right? Like formation, right? So
Oh man, that's why. It's like, why isn't it working? Not even taking its hair back from the room. Hold on, guys. I need to fix this. Oh, jeez. No wonder, huh? Wasn't really working. What? Jeez. It's hot. I'll be back. I need to get some duct tape. Oh, there goes China. Put China back up. Oh, China. All right, let me get some more ice and we'll start again. We're what? We're nearly 1/3 there. Look at that. I'm back.
Embryogenesis is the creation of an embryo, right? Where do you think embryogenesis begins? How does embryogenesis start? Remember?
In the fallopian tube.
The end of the electron menstrual cycle, we have two ways it could go. So either you get blank and implantation occurs, or you don't get blank and you get your period, right? What is the blank? Fertilization, right? So now we're going to talk about what happens when fertilization does occur. Does anyone remember where I specifically said fertilization normally occurs?
Fallopian tube.
In the fallopian tubes. In the fallopian tube. So the ovum, right, the egg cell that is released when we get the LH spike, blah, 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 travels through the fallopian tube, and a sperm, if injected into the woman, right, if the sperm travels through the vaginal canal, through the cervix, through the uterus, right, it can reach the fallopian tube and fertilize the egg, right? Is there LH spike, blah, 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 travels through the fallopian tube, and a sperm, if injected into the woman, right, if the sperm travels through the vaginal canal, through the cervix, through the uterus, right, it can reach the fallopian tube and fertilize the egg, right? Is there one sperm trying to do this? No, there's thousands, right? There's thousands. Because every time a man ejaculates, there's hundreds of thousands, maybe millions of sperm cells that get released, right? And those sperm cells will swim inside of the women's reproductive system until it gets to the egg, right? All right, cool. So what do I have in my notes over here? The secondary oocyte is ovulated from the follicle on day 14 of the menstrual cycle, right? Very specific fact, right? Fertilization usually occurs in the widest part of the fallopian tube called the ampulla, right? So if we have a little crude drawing right here, here's the top of the uterus, right? Usually occurs in the widest part of the ovulated from the follicle on day 14 of the menstrual cycle, right? Very specific fact, right? Fertilization usually occurs in the widest part of the fallopian tube called the ampulla, right? So if we have a little crude drawing right here, here's the top of the uterus, right? And there's the part that we usually implant inside of. And then we look at the fallopian tube. Normally, fertilization will occur in the widest part of the fallopian tube, right? Known as the ampulla. The ampulla, right? Okay. Inside of sperm, right? Inside of sperm, we have this little tip. And the sperm tip is full of acrosomal enzymes. Acrosomal enzymes. What are enzymes? They're just little special proteins, right? Acrosomal enzymes basically function to penetrate the egg. You have an egg cell, acrosomal enzymes basically eat through that little, like, layer, and then everything goes into the egg. The genetic material that's inside the sperm can get into the egg, right? Okay, cool. So inside of the book, if you guys have the book or if you're looking at a PDF, you will find this diagram. You'll find this diagram of something known as the cortical reaction, right? Something known as the cortical reaction. You'll find that diagram, right? Okay, what is the cortical reaction? The cortical reaction basically...
Hold on, buddy. I don't really have the same book, but there's a book my friend gave me. Seriously? Chapter 13. Oh, it doesn't even have it here. Oh. What? Oh, it sort of does.
happens so that no more fertilization can take place, correct? Because think about it. Think about what we have going on over here. We have a sperm, right? And it's 23 chromosomes. And we have an egg cell, and it is 23 chromosomes. When the sperm penetrates the egg and puts its genetic material into the egg, we now have a cell that has 46 chromosomes, 2 of every type, a pair of every type, right? If the egg did not undergo the cortical reaction and does not shut down its wall to any more fertilization, what's going to happen? It's going to go from 46 to what? 69, right? And it could go from 69 to what? To 23 more than that, right? And that's 69. Cortical reaction, free type. And it's 23 chromosomes. And we have an egg cell, and it is 23 chromosomes. When the sperm penetrates the egg and puts its genetic material into the egg, we now have a cell that has 46 chromosomes, 2 of every type, a pair of every type, right?
Yeah.
If the egg did not undergo the cortical reaction and does not shut down its wall to any more fertilization, what's going to happen? It's going to go from 46 to what? 69, right? And it could go from 69 to what? To 23 more than that, right? And that's not, in medical school, we use a very grim term. It's not, what is it? More than that. Down its wall to any more fertilization, a pair of every type, right? If the egg did not undergo the cortical reaction and does not shut down its wall to any more fertilization, what's going to happen? It's going to go from 46 to what? 69, right? And it could go from 69 to what? To 23 more than that, right? And that's not, in medical school, we use a very grim term. It's not, what is it? Viable with life. It's not viable with life, meaning that it's not going to grow. It's not going to develop. It's not going to live, right? So if you have 69 chromosomes, you're not an embryo. You are just a cell with 69 chromosomes. So unless the egg were to shut down its walls to any further fertilization, that might happen, right? So the cortical reaction is a way of the egg cell stopping that whole process, right? And that's what you have to know about that. Anyone here know twins? Any of you guys know a pair of twins? Yeah? Do they look the same? No? Do yours look the same? No? Anyone else? They look the same, right? Why? Why do the twins you know look the same, but the twins that you know and you know don't look the same? How does that happen? Well, what determines our looks? What determines our looks? You see someone who is like quite attractive. You walk up to them, oh man, you got good genes, right? You got good genes. Then you're kind of right. What determines our looks? It's our genetics, right? It's our genetics. I have brown skin because of my genetics. I have brown eyes because of my genetics. I'm this tall because of my genetics. It's coded into my body, right? So if twins look the same, that must mean that their genes are the same. How does that happen? Well, these are called what? These are called identical twins. Identical twins, right? And identical twins, at some point in the development, this either cell or ball of cells or whatever it was, split in half. And one half went one way and one half went another way, and they developed into 2 different embryos, fetuses, whatever it may be at that time, right? That's what happened. That they came from the same fertilized egg. That identical twins came from the same fertilized egg. These are called monozygotic twins. Identical is a colloquial term. These are called monozygotic twins. What does mono mean? Not the disease you get in high school. What does mono mean? 1, right? So a single zygote splits into 2, and this has a higher likelihood of resulting in conjoined twins, which is a very big problem. It's hard to fix, right? So if this is monozygotic twins, what's the other one? Dizygotic, right? Dizygotic. Dizygotic twins. 2 egg cells, 2 sperm, right? Now, here's the question. Bless you. That egg cell, right? Like egg cell A and egg cell B, right? Came from the same person with the same parents. And like, let's say it was my mom. Let's say that I had a twin, a dizygotic twin who looked nothing like me. Those 2 egg cells came from the same person with the same parents, same genes. Every single cell in her body has the same genetic information. How the fuck? Like, why do they look so different? Why do all of my parents' children look so different? Remember, way back in the beginning when I asked you, what's the event that accounts for genetic variability? Crossing over. Because there is a different combination of my grandparents' genes from my mom and my grandparents' genes from my dad in the egg cell and the sperm cell that made me, that made my hypothetical twin. I don't have a twin. I'm the youngest. I have 2 older sisters, right? I'm spoiled out of my mind, right? Only son, youngest, 2 older sisters, right? And I'm brown. All right. You know my mom loves me. All right, so that is what we're talking about. That's why I asked in the beginning, right, about the whole crossing over thing. So we're talking about twins. You're like, wait, but that doesn't really make sense. But you know what makes even less sense? Why do identical twins not look exactly identical? Why do identical twins not look perfectly identical? They have the same genes, keyword, different one. Different, not necessarily same genes means same alleles, right? Different expression. Your gene expression is completely contingent upon your environment, right? The environment around you will influence your gene expression to a degree that is different than every single other person, right? The amount of time I spend in the sun, the amount of carcinogens that I inhale by living in New York City, right? The number of fucking chicken cutlets that I've eaten in my life will probably have some sort of effect on my gene expression, right? So when we talk about monozygotic twins who are exactly the same in their genetics, they're still not going to be perfectly the same, right? They're not going to be perfectly the same. Do I make myself clear? Cool? That's all you got to know about twins. So to recap, for the people who just walked in, monozygotic twins, also known as identical twins, are twins that come from the same fertilized egg cell. And dizygotic twins, also known as fraternal twins, who don't look exactly the same, come from two different fertilized egg cells. Why are they called fraternal twins? Because fraternal means like brotherhood, right? And a good way to remember that is that the identical twins, right, they both came from mom, right? But the fraternal twins, the brotherhood twins, are two sperm cells had to come from dad. The dad had a bit more work to do for fraternal twins. Make sense? Remember, the terms are monozygotic and dizygotic. Mono meaning one, di meaning two, right? Cool. What's a zygote? I'm throwing that term around. What's a zygote? A fertilized egg, right? The moment an egg gets fertilized, it's a zygote, right? So, to recap the menstrual cycle from last time, I'm going to go a bit further back. So, we have the follicle, followed by the LH spike, and the follicle ruptures. So we get the ruptured follicle, which is also known as the what? It becomes the corpus luteum, right? The ruptured follicle, under the influence of luteinizing hormone, becomes the corpus luteum. Once it's the corpus luteum, this begins spitting out what? Progesterone, right? In response to LH, right? It begins spitting out progesterone. Progesterone is used to maintain the uterine lining, correct? Now, the ruptured follicle also comes with an egg cell, and that egg cell will travel into the ampulla of the fallopian tube and get fertilized. And this is now called a zygote. That's called a zygote, okay? That is a single cell, a single 2N cell, okay? The ampulla, A-M-P-U-L-L-A, the widest part of the fallopian tube, known as the ampulla. By the way, for people who are new here, I'm Yusuf. Nice to meet you guys. If you guys get a rather difficult question right in my class, I will throw you a dollar answer. So, let the games begin. All right. A zygote undergoes rapid cell division in order to turn into a functional growing unit known as a embryo/fetus, right? And that division is known as what? Very funny word for middle schoolers to say. Cleavage. Cleavage. And the moment it undergoes its first division through cleavage, it is no longer known as a zygote. What's it known as? It's known as an embryo. So, in order of vocabulary, unfertilized equals egg or ovum. Fertilized, one cell equals zygote. Greater than one cell equals embryo. That's why I call it embryology, right? The book makes a note about indeterminate and determinate cleavage. I don't think it's so important, but indeterminate cleavage results in cells that can still develop into complete organisms. So basically, pluripotent cells that can become anything they want, and determinate cleavage results in cells that are determined or committed to differentiating from one specific thing. Right? That makes sense? If that did it, don't worry about it. Okay. The embryo will eventually become a solid mass of cells. That is known as a morula. All right, so mass of cells, morula. We're going to keep our vocab separate over here. I know it's quite small, but I'm trying to write bigger over here. So the mass of cells is known as a morula. The mass is a morula, right? Now, this all seems very good. Out of order. Yeah. This all seems very fast, right? So look, we got fertilization. That makes the zygote. The zygote undergoes division. The zygote becomes an embryo. The embryo becomes a solid mass of cells. The embryo is now a morula, right? It's going to keep going. Because this is just like, this is the big bang right here. We're like femtoseconds of shit happens between now and then. It becomes the biggest thing in the world, right? So what happens next? The morula becomes what? This is the confusing part. It undergoes a process known as blastulation. Guys, if this makes no sense, trust me, it doesn't make sense to me either. It's just in the book. Undergoes a process of blastulation, right? It becomes a blastula. That's what's called blastulation. What is a blastula? It's a ball of cells, right? It's a ball of cells with a little cell mass at the top. So these are all cells over here. And a lot of empty space. Okay? Right? And that empty space is called a blastocele. C-E-L-E. Blastocele, right? So, what's the blastula? It's a hollow ball of cells. Okay? Keep the vocab somewhere else, so you can look back at it later. If you need to read anything, please let me know. The blastula moves through the rest of the fallopian tubes, and this is what gets implanted. Blastula, And then I'm just going to go implantation. Implantation, right? Okay. They talk about the amnion and the chorion and trophoblast and blah, blah, blah. Amnion, chorion is above your level of knowledge. It's for me. I need to know what those are. You don't, okay? That's going to be a step 1 question, not an MCAT question. But what you need to know is that the amnion and chorion are basically just supporting structures of the embryo, right? Now, all of this is still called embryo, right? These are all different phases of the embryonic growth. These are all still technically called embryos. Remember, anything greater than 1 cell is an embryo. I wrote that specifically, right? These are all still part of embryo. These are all just different kinds of the embryo, different phases that move through, right? Okay. Has anyone heard the word amnion before? There you have. Amniotic fluid. Right? What is amniotic fluid? Amniotic fluid is just the fluid that you float around in when you're about to prepare to be born, right? Okay, cool. Oh my god. Okay. This is one of my least favorite things to ever learn / teach in the world. I hate it. I think it is so baseless, and I never understood why I have to learn it until I got to medical school. So it's not going to make any sense to you yet, but it will make sense in your medical school. The next process, so I'm just going to leave this here, implantation, right? It's called gastrulation. So blastulation made a blastula. Gastrulation is going to make a gastrula, right? This creates a gastrula. What is a gastrula? So what happens is that the blastular membrane invaginates, right? What does invagination mean? It means to come into oneself. So my hands like this and they invaginate, right? And that invagination, right, brings the cell mass in towards the other side of the blastula. So what happens that invaginates here, it creates a little C, right? And this circle I drew, those are all cells, right? So then what it looks like after this, it kind of looks like this. Where those are all cells everywhere, right? Cell. And they look like this, right? That's the gastrula. And you can find the diagram in your book, of course, right? Okay? And what happens here is that inside of this, there are 3 distinct layers. Who can tell me the name of those 3 layers inside of the embryo?
Endoderm,
Anyone?
mesoderm, ectoderm.
Very good. Do you catch? Close. All right. The endoderm, the ectoderm, and the mesoderm. All right. This is when we start using our fancy colors, right? And I put them in red because I fucking hate learning about all this. All right. So we have the endoderm, the mesoderm, and the ectoderm. You know what's so funny? I will literally never get monetized on YouTube because of the amount of time that I spend cursing in my videos. I don't give a fuck. Okay. Cool. Are you guys following me here? This is much better learned through diagrams. So this is where we're going to leave it now, and we're going to talk about these 3 things right here. Does everyone have this down? If you don't, take a photo. Let's go. And we're moving. We're moving quick. Take a photo, take a photo. I'm going to get my caffeine fix. I'm just lighting her up. She's been laughing at all my jokes. You know my mom the other day? So I was at home. Because my mom got getting surgery soon. She has a tear in her shoulder. So I went to go visit her because I'm not going to be able to come like the day of her surgery. She didn't want me there either. She's just like, oh, like stay in Brooklyn. Like it's going to be overwhelming for me, blah, blah, blah. All right, mom, I'll see you the next day, right? So you know, everything goes good, right? But I was with my mom, and she was just like, oh, like, how's your program going? I was like, oh, it's great. She's like, can I watch your lectures? Like, no, you cannot. You can't watch my lectures. Oh, no. I had like one day, you guys have to remind me to like not curse for like 15, 20 minutes of lectures. I was like, guys, look, I'm such a good teacher. All right, so these 3 things are known as the primary germ layers, right? The primary germ layers, don't worry, I'll rewrite them right here. So the primary germ layers. Number 1 is the endoderm. Why did I look over that? I know these two. Okay. This develops into something. It is the epithelial lining of digestive and respiratory tracts. Someone smarter than me in cell biology, tell me what epithelial means. Louder. Kind of. It's the epithelial lining is like the functional lining, right? The epithelium of something. It's like the tissue that, if you're talking about lung epithelium or liver epithelium or something, it's like a functional tissue of that organ, right? It's like the functional lining of the digestive system, the stuff that secretes all the digestive enzymes, blah, blah, whatever it is, right? I don't have like a solid grasp on what epithelium means because we use it for everything. It's a very vague term, right? Also included in this are the pancreas, the thyroid, the bladder, and else, well, the end of it, right? See, I'm trying to write big. E. Nervous system. The ectoderm also produces some stuff in the nose and also produces stuff in the ear. And you're just like, why the hell? Very next. The epidermis, the hair, the nails, right? The epithelia of the nose. And one more very important thing. The nervous system. This is what you'll be asked about most often. The nervous system. The ectoderm is contribution to the nervous system, correct? As well as the lens of the eye and the inner ear canal. You'll realize when you get to medical school, there's a part of the development of the brain that also produces some stuff in the nose and also produces some stuff in the ear. And you're just like, why the hell? And then you realize it's all from here, right? Cool. Really, really specific thing that they for some reason ask about. Before I get there, I'm going to teach you guys more language stuff. You guys know about epinephrine, right? Epinephrine. And you guys know about adrenaline, right? What are these? Yeah, right? So this one's for allergies and this one's for fight or flight, right? They're the same thing. They are the same thing. Epinephrine is adrenaline. Adrenaline is epinephrine. Okay? Why? Epi in Greek means above. Nephros in Greek means kidney. Above the kidney. And that INE is a hormone or a molecule. The molecule is released from above the kidney. Ad in Latin means next to or near. Renal in Latin means denoting the kidney. Adrenalin is a molecule produced next to the kidney. Epinephrine and adrenaline are talking about the same thing in two different languages. That's why norepinephrine and noradrenaline are also the same thing. Does that make sense? Okay? However, the gland is not called the epinephral gland. It is called the adrenal gland. When you get to medical school and you hold the kidney in your hand, you'll see this soft, squishy, fat-surrounded thing on top of the kidney. That is the adrenal gland. And it is wildly important to your survival. Okay? You good? Very cool. Okay, so why did I bring that up? Because for some reason, the MCAT book makes a very specific distinction about the differentiation the difference in development of the adrenal cortex and the adrenal medulla. The adrenal cortex is derived in mesoderm, whereas the adrenal medulla, or medulla, is derived in ectoderm. Does that make sense? The adrenal gland is organized like this. Here's the kidney, right? Here's the little blob, right? You have to eat for enough arterioles, blah, blah, whatever. And on top of the kidney is the adrenal gland. And I'm going to zoom in. And the adrenal gland kind of looks like this. Here's the top, here's the bottom. And you have a cortex and a medulla. And the cortex and a medulla, right? This is part of the adrenal gland right here. This is the medulla. And the cortex is split up into 3 layers. 1, 2, 3 of the cortex and then the medulla. They're saying that this and this are derived in 2 different derm layers. Mesoderm, ectoderm. And that's because they secrete different things. Norepinephrine and epinephrine come from the ectoderm. A lot of other hormones come from the adrenal medulla, the ectoderm. A lot of other hormones come from the adrenal cortex, which is derived of mesoderm. I don't know why they make that specific differentiation, but they do. Understood? Cool. You need to know that? No. But if you wanted to, those 3 layers inside the adrenal cortex are known as the zona glomerulosa, the zona fasciculata, and then the zona reticularis. And those 3 zones will produce 3 different things. It's very difficult for me to reach back in my memory and remember that, because that was a couple months ago. And I'm not learning kidneys anymore until 5 weeks from now. Okay. What's differentiation? We tell you differentiation in a very general sense. It's when cells develop into different cell types, right? That's differentiation, right? So what happens is that every single cell, like if I took a cell out of my muscle in my leg, and I took a cell out of my skin, I took a cell out of my nose hair, and I took a cell out of my eyeball, right? And I karyotyped all and I ran the genes. Remember what we said in the first thing? They would all have the same genes. How did differentiation occur? Selective transcription. Selective transcription, right? Selective transcription leads to differentiation. Okay. Selective transcription leads to differentiation. Welcome, welcome. How are you guys? Good. Take a seat. Okay. Okay. Guys, I'll always tell you when I'm incapable of doing something, right? That's why I have other people who are going to come in here later and teach the stuff that I can't teach. Contrary to popular belief, I do not know everything. Okay. I cannot properly teach you neurulation. This is neurulation. It is when the ectoderm becomes this thing called the, I don't even remember what it's called. It's called the neural tube, and the neural tube defects happen, and the neural tube grows out into the spinal cord ball. It's like this crazy thing. Go watch a video. I'm being so dead ass. I cannot teach this stuff. I'm so bad at it. So what I want you to do, even if you're at home, right? I want you to look up on YouTube, neurulation, embryology, basics. Neurulation, embryology, basics. But to run you through it.
Yeah, I might still have to watch that.
Very, very simply, the nervous system is, of course, derived of ectoderm, right? And what do we say ectoderm was? Right in the gastrula, we have a little gastrula, we have 3 cell layers, right? And the endoderm is the most inside, and the ectoderm is the most outside, and the mesoderm is the middle, like Sasha said, right? Okay, so basically what happens is that the ectoderm, like it invaginates, and then this little cell mass comes up out of it, and then you fold over it, and that cell mass becomes the neural groove, and the folding of the neural folds, and those all become different parts of the spinal cord. And as you can tell, I have no fucking idea what I'm talking about, right? I don't know what's going on there. I'm not good at it. Hopefully, when I learn neurology in a couple months, I'll know a little better, and I'll come back to you, and maybe we'll re-explain this, right? But for now, we're gonna move on. Because I am not going to sit here and lecture you on stuff that I don't know. I'm much more comfortable telling you that I don't know. Why? Because I'm 23, not 45. Okay? And if one of you guys does learn, and you feel like you do understand it to a degree that you can teach me, please do. That's a genuine question. If any of you guys know it well enough, please do teach me. Okay, let's talk about problems in early development. Okay, what do you call a substance that can cause a problem in a fetus or an embryo? What do you call them? So problem, like molecules that cause cancer are called carcinogens. What do you call teratogens? Teratogens. Right, so teratogens are substances that interfere with embryonic development. That was so funny, like a new person walked in and then heard me explain that I don't know jack shit and just walked out. At least you guys know that I know something, right? Okay, cool. Alcohol, drugs, prescription drugs, warfarin, heparin, like these things, environmental chemicals, other carcinogens, et cetera, right? Even certain infections. So like in medical school, you're going to learn about these things called torch infections. And those torch infections are specific infections that you can pass on to your baby. Toxoplasmosis, rubella, CMV, like these infections that can be passed on to the child, and they cause horrible, horrible birth defects, like really, really bad ones, right? As you just learned earlier this week about a heart defect that happens in children, it's called transition to the great arteries, is where your aorta and your pulmonic artery are swapped. Like they just swap places, and somehow maternal diabetes causes that. It's very strange, right? Other stuff, maternal health, diabetic mother, and the biggest thing, folic acid deficiency. Folic acid or folate, for my women out there, whenever you are, if you plan to get pregnant, if you want to have children, right? If you go see a perinatal doctor, they will put you on a folate supplement, right? Folate is vitamin B12. And it is wildly important to neural development. If you don't take enough folate, your child will most likely be born with a nervous system issue. You guys ever heard of spina bifida? Spina bifida can be caused by folate deficiency in the mother. All right, speaking of vitamins, all the B vitamins, which you need to learn later, have different names, right? B1 is not just B1, it's also called thymine. B2 is riboflavin. B3 is niacin. You don't need to write this down, we're going to go over it later. B5 is pentanoic acid. B6 is pyroxidine. B9 is, I completely forget B9 like all the time, and B12 is folate. B9 is really important though. I don't know why I'm forgetting. I'll probably remember in a couple minutes. Okay, so let's talk about mechanisms of developing. So let's look at the time real quick. 7, cool. We started a bit late because I'm locked out of the lecture hall, which sucks, right? But if you guys are here the whole time, we're here till 9. Right. So let's talk about mechanisms of development. Simon, how are you? Cool. We're going to talk about 2 things. Number 1 being specification. Let's talk about 1A and 1B. 1A is specification. It's when a cell that is capable of doing whatever the fuck it wants reversibly designates itself to one job. So cell reversibly, right? It reversibly designates to one cell type. A pancreatic cell, a liver cell, a tongue cell, a brain cell, right? Determination is then what? It irreversibly commits to that cell type. So it's that, but irreversible. And what's so irreversible about it, right? It's like something within the genetic code. It's like a specific mRNA transcript or signal, blah, blah, whatever. It has to become that specific cell now, right? And the second thing is differentiation. Differentiation is when the cell changes structure, function, and its actual internal chemistry to become that cell type. Structure, function, chemistry. Okay? Cell changes structure, function, and chemistry. What's up, man? So basically, you bring up a really good point, which I brought in the book, right? So I would say you know someone's listening when they ask the question that you're going to see next. What's up? Huh? Oh, yeah. There you go. See, he asked. I mean, all right. So, yeah, basically what happens is that in the embryo, there are stem cells, right? And those stem cells are what we call pluripotent. They can become anything, right? Once specification happens, or determination happens, they're like multipotent. They become a bunch of things within that specific cell type, right? But most of our cells
Totipotent, right?
are unipotent. They can only be that specific thing. So potent meaning the potential. The potential of the cell to become what it wants, right? All right. So one thing I want you to take away from this: an increase in differentiation equals a decrease in potency. Anyone here ever heard of Henrietta Lacks? Right? Where we took stem cells from a person who didn't like agree to them, and now we use those stem cells to research and save millions upon millions of lives. Anyone here ever heard of Henrietta Lacks? Right? Where we took stem cells from a person who didn't like agree to them, and now we use those stem cells to research and save millions upon millions of lives. Her family was never like compensated for that. If you haven't heard about it, go research it. It's very interesting, right? But we use undetermined, unspecified cells to grow anything we want out of them by using those sort of transcription factors and signals and blah, blah, blah to turn them into specific cell types. So we can grow pancreatic cells, we can grow blood cells, we can grow tongue cells and brain cells and blah, blah, blah. We can do it from undifferentiated cells. Does that make sense? Yeah? My dad like heard me teaching one time, and obviously I started like really tutoring when I was locked up in my basement during COVID, right? So I was studying for the MCAT, I was tutoring and eating and sleeping, and that's pretty much all I did. And my dad would always hear me say, like, does that make sense? Right? Does that make sense? Does that make sense? And he like, for some reason, he like comes up to me and is just like, do you say that like every single time you explain something? I was like, I don't know. So ever since then, I've been noticing it. And I think I do say it like every time I wrap up a topic, I say, does that make sense? And you guys are free to say no. Yeah, does that make sense? Like just say it like every 20 seconds. Okay, recently I've been starting to say like, do I make myself clear? Right? That sounds like scary almost, right? Okay. They talk a little bit about cell-to-cell communications, right? So we're going to skip over like the inducer stuff because it's not super high yield, right? We're going to talk about the types of cell-to-cell communication. Also, for anyone who wasn't here before, right? Or wasn't here by previous lectures, whenever I say that something's not super high yield, it means in like the 6,000 MCAT questions I've done, it just hasn't come up all that often. So rumor has it that it won't come up again later, right? That's just like the theory in my head. And 9 out of 10 times I've been right. Okay, cool. All right, so let's talk about types of cell-to-cell communication. Types of cell-to-cell communication. There's 4 types. Autocrine, juxtacrine, paracrine. What's the last one? Endocrine. So I'm gonna nerf you, man. You? Which one are you? Oh, there you go. So I'm gonna nerf you too. You guys are smart. All right. Autocrine, juxtacrine, paracrine, and endocrine. Right? Autocrine looks like this. The signal acts on the cell it came from. Juxtacrine looks like this. Immediately next to it. What we call the adjacent cell. Paracrine looks like this. Local cells. And we have a whole system named after the last one. Endocrine acts on far away cells.
Good.
Good.
Let me just take the outcome real quick.
Who can name me an endocrine organ?
Looking for that. The pituitary gland, hypothalamus.
The brain, yeah, like hypothalamus, stuff like that, right? The pancreas, insulin, glucagon, right? The thyroid, T3, T4, right? Parathyroid, calcitonin, right? Stuff like that, right? The ovaries, the testes, estrogen, progesterone, testosterone, and the list goes on. We have a whole chapter on endocrinology, and I sure hope you get to see me give that lecture. What's up? Cool. I know we're moving fast. We just have to get through a lot of information. All right. We're moving. Yes, following along? Okay. We talked a little bit about cell death before. We're not going to touch on it again. What are the two types of cell death you're responsible for? Apoptosis and necrosis. What's the difference? Apoptosis is controlled, organized, right? It is deliberate. And it's controlled by what? What in the cell has control over apoptosis? The mitochondria, right? I was like mitosis, nothing about mitochondria, right? What about necrosis? Necrosis is just like whenever the cell gets shot in the back of the fucking head, it just dies, right? So like deoxygenation can cause necrosis, like some poisons can cause necrosis, stuff like that, right? So when you have a heart attack, right? Your coronary artery is blocked, you get a loss of blood flow, immediate lack of oxygen in the myocardial tissue. What's gonna happen is you get ischemia. That ischemia leads to necrosis, right? You get necrosis of that area of the heart, it no longer pumps, it no longer functions. That's why heart attack is a big thing, right? And you get this thing called contraction and necrosis, right? But it's not apoptosis because the heart didn't kill itself. Whenever you have apoptosis, that's the cell killing itself, right? Okay. So a little bit about regeneration, right? What's the regenerative capacity of most tissues in the body? Quite low in general, right? But who can name me some tissues that are very good at regenerating? There's one really big one. Sorry? That's different. So those are a little different, but organ tissues. A little different. An organ specifically, it's really good at regenerating. The liver, who said it? The liver is fantastic at regenerating. Okay. Senescence and aging, right? We're not going to talk about aging on a human level because I don't want to, because it scares me. Not really, but maybe I'm doing, right? What we are going to do is talk about aging on a cellular level. In your chromosomes, greatest chromosome I've ever drawn in my life, right? At the ends of the chromosomes, you have these things, right? You have these things. What are these things? Yeah, telomeres, right? Or telomeres. Telomerase, you ever heard of that? Telomerase, right? Anyway, gotcha. Back row. Okay, so telomeres, right? Every single time that your cells divide, the telomerase has to copy over those telomeres, right? Because think about it. When you read DNA, let's say it's DNA, and you're reading, you gotta stop. You gotta stop synthesis of DNA somewhere before the end, because you can't just stop when there's nothing in front of you to catch on, right? So imagine that it needs to cut somewhere. Right? It reads, cuts. But if there's nothing at the end of the DNA to cut, you'd just be shortening the DNA every single time you do a replication, right? So what happens is that when we read the DNA, we add on this little bit of nonsense, right? It's like when you haven't hit your word count on an essay yet. You just start going to the white font and start typing nonsense, right? So it's the nonsense. So what happens is that we read the entire transcript of DNA and we cut off a little bit of nonsense, right? But you can imagine, after many cell divisions, that nonsense starts to disappear. And now you are cutting away part of the DNA. And it's possible that you cut off a functional part of the DNA. And that's when that cell dies. So after enough replications, the cell will naturally die. Does that make sense? Cool. So my telomeres are shorter than they were 23 years ago. By how much? I don't want to know. All right. This is most possibly the stupidest organization of a chapter I've ever seen in a textbook. Because we just talked about embryology, blah, blah, blah. But you guys know how we haven't covered the circulatory system yet? They throw fetal circulation into the chapter, even though you learned nothing about cardiology and nothing about blood and nothing about circulation. But thankfully, you have me. So what we're going to do is everyone's going to put all of their shit down. Close your laptops, flip your books, don't touch them. Everyone's going to listen to me talk for 10 minutes. And we're going to retake these notes in the lecture on cardiology. But you're going to listen to me talk now and try to understand, okay? Because if you were to take notes while I'm talking, we wouldn't get through the information we have to get through today. Because Kaplan's fucking stupid. Fuck you, Kaplan.
Oh man, that's funny.
I love their books. Their information's fantastic. Whoever designed the textbook deserves a noose around their neck. Okay, we're talking about fetal circulation. Now, you guys know that a baby can't breathe on its own, right? And since it can't breathe on its own, it can't oxygenate its tissues on its own. And since it can't oxygenate on its tissues on its own, it's fucking stupid. It can't do anything, right? Okay, so since the baby's stupid, it needs its own method of getting blood into its body. The fetus is a parasite, okay? It is connected to the mother via an umbilical cord. And inside of that cord, there is an artery and there's a vein. The artery carries blood towards or away. Away. And the vein carries blood towards. So this is one of the only examples where an artery has deoxygenated blood and a vein has oxygenated blood. Who can tell me the other example? Dude, shut the fuck up. Drive me crazy. Why do you know my answers? And you caught it. All right. Yeah. There you go. No, good. You know, you're actually doing fine. Okay. So the umbilical arteries carry blood away from the fetus and towards the placenta, right? The placenta is the interface between the mother and the child, right? Don't eat your placenta, by the way. I don't know who the fuck started the whole trend of eating the don't. That's disgusting. Okay. We're going to go over very, very basic cardioanatomy. Because without understanding cardioanatomy, you won't understand fetal anatomy because it's different, right? And why the fuck would you teach the different thing before you teach the normal? This is your heart. Hopefully not, but this is your heart. It's split into 4 chambers, and it goes in alphabetical order. It goes from A to V. It goes from atrium to ventricle. Now, you must think I'm stupid because right is on the left and left is on the right. I'm not stupid. For some reason, someone let me in medical school, right? You're looking at the patient. You're looking at me. My right is on this side, my left is on this side. You're looking at the heart. What is this? That's plugging into the right atrium. This is the vena cava. It is the main vein in the body. All other veins drain into the vena cava, okay? You have the superior and inferior vena cava. The vena cava will put blood through the tricuspid valve into the right ventricle. The right ventricle will pump out of the, so we're going to follow this arrow up, and pump out the pulmonary artery, which is the other deoxygenated artery. The pulmonary artery carries blood towards the lungs, and the lungs bring it back to the left atrium through the pulmonary veins. Oxygenated vein. That's weird. That's different. The left atrium goes through the mitral valve, or the bicuspid valve, into the left ventricle, and the left ventricle pumps out the aorta, and to the rest of the body. And the body eventually cycles it back to the vena cava. Very, very, very much simplified. Anyone who's ever taken anatomy will tell you that this is the simplest way I can possibly describe this, right? Because the aorta is not there, it's up here, obviously. Okay. There are 3 main differences inside of the fetus, okay? So what we're going to do is I'm going to take you step by step through each thing. Okay, we go, let's say vena cava, right atrium, right ventricle, pulmonary artery, lungs. Lungs goes to the left atrium, to the left ventricle, to the aorta, to the arteries, to the arterioles, to the capillaries, to the veins, and back into the vena cava. Here you have the tricuspid valve, here you have the mitral valve, here you have the aortic valve, I'm not going to write AD because that means something else, and here you have the pulmonic valve. Okay, there are valves everywhere. Okay, now, Jolly answer question. How does the blood in the veins not just pull back down toward the legs? Not you. No. Not necessarily those that's in the heart. No. No. In the veins, there are these things. There's one-way valves. They don't open or close, but what happens is that the blood flows through them, and when it falls back down, it closes the valve on its way back. So it can't go backwards. Right? Okay. Now, what I want you to know about the fetus is that A, it's stupid, right? And B, its lungs don't work. Its lungs do not work. So what we need to do is circumvent the lungs. We need to make sure that no blood goes to the lungs, right? How do we make that happen? What's the first thing we have to do? We have to make sure that blood doesn't go here. So we need to somehow knock out the pulmonary artery, right? We need to take it and knock it out. So the first thing, and this is when you can start taking notes. The first thing we do is called the ductus arteriosus. The ductus arteriosus, or the DA. The ductus arteriosus is a connection between the pulmonary artery and the aorta. So that blood exiting the right ventricle goes straight into the aorta. And skips the lungs. You see how that happens, right? Okay. So we're going from here straight. We're going from here straight to here. We skipped the whole left side of the heart. We never went to the lungs. We never even tried to get oxygenated. We just skipped right over. I know it was really quick and it's very basic, and we're going to go over it again when we go over cardiology, but you guys understand what I'm saying, at the least. Okay?
Okay.
It's not my fault, I promise.
Okay, but then how does that?
We need another way to make sure that, so we don't want blood leaving the pulmonary artery. We just talked about that. But we also don't want blood overriding into the right ventricle. What's the second thing we have, Sarah? In the heart. Fossa ovalis after it closes. This is known as the foramen ovale. What does foramen mean? Hole. Hole, right? The foramen ovale is a hole between the right atrium and the left atrium. Right? So it's a hole right here. And what happens is that blood is allowed to leak into the left atrium and just exit into the left ventricle and out the aorta. So we go from here all the way to there. We skip the lungs, right? And one more thing. If we're skipping the lungs, how does the fetus get oxygen, right? Well, you have to erase some of this, right? The fetus gets oxygen by doing this. So I'm going to say body and over here, right? In the circulation, we get out towards the body, and there are these little umbilical arteries that pick up blood. Umbilical arteries. And I'm going to shorten arteries to AA. That's how we say arteries. A period would be artery, AA is arteries, right? So either the body goes towards the vena cava or it goes out towards the umbilical arteries. The umbilical arteries take the blood to the placenta, where the placenta injects oxygen into them, right? And then it goes back through the umbilical veins. And the umbilical vein plugs into the fetus's body through a third structure known as the ductus. If one was the arteriosus, the other one is the ductus venosus. It's where the umbilical artery, or the umbilical vein, sorry, plugs back into inferior vena cava, I believe. But don't quote me on that. So I'm just going to say venous system of the fetus. The ductus venosus. And all three of these things close after birth because of increased oxygen pressure and left atrial pressure and everything closes up, right? I see a lot of confused faces. I know. We're going to go over this one more time. Yeah, so if the umbilical vein plugs back into the venous system of the fetus. Okay. You guys ready? I'm going to erase that. I'm just going to show you what we're talking about. All right. You're going to physically see how we skip the lungs. Pens down, everyone on the board. The vena cava, right atrium, right ventricle, pulmonary artery, lungs. Left atrium, left ventricle, aorta, body. And actually I'm going to say arteries, arterioles, capillaries, veins, vena cava. Okay? We're trying to avoid the top right corner. We want to get away from here. Number one was what? The ductus arteriosus, which takes the pulmonary artery and plugs it into the aorta. That's pathway number one that we use to avoid that right corner. So I'm actually going to do that in different colors so you can see the properly. Pathway number one, the blood leaks through here. Okay? And this is deoxygenated blood because it came back to the heart, right? Well, it's a mix of deoxygenated and oxygenated blood, and we'll talk about that in a second. Number two, what else did we do? We made the foramen ovale. The foramen ovale is a septum, it's a septal hole, right? In the septum of the heart. So it connects the right atrium and the left atrium. Look, we skip over the corner. And this is the ductus arteriosus. This is the foramen ovale, right? And the last thing is how do we get oxygen? When we are inside of the body, somewhere along here, the blood exits through the umbilical artery and goes to the placenta, where it gets oxygenated and plugs back into the veins. So we take oxygenated blood and plug it into the veins that are deoxygenated. So the fetus's entire body is a mix of deoxygenated and oxygenated blood, which is why if you've ever heard, fetal hemoglobin has a higher oxygen affinity than ours does. Is this making sense to you guys? Do you see how we skip over the lungs in each and every single thing that we did? We will re-explain this when we go to cardiology. You just can't leave this chapter without hearing this at least once. Does that make sense? Does anyone want me to repeat anything, or do you have any questions at all because we're leaving? Yeah, take a photo. Take your photos, do your dance, take a shot, right? We will go into great depth about this when we do cardiology. Because there's not much else to know about cardiology other than like how things go. So we're gonna have a little bit of time to talk about this. Okay. When is the embryo known as fetus? At what time? Unanimously, around 8 weeks. 8 weeks. Pop quiz. When is the heart fully developed? When are the 4 chambers of the heart developed? 4 chambers at 4 weeks. Right? They do. So I had like I think the question back your head is how do they close, right? They close. They all close. So no, they just close. They like the foramen ovale. It looks like this. So you have the heart septum, right? The thing that splits. This is the right atrium. This is the right. Left atrium, and this is the septum. The foramen ovale is basically just like there's a hole here, and then the valve that comes like this, right? And what happens is that increased left atrial pressure at birth forces the valve against the septum, and it closes. The ductus arteriosus, there's a decrease in prostaglandin synthesis, which causes it to kind of just like close onto itself. And the coolest thing is, remember how it was the connection between the aorta and the pulmonic artery? The ductus arteriosus becomes the ligamentous arteriosus, which is the only ligament that anchors the aorta to the pulmonic artery. Very cool, right? And then with the ductus venosus, venous pressure just slows that as well. So it will be in the little link like that I sent out in my thing. Come see me after, or if you have to leave early, I'll pull up the QR code for you. There's a QR code you can scan. You'll get on the email list, and it's there every time. Yeah, no problem. All right, guys, that's chapter 3. We're done. Okay, we're gonna stop the video. We're gonna talk about the nervous system and what makes your brain go tick, right? But it's not really only your brain, is it? Right? So has anyone ever here seen Schoolhouse Rock? Like, you know, you guys remember those old Schoolhouse Rock things? They had like the Constitution, that one like adverbs and adjectives. If you haven't seen it, there's a fantastic Schoolhouse Rock video about the nervous system. So I want you guys at home to go look up the nervous system. Schoolhouse Rock. And you're gonna hear this awesome song that goes like, "There's a telegraph line, you got yours, and I got mine. It's called the nervous system." I don't want to spoil it anymore, but it's a great song, and it gives you a very basic rundown of what's included inside the nervous system, right? Now, in the nervous system, obviously, we have the brain. I mean,
Sure.
message is clear. It says there's something bugging you and buzzing in your ear. The results can be quite itchy, so what is your reply? Tell your mom to swat that fly. Hey, there's a telegraph line. You got yours and I got mine. It's called the nervous system, and everybody understands those telegram commands. And you know that everybody better listen. The central nervous system is the brain and the spine. The brain controls the system and the spine is the line. Telegrams come in to tell what's happening to you. Then telegrams go out to tell your body what to do. There's a telegram for you, sir. Better read it on the spot. It says your hand is near a stove that's very, very hot. The results can be quite painful, and there's no time to think. Quick, pull that hand away and get it to the sink. There's a telegram line. You got yours and I got mine. It's called the nervous system, and everybody understands those telegram commands. And you know that everybody better listen. Your peripheral nerves, they go all out, delivering those messages your senses send out. From your hearing and touch to your sight and taste and smell, they let your brain react to all the messages they tell. There's a telegram for you, kid, and it's at an awkward time. It says you've gotta go on stage and you forgot your lines. You're gonna be embarrassed cause this telegram's a rush. Your heart starts beating faster and you blush. Hey, there's a telegraph line. You got yours and I got mine. It's called the nervous system, and everybody understands those telegram commands. And you know that everybody better listen. The autonomic system has a hold of you, controlling automatically some things that you do. Your breathing and your heartbeat just go on naturally. And when you're scared, your nerves rev up the speed. Hey, there's a telegraph line. You got yours and I got mine. It's called the nervous system, and everybody understands those telegram commands. And you know that everybody better listen.
Bravo. Pretty cool. Pretty cool. Pretty cool. All right, we're about halfway. I'm gonna take a small break. What's going on? What's going on? I'm gonna take a small break. I'll come back maybe like 15 minutes or something. Yeah. Or actually, but let me hold on. I have to send an email out, I think. I don't like doing it on Chrome. Chrome. Excuse me. This one. Yeah. It's fine. Shoot, seriously? Maybe I haven't logged in yet. I wanna hold your hand. I wanna hold your hand. Hold your hand. I wanna hold your hand. I don't know. I thought it had sent me something, but maybe they didn't. Oh, I hear. Oh, yeah. Never mind. That's fine. I'm not worried about that. Never mind. Anyways, yeah, though. I'll be back, guys. We'll finish it today, though. Don't worry. We're halfway. Might as well, right? Might as well, right? Clean up here a little bit. the air in here. I have the AC, and it still feels like it's not working. Unless I'm just, I don't know, not feeling the temperature. Yeah. Well, we got a banana. Banana. I hope there's no fluctuations with the light in the video. It kind of gave me a couple headaches. Actually, maybe I need to change. Hold on, guys. I need to just change. Change into something a little bit lighter. All right. All right, I think we're good now.
Right? Those nerves send messages throughout your entire body to regulate when you wake up, when you sleep, when you move, when you do anything, right? The nervous system is necessarily the path of communication between the brain and the rest of the body, right? I'll tell you a really grim story, right? Inside your neck, right? Here's your head, and here's your neck, here's your nose, here's your eye, right? Inside your neck, there are these two little bones, right? And one of them is called C1, and one of them is called C2. And C2 fits into C1 and allows you to turn your head, right? And they're little spinal vertebrae. So the spinal cord moves through them. They're part of the spine. When you hang someone, when someone sends death by being hanged, right? What happens is that their C2 detaches from C1, and their spinal cord gets crushed. And basically, their brain is separated from communication with the rest of their body. And they die. That's how they die. That's how hanging works. This is called an internal decapitation.
That's what you gotta do with the mice. The mice at the research lab. Banana.
So it's not that you're decapitated from the outside, that's what cut your head off, but someone removed the C1 from the C2, and now your head is dislocated from the rest of your body. That will kill you in 99% of instances. There's like one case study I read of an internal decapitation by a car accident where a kid was brought into like a neurosurgeon who like reattached his fucking dens and axis, and like, it was ridiculous. It's crazy, but normally that'll kill you. So that should show you how.
What's the story?
This like there's like one case study I read of an internal decapitation by a car accident where a kid was brought into like a neurosurgeon who like reattached his fucking dens and axis, and like, it was ridiculous. It's crazy, but normally that'll kill you. So that should show you how important the spinal cord is, right? It is the way that we get messages from command central to the rest of the body, right? And when you are in medical school, right, you will have the honor and the absolute privilege of participating in an anatomy lab, right? And in this anatomy lab, one of your labs will most certainly be called a laminectomy. It is where you take the back of the spinal cord and chop through it with a chisel, and you remove the bones, right? And what you get to see is the sheath of the spinal cord, and you'll cut it open and unravel the layers until you can see the spinal cord. It's a beautiful story of mine. which still like kind of makes me emotional to this day. There is a little device in an anatomy, not a device, it's a thing, it's like an instrument in an anatomy lab, and it just looks like a pen. If this thing were thinner and made of metal, the little hook at the back, a very dull hook, that's not a probe. You just use it to move stuff around an anatomy lab. What happens is that you can see the spinal cord, right? It kind of looks like this, and it looks like this, and it has little branches going off at each level, right? And what I did is I took my probe and I snuck it under the spinal cord and I lifted it up. I just lifted the spinal cord gently with my probe, right? And at that moment, I was a 22-year-old first-year medical student sitting in a cold basement of a medical institution, holding someone's fucking spinal cord in my right hand with a stainless steel device. Every single motion, and it was like down here, like their spinal cord down here. So every motion that that person had ever made with their legs, their toes, their muscles, everything they felt on their skin at that level, everything that they ever did willingly and unwillingly, every motion they ever took, every inch they ever moved, I was holding inside my right hand. If that does not display to you how important this structure and what we're about to talk about is, I would reevaluate. Because this is important shit. And one day, some of you may become physicians who work with things as important as this. And by some of you, I mean all of you. Because one day you'll realize that everything in medicine is as important as this. Okay? So let's talk about the nervous system.
Let's do it.
The basic building block of the nervous system
What
is
division?
Neurons, right? Neurons. Okay, these are neurons. What are they? They are cells capable of what? There's like a very specific definition I'll give you here. Transmitting and translating electrical impulses.
So light.
Into chemical signals.
Maybe if we can get through at least endocrine today. If we can finish endocrine, I'll be happy. What looks right?
This is a very crude drawing of what a neuron looks like, right? And honestly, in between here, there are little spaces, right? Ninja Nerd on YouTube has a fantastic video about the nerves and the nervous system, neurons, and everything we're talking about. I urge you to watch his video because his illustrations are much better than mine, right? I don't care for illustrations. He does. Okay. These ends of the nerve, this is a nerve cell, right? So when we talk about like, differentiation, one of the cells inside of that like embryo we talked about prehistorically a long time ago is this. This is a neuron, right? These ends, they're called dendrites. Dendrites, D-E-N-D-R-I-T-E-S, dendrites, right? And they receive messages from other cells. What's up, buddy? How are you? Receives messages from other cells, right? So we receive a message here. Where do you think the message goes? That way, right? That way. This is called the cell body. It's like where all the cell HQ, it's like where the nucleus is and everything like that. So this doesn't look like a normal cell, right? But it is. Cells mitochondria, cells does all the stuff, blah, blah, whatever, right? This is just a very specialized type of cell, right? This right here is called the axon hillock. It's like where we begin to send the message, right? This stuff, like this stuff surrounding the entire thing, like this cord goes through. All this stuff surrounding the cord, this is known as myelin. M-Y-E-L-I-N, myelin. Myelin. And it's an insulating material. And these, what we call unmyelinated sections inside of the neuron, these are called the nodes of what? Nodes of what? Ranvier. Because he's French and pretentious. The nodes of Ranvier, right? This is the axon. And these are the nerve terminals or axon terminals. What does terminal mean? It means the end, right? Okay. Cool. Myelin, specifically, is produced by a type of cell called, does anyone know? They're called oligodendrocytes. So these are produced by oligodendrocytes. Interestingly enough, when you get to medical school and you do histology, right, you're gonna look at a cross section of a neuron that goes like this, and when you look at it, you're gonna see the oligodendrocyte, and like this is the cord, and it wraps around the cord like this, like a spiral, and it makes myelin, and it makes that beefy insulation around around the little cord, around that part of the cell, right? The axon, around the axon. So that cord is the axon, right? This is the axon terminal. That's the way that the signal. This is called signal propagation. You take a photo. So the axon is surrounded by myelin on its way down the axon terminal, right? So let's talk about it in the terms you need to know. This signal is received on the dendrites and transferred to the cell body before getting compiled at the axon hillock, traveling down the axon between the nodes of Ranvier to the nerve terminal where the signal is passed on. That's what you need to know, okay? But what did I just say? The signal travels between the nodes of Ranvier. So the one thing I want you to really get in your head and take away from this whenever you see it, the signal doesn't travel through the axon the whole way. It jumps from node to node to node to node. It jumps, right? Why does it do that? Because it's faster than traveling down the whole thing somehow. It's somehow faster than traveling down the whole thing. It makes all the conduction quicker. Now, guys, this is one thing I say to all my students. Orgo, MCAT, bio, everything, physiology in medical school. Think of everything in science as dramatic as possibly can be. You think you know how fast it's happening, you jump. It's 1,000 times faster, 1,000,000 times faster. I was called Dr. Litzy. Professor Litzy once gave us an example. This is glucose, right? This is glucose. And glucose can close itself into a structure like this. And it closes itself, opens itself, closes itself, opens itself, closes itself, opens itself. How often? I would guess. How often do you think that happens? And it closes and opens, right? How many times a minute does that happen? My guess is like 6, 7, a billion. As they go closer, yeah. This happens 1 million times per second. So 60 million times a minute. Glucose closes and opens back up. It closes and opens back up. I can't comprehend that. That's incomprehensible. I don't even know what a million of anything looks like, right? I wish I did. Like it's probably money, but I don't know what a million of anything looks like, right? So that is incomprehensible. And that is just a molecule opening and closing. This, we're talking about electrical signal conduction. This is ridiculously fast, right? So when you think about how quickly this jumps from thing to thing and how quickly this goes and like propagates, we're going to talk about in a second, it is lightning quick. Because if it wasn't, you would be dead. You'd be dead, right? I tutored some of the NS1s over at Downstate, and I tell them like every single time I turn to them, I say, if it didn't, you'd be. They all say in unison, everyone, dead. Okay, you'd be dead, right? Okay, does everyone have that down? Is that? All right, let's keep talking. So, like I said, myelin, the biggest thing, it's an insulating material, right? So when you talk about demyelinated sections of the axon, those are nodes grandiae, which the signal can propagate down. So what does insulation do? It stops the signal from being transmitted, right? So since the whole axon is surrounded by insulation and those specific points aren't, the signal will go through those points. It's the easiest path of conduction. Okay, cool. Neurons are not physically connected to one another. This is the axon terminal of one neuron. And I'm going to zoom in on this specific part, right? And this is going to be the dendrite of the next neuron. And I'm going to zoom in on this specific part. So this is the axon terminal, and this is the dendrite on a very, very, very microscopic level, right? What is this space between the two of them?
Synapse.
This is called the synapse. The synapse. It's called the synapse, right? Or the synaptic cleft, right? Well, okay. The nerve, this is technically called the synaptic cleft. The nerve terminal plus the synaptic cleft plus the next dendrite is the synapse, right? And sometimes we say the two nerves synapse onto one another. We use it as a verb as well. They synapse onto one another, right? Okay. I don't know why they start talking about like, yeah, I kind of get it. Okay, so we're gonna come back to this, right? But I want you to know that that's how that happens. Because before we talk about signal propagation through a synapse, we need to talk about the different types of neurons and the different actions of neurons throughout the body. Everyone good? Cool. Trust me, this chapter goes quick. It's really short. Like all the long chapters at the beginning, it's like a cell, reproduction, blah, blah, whatever. These go quick, super quick. Okay. So multiple neurons can be bundled together, and that's called nerve, right? And you have sensory nerves, motor nerves, and mixed nerves. There's sensory motor. Some of them are just motor, some of them are just sensory, correct? Okay. So the central nervous system is the, if you listen to Schoolhouse Rock, the central nervous system is the brain and the spine, right? That's what it is. The central nervous system is the brain, and they say spine because they're talking to children, is the brain and the spinal cord. That's the central nervous system. The peripheral nervous system, right? The peripheral nervous system, central nervous system, peripheral nervous system, right? Whenever you see CNS, DNS, that's what we're talking about. Central nervous system, peripheral nervous system. When we talk about that, the central nervous system, the peripheral nervous system, we're talking about the difference between the brain and spine slash the whatever else. Anything else that exists inside of the nervous system. So all the peripheral nerves. So they make like a little note inside of the book where in the peripheral nervous system, all the cell bodies are trapped, like synced into ganglia, and the central nervous system, they're split into tracts. You don't necessarily know that until you get to like medical school. And then we talk a little bit about glial cells. So glial cells are just like cells inside the nervous system that support the nervous system and blah, blah, whatever. In your books, they make a very, very specific distinction between like astrocytes, epithelial cells, microglia, oligodendrocytes. I don't know enough about neuroanatomy yet to properly teach that. That's the second time they say that. I'm good enough at this stuff to teach it to you. But they're just simple facts. You look at the book, you write them down, you're good. Okay? So look at your books and just look at the cell organization of the nervous system, right? Now, this is where I start to lose people. And it's funny because the first time I ever encountered this, I was probably 16, and I'm still learning about it to this day. I still don't know everything about this. We need to talk about action potentials. Who knows the action? What an action potential is? Any action potential. Who doesn't know what an action potential is? Okay, so we're going to talk about action potentials in its entirety, right? So I'm going to draw a graph. And here we're going to have voltage. These, remember, the nervous system is electrical conduction. And here we're going to have time. As time goes forward on the x-axis, right? So this is called an action potential. Before we do that, we need to talk about the inside and outside of cell. Here is the cell membrane. Inside the cell, there's a collection of positive and negative charges, right? And outside the cell, there's a collection of positive and negative charges. When we speak of the voltage of the cell, what we are saying is the difference between the inside and the outside. So voltage equals in versus out, right? Because what is anyone who took physics? What is voltage? It is an electrical conductive differential between 2 points, right? A 9-volt battery. There's a 9-volt difference between positive and negative terminal of the battery, right? When we talk about voltage of the cell, we're talking about the inside versus the outside. So what we call the resting potential of a neuron, it's at negative 70 millivolts, meaning that the difference in electric pressure or movement or whatever it is, whatever you want to call it, between the inside and the outside is negative 70. So is the inside more negative or more positive? The inside is more negative. Right. So that is where our action potential begins.
Why is it so short?
All right. What happens is that the dendrite receives signals, right? And those signals begin to do something called depolarization. What is depolarization? It's making the cell more positive. Right? It's making the cell more positive. So as the neuron gets signals, this little slope begins to climb. Right? Okay? Yeah? You should be taking this graph down, by the way. This graph we're going to build up. And it's going to climb until it reaches something known as what? Does anyone know? The point of no return? It's called threshold potential. I'm going to write A, and you're going to write A. This is threshold potential. And this might not make a lot of sense right now, maybe just gibberish, but wait, we'll put it all together at the end. Threshold potential is at roughly what? Negative 50 millivolts. Negative 50 millivolts is threshold potential. Why is it called threshold potential? It's because at this point, there's no more going back. A full action potential will be realized, right? So in this segment, we're going to call this segment 1, right? We're going to explain it later. That's segment 1, right? At threshold potential, we're going to climb, we're going to reach an apex, we're going to come back down, and we're going to come back to here.
Yes, sir.
That is the whole action potential over time. And now
Yes,
we need
sir.
to explain what happens at each and every single one of these points, okay? Don't get nervous. This is 2, and this is like what? Positive 35, right? This is section 3. And this part right here is section 4. Section 4 is really important, right? So now what we're going to do is we're going to explain those different sections of the graph, right? So section 1 is, and what neuron are we talking about here? Which one? Are we talking about the one that's receiving the signals, or talking about the one that's sending the signals? Talking about the one that's receiving them, right? So it all starts, let's say that this is the neuron, right? And this is the axon, and this is its terminals, right? It all starts with receiving signals where? Receiving signals on the dendrites, right? So as it receives signals on the dendrites, those signals begin to depolarize the neuron, meaning that it's becoming more positive, right? Depolarization makes the cell membrane more, or the cell more positive, right? And more positive means that there's less negative charges with respect to the outside, or there's more positive charges with respect to the outside. You guys following? Yeah? So number 1 is excitatory. Excitatory because we go in the positive direction, right? Signals from the, or sorry, not from the, like it could be from another neuron, or it could be from the brain, whatever, right? Excitatory signals begin to act on the dendrites and depolarize, make positive, the neuron. And this leads us to threshold potential. That brings us to threshold potential. And once we're at threshold potential, there's no going back. The full action potential is all or nothing, right? So if you don't reach threshold potential, it's nothing. If you do reach threshold potential, you go. Does that make sense? Cool. Section 2. If threshold potential is reached, voltage gated sodium channels open and allow sodium influx. Allow sodium influx. What does influx mean? Influx means things entering the cell, right? So sodium is what? Positive or negative? Positive. It's an ion, right? It's a positive ion, or what we call a cation, right? So those cations, and what? Okay, so break it down. Voltage gated sodium channel. It is a channel that does not open until a certain voltage is reached. What do you think that voltage is? Threshold potential, right? Okay, so until we're at negative 50 millivolts, that's the worst 50 I've ever drawn in my life. Until we're at negative 50 millivolts, those channels don't open. And once we're at negative 50 millivolts, remember, it's dramatic. They completely open up. And sodium is just allowed to flood into the cell. Why does it go in and not out? It's because the concentration of sodium outside the cell is far higher than inside the cell, right? And it's just following laws of diffusion. The moment you open those channels, it's going to flood into the cell. It's just going to completely destroy the inside of the cell, right? That's how it happens. Okay. So when it's allowed to do so, it will go. And after some time, it will close, right? And the sodium channels, I want you to write, I'm not going to put this on board, but write it down. The sodium channels are quick to close. They are quick to close, right? Because the third step is that voltage gated potassium channels open and permit potassium efflux. What is efflux? Movement out. Why is potassium moving out? Well, number 1, the potassium concentration inside the cell is far higher than the concentration outside the cell, so just following simple laws of diffusion. That's number 1. Number 2, the inside the cell is now positive. So you can imagine that there's an electro-repulsive force that forces positive charges out of the cell. Now, why doesn't the sodium leave? Well, that's because the sodium channels are closed now, right? But the potassium channels are open. So the electro-repulsive forces push the potassium out of the cell. Does that make sense? Remember, that for things to leave and enter the cell, you need a channel, unless you're just allowed potassium to the outside. Yeah, so voltage-gated potassium channels open and permit potassium efflux. Why? Because the potassium concentration inside the cell is far higher than outside the cell. And number 2, there's
this is happening to anybody else.
Voltage right now, positive 35 millivolts. When I say the voltage meant, is the difference between the voltage inside the cell and outside the cell.
Changes in the lighting in the video.
In the cell, and we're forced
Sorry, hold on. The changes in the lighting in the video is actually giving me like some headaches.
them out. But they can only get forced out through channels that are open. And right now, in this phase of the action potential, what we call the downstroke,
God.
right, or the repolarization, the potassium channels are open. This is also known as repolarization. And one more thing, a very specific term, hyperpolarization. Why is it called hyperpolarization? Because remember, negative 70 millivolts was the resting potential, right? But are we at negative 70 millivolts anymore? No, we're hanging out down by like negative 80, negative 85, right? So what gives? Why wouldn't the neuron just go straight back to rest? It's because the voltage-gated potassium channels, remember I said the sodium channels were quick to close, but yes, these are slow to close. These are slow to close. So instead of snapping open, snapping shut, they snap open, and then they're like, we're gonna close, it's gonna happen, oh my god, and then they close, right? So since they're given so much time before they close, and the potassium concentration is so much higher inside the cell than outside the cell, a lot of potassium leaves the cell, and almost too much of it leaves the cell, so it hyperpolarizes. All right, cool. This is known as hyperpolarization. It's 2 negative now, right? So in order to get back to resting potential, we have to go through number 4. So sodium, potassium, ATPase, or what's called a sodium potassium pump, activates to restore membrane potential. Remember sodium potassium pumps? 3 sodium out, 2 potassium in, right? And since you're kicking out, or whatever, it's something in that direction, but it's 3 sodium for 2 potassium, right? So since you're doing that, you're reestablishing this sort of gradient, and slowly over time, it levels back out to resting potential. What does this have to do with signal transmission? Everything. This is the signal. This is the electric signal that's getting translated down the line. So when we talked about before, look back in your notes, if you drew up a whole axon like thing, whatever, and you did it, I have a photo of it right here, right? So this is actually the book, this is what it looks like. So if you look over here, right, that signal is going to start here and propagate and propagate and propagate. The action potential happens at those different points in the neuron. What are those points called? The nodes around the A. I'm gonna check to see if my microphone's still on, because last week my fucking microphone died. An hour and 50 minutes. I had to re-record 45 minutes of lecture. It sucks, right? So propagate until we get the axon. At the end, the axon gets translated into a chemical signal, which we're gonna talk about in a minute, right? So that is the signal, right? Let's talk about specifics of what's special about the signal. The hyperpolarization. So there's a specific thing, and I make this connection all the time because the MCAT also wants to know this, but they don't really talk about it. There's something called a refractory period. Okay, so when men climax, when men ejaculate, right, they're unable to do so for a certain number of minutes or maybe even hours until after they're done, right? That is known as a refractory period. In the same sense, the neuron is refractory inside the time that it is hyperpolarized. It cannot send another signal, right? So this is known as the refractory period, or the time that's getting back up to resting potential, because you can't really wake it up until it's back at resting potential. So it cannot send another signal. Does that make sense? Cool. Yeah. This happens not even in seconds. This happens in fractions of seconds, right? Remember, quicker than you can ever believe. All right. Now, can I erase some stuff on the side over here? Yeah, I just want you guys to look at this. So there's two different types of refractory periods. Number one is an absolute refractory period.
Is it my monitor?
So this is no signal at all. No matter how many incoming signals the dendrites are processing, there's no action potential at all. The neuron's asleep. It's in a coma. It is practically dead. You don't even know it's breathing. It's not happening, right? Does anyone know the name of the other refractory period? There's absolute refractory period and a relative refractory period. A relative refractory period. The neuron will only respond to a strong enough signal. So if you poke the neuron, nothing's gonna happen. If you take a machete and chop its arm off, maybe it'll send a signal. Now, what do I mean by that? Okay, this part's phase one right here, right? Phase one actually kind of looks like this. So we're at resting. Everyone, like, squint your eye.
We will actually probably
Let's look at this.
slow it down a little
So
bit.
there's two different types of refractory periods. Number one is an absolute refractory period.
Maybe go normal here. You want to know what he's talking about?
So this is no signal at all. No
Let's speed
matter
up a little
how many incoming signals the dendrites are processing, there's no action potential at all. The neuron's asleep. It's in a coma. It is practically dead. You don't even know it's breathing. It's not happening, right? Does anyone know the name of the other refractory period? There's an absolute refractory period and a relative refractory period. A relative refractory period. The neuron will only respond to a strong enough signal. So if you poke the neuron, nothing's gonna happen. If you take a machete and chop its arm off, maybe it'll send a signal. Now, what do I mean by that? Okay, this part's phase one right here, right? Phase one actually kind of looks like this.
Yeah.
So we're at resting. Everyone, like, squint your eyes and really zoom in because I don't have much space over here. So it's receiving signals on the dendrite, right? And those signals may go like this, and like that, and like that. And like, it's like little humps, right? They add together, right? So that's a small signal. So if you're in this period right here where it's coming back to resting potential, and you got a really small signal, it's just gonna come straight back down. But if you get a huge signal, you might just kick straight up to threshold potential and get the rest of the action potential, right? Because once you're here, you're gone. Does that make sense? Okay, right? So now, going back to that all or nothing principle, right? So it's possible that you get a bunch of signals on the dendrites, but all of them only add up to like this, and this is threshold potential right here. They only add up to this, you fall straight back down. Or they only add up to this, you fall straight back down. But the moment they all add up to this, you're flying. Does that make sense? All or nothing. If you do not get up to threshold potential, you do not get a signal. Does that make sense?
Yeah.
Take a photo of the board, everyone. Yeah, what's up? Huh? Oh, so the neuron will only respond to a strong enough signal. A strong enough signal. The absolute refractory period is very early in the hyperpolarization. The relative refractory period is very late in the hyperpolarization. Does that make sense?
Yeah.
Very cool. And this is how we propagate signals throughout a neuron. This is through the neuron. What happens when we get to the end of the neuron? This feels so cool because, like, I always used to watch lecturers and like watch their lectures and stuff like that and see how they completely fill the board with information. And now, like, I'm doing it and it feels cool. It feels nice. Because I've been doing this in my little man cave of a room since like 2020. It's ridiculous. Yeah, it feels good. Are we all done? Does anyone else want to take a photo of the board before it goes away forever? Forever hold your peace. All right. Part of history now. What time is it? Oh my god. Dude, time really flies when you're up here. That's crazy. Okay, so I have the 6 steps right here the way I wrote them. So cell reaches threshold potential, voltage gated sodium channels open in response to depolarization. You reach positive 35, the sodium channels closed, and the potassium channels open. The voltage gated potassium channels hyperpolarize the neuron, and then the Na plus K plus ATPase or sodium potassium pump activates and restores resting potential. Okay, the reason I call it Na plus K plus ATPase and not a sodium potassium pump is because when people call it sodium potassium pump, they forget that it uses ATP. So whenever someone says, if any molecular says sodium potassium pump, I write this. So I always remember that it uses ATP. Fun fact, greater than 50% of the ATP you will ever produce goes into these pumps. That's how important they are throughout your body. Okay. By the way, for impulse propagation, you know how I said that the signal jumps through the nodes around the A? There's a specific name for that. Does anyone know it?
Saltatory conduction.
Yeah. Nice. It is known as saltatory conduction. Saltatory conduction. And that is when the impulse, quote unquote, jumps from node to node. Saltatory conduction. We need to talk about neurotransmitters and the synapse. Oh my god. Guys, I'm not gonna lie to you, I'm fucking dying up here. I'm so tired. All right, so this, again, what was this and what was this at the end of the last thing? This is very, very zoomed in. This is the axon terminal, right? And this is the dendrite. And these neurons have specific names. This is the presynaptic neuron, and this is the postsynaptic neuron. So creative. The one before the synapse is called the presynaptic neuron, And the one that's postsynaptic, or the one that's after synapse, is called the postsynaptic neuron, right? Pre and post. Cool. Neurotransmitters are stored, right, in membrane-bound vesicles of the presynaptic neuron in order to be released. So these are little vesicles, and there are neurotransmitters stored inside of here. And what happens is that when the signal reaches here, there's an influx of calcium, right? There's a calcium influx into the neuron that pushes these neurotransmitters out into the synapse, right? Good? Understood?
Yeah.
Yeah, so once action potential reaches the nerve terminal, voltage gated calcium channels open, allowing calcium to flow into the cell, and this pushes the vesicles out to fuse with the membrane, neurotransmitters are released into the synapse, right? So when you study general biology and you learn more about vesicles than I taught you, you will realize that vesicles are just little membranes floating around, little balls of membrane. And what happens is that when they get pushed, they fuse with this part of the membrane. They fuse, like this membrane, it opens up and it becomes an integral part of the rest of the membrane here and here, and then it pushes the neurotransmitter out. You guys imagine that? Look at this little ball, right? And there's the membrane right there. It fuses like this, and when it grows out, it spits out the neurotransmitters, right? Does that make sense? You guys can imagine that, right? So those neurotransmitters are spit out into what? What's this? The synaptic cleft, right? So the neurotransmitters, so look, action potential leads to calcium influx, can't spell, leads to neurotransmitter release into synaptic cleft. I know it's very small. So action potential leads to calcium influx, leads to neurotransmitter release into synaptic cleft. Does that make sense? Yes? Cool. Once the neurotransmitters are here, what do you think exists on the dendrite?
Receptors.
We have molecules and we have
Receptors.
receptors. We have receptors.
Receptors.
And the neurotransmitter will fit into the receptor
Ligand gated.
and produce an excitatory potential inside of this neuron. And if we have an excitatory potential that's strong enough in the neuron, what will happen? An action potential will travel down this neuron. And the next neuron, the neurotransmitter will be released onto that. And if the potential is large enough, it will depolarize and create, another action potential. That's probably really gross with the microphone, like burp in the middle of speaking. Okay, so neurotransmitters attached, depolarize if it's an excitatory potential, and then relate onto the next neuron, and the next neuron, and the next neuron until there's a neuron that fires onto something else, like a muscle or an effector organ or something, right? And those neurotransmitters are special, right? Those neurons hold special neurotransmitters in order to create changes inside of the target tissue. Does that make sense? Is everyone following here? Can you kind of imagine, can you map out how a potential moves through the nervous system? You guys get that? Okay. One very important thing, right, to talk about, and we're going to talk about the types of receptors later. We're going to get to that later in the chapter, later in the book. But one thing we're going to talk about is the mechanisms by which neurotransmitters, their signals are stopped or continued, right? Because you can't just have this shit all the time. You can't just have neurotransmitters fire on receptors all the time. There's three things that can happen. So are you guys okay with this? I'm going to erase, right? Cool. So what are the three things that can happen in order for a neurotransmitter, I just gave you one of the answers. In order for neurotransmitters to stop firing. So the first one is enzyme breakdown.
Enzyme degradation. Yes, sir.
Enzyme.
What's the second one? Come on, guys. We should know this. Anybody in the chat? Anybody in the chat know this one? No, nobody. Come on. It's
breakdown.
Presynaptic reuptake. Or presynaptic cleft reuptake.
Right? So the neurotransmitter is broken down by an enzyme. And if it's broken down, it can no longer bind to the receptor, right? And if it can't bind to the receptor, it can't produce any signal. Does that make sense? Cool. Very cool. Number two. And we're going to talk about this one in a second. I'm going to ask you guys a very, very large, over-the-top Jolly Rancher question. So prepare yourselves. This is called reuptake. And if you already know the question I'm going to ask, shut the fuck up. So this is when the presynaptic neuron will take back the neurotransmitter. Will take back the neurotransmitter. It'll bring it back into itself and recycle it and use it again. What's the last thing? So it can either get broken down or taken back, right? Or what?
Diffusion.
It can leave. Diffusion.
Outside the cleft.
It leaves. So here's the synaptic cleft. All the neurotransmitters, they leave. That's it. And they get broken down somewhere else in the body. So there's a disease in the body. It is called pheochromocytoma. It is a specific type of tumor. For anyone who's seen House MD, they talk about it all the time, right? A pheochromocytoma is a neurotransmitter releasing tumor, and it releases epinephrine and all that stuff, and all these like hormones that kind of upregulate your sympathetic nervous system, right? And one big way we diagnose it is by looking at the breakdown products in the urine. So we look for homovanillic acid and phenylmandelic acid, and we look for these breakdown products of epinephrine in the urine. And if we see a lot of breakdown products, that must mean there's a lot of neurotransmitter in there, right? And if it's too much, if it's outside of the normal amount, it might be a tumor. So that's important in enzyme breakdown. Here's your Jolly Rancher question. In patients who are depressed, patients with depression, they take a specific class of medications called SSRIs. Has anyone ever heard these? SSRIs, right? Maybe some of you have been on them, right? It's very common, right? Very common medication that's prescribed. I know a lot of people who have been on SSRIs, right? These are known as selective serotonin reuptake inhibitors. And another one is called an NDRI, a norepinephrine dopamine reuptake inhibitor. I want you to tell me why these work to reduce the symptoms of depression. Given only the information I've given you, I want you to tell me how they work.
Why would what? What's the question?
Work to reduce the symptoms of depression.
Reduce the symptoms of depression. I mean, I think you'd be able to fire more action potentials, right? And if you fire more action potentials, then let me think about this. Hold on. I think you fire more action potentials. Wouldn't that allow for more more cell responses, I think, right, for the target cell, depending on what the message is to a specific target cell to do a specific function, or to have a specific response, right?
Given only the information I've given you, I want you to tell me how they work. You're both correct. Did you catch?
I don't know what they said.
Oh, fuck. You're so damn far away. All right. Selective serotonin reuptake inhibitors inhibit the reuptake channel of serotonin in the presynaptic neuron. When this happens, selective serotonin reuptake inhibitors.
Oh, never mind. I'm completely wrong. Selective serotonin reuptake inhibitors. Serotonin reuptake inhibitors. Reuptake inhibitors. Actually, I think it's the opposite. It's going to inhibit the reuptake of neurotransmitters. To not allow a specific response. Oh, and that would stop the symptoms, right? Yeah, I think that makes sense. That makes sense to me.
Inhibit the reuptake channel of serotonin in the presynaptic neuron. When this happens, serotonin is allowed to stay inside of the synaptic cleft for longer. And if it can stay in the cleft for longer, it can act for longer and increase feelings of happiness and combat depression.
Okay. Yeah.
Same thing with norepinephrine and dopamine. That is how these drugs work. Understood?
Yeah.
Very good. You're done, guys.
That was way out. Okay. Never mind. I was thinking something else. No, that makes sense.
Don't go asking your therapist for SSRIs. Just because I said so. I will find out. So we talked a little bit about the organization of the central peripheral nervous system, and now we're going to talk a little bit about like more specific organizations there. So let's talk about the CNS and the PNS. We haven't talked about the words afferent and efferent. Afferent arrives, it brings signals into the central nervous system. So an afferent neuron is when I touch my finger right here, what is telling my brain that I'm being touched, that's the afferent neuron. It is arriving, the signal's arriving at the brain, at the central nervous system. The efferent exits. Whenever my brain sends a signal to a muscle to contract, that is sent by efferent neuron. The efferent exits. Sends signal to periphery. Interneurons are found between other neurons. They're located specifically in the spinal cord, and they usually control reflexive behavior, right? So basically, like if I touch a hot stone, right, that signal will be transported into my spinal cord, right? And in the spinal cord, there's a neuron that's going to fire back, but also relay the thing up to the brain, the message up to the brain. So this is my spinal cord right here. And what that does is you'll realize if you ever touch something that's hot, you'll pull your hand away before you can feel the pain. That's because of an interneuron. That's the reflex that happens, right? Sometimes like when you're walking, your leg buckles, you like go like this before you realize that you're tripping. That's another interneuron. It comes up to the bottom of the spinal cord, fires back, and then it's like sends the signal at the same time. Does that make sense? Cool. Yeah. Yeah. So efferent, it exits and it sends the signal to the periphery. Sends the signal to the periphery, right? We talk about reflexes, and then blah, 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 blah. Okay. Talk about some more divisions, right? So we have the nervous system, right? And the nervous system is obviously split into central and peripheral. The peripheral nervous system is further split into somatic and autonomic. Somatic is what's under your control. So I'm able to make a fist, I'm able to flick my finger, I'm able to lick my lips, I'm able to talk, I'm able to move my head. That is somatic, right? Autonomic nervous system is everything that's not under your control. My breathing, my ventilation, my heart rate, right? My digestion, right? Certain smooth muscles that are moving around, like my vasculature. Those things are autonomic. And the autonomic is split into two divisions as well. What are those two divisions known as? The most famous in here, like every psychology course about the nervous system. Sympathetic and parasympathetic. Sympathetic and parasympathetic. So, like I said, the somatic nervous system consists of sensory and motor neurons distributed throughout the skin, joints, and the muscles. There's sensory neurons, afferent, inferior, blah, 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 right? So that's like sensation and control of whatever's going on. Autonomics, right, are they generally regulate the heartbeat, respiration, digestion, glandular secretions, like how quickly you secrete hormones and blah, blah, and everything like that, right? It is independent of conscious control, right? And two very specific terms. In the autonomic system, there's two neurons in the pathway. The first one is called the preganglionic, and the second one is called the postganglionic. And the ganglion is just this collection of cell bodies that helps to process the information. I'm gonna write a finger over here. So pre-ganglionic and post-ganglionic. Right? Cool. There is a misconception among students everywhere that the nervous system, the parasympathetic and sympathetic branches, antagonize one another. Well, they do, right? But that one is active and the other one's just off. And that one is off and the other one's active. Like, that's not true. There is a level of sympathetic tone and parasympathetic tone that can be modulated at all times. Sometimes with stress, when I'm about to walk into an exam, or walk up to race a race, or walk up to do a certain procedure or something, right? My sympathetic tone increases, right? And right after I've had a meal, if I'm laying down in bed, if I'm relaxing, my parasympathetic tone increases, my sympathetic tone decreases. It's like controlling the volume on a little DJ turntable, right? So it's not that they are always against one another; they just function to do different things. So the parasympathetic nervous system, its goal is to conserve energy.
What do you say about the pre and post?
To conserve and the tone decreases. It's like controlling the right finger over here, right? And two very specific terms. Like I said, the somatic nervous system consists of sensory and motor neurons distributed throughout the skin, joints, and the muscles. There's sensory neurons, afferent, inferior, blah, 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 right? So that's like sensation and control of whatever's going on. Autonomics, right, are they generally regulate the heartbeat, respiration, digestion, glandular secretions, like how quickly you secrete hormones and blah, blah, and everything like that, right? It is independent of conscious control, right? And two very specific terms. In the autonomic system, there's two neurons in the pathway. The first one is called the preganglionic, and the second one is called the postganglionic. And the ganglion is just this collection of cell bodies that helps to process the information. I'm gonna write a finger over here. So pre-ganglionic and post-ganglionic.
Okay.
Right? That the nervous system, the sympathetic tone, the volume, to conserve energy, right? Or the other. Why did I say constricting the bronchi? Well, when you need it to be a lot easier to breathe, you need it to be easier to breathe to conserve energy, right? And it's associated with resting, lowering the heart rate, constricting the bronchi, right? Sleeping, stuff like that. One of those things is like the other. Why did I say constricting the bronchi? Well, when you need it to be a lot easier to breathe, you need it to be easier to breathe when you're doing a lot of heavy exercise, right? Right? So the sympathetic system dilates the bronchi. The parasympathetic system relaxes them. You don't always want them to be breathing like 100% efficiency. You don't always want something on, right? So, and this is mainly controlled by a neurotransmitter known as acetylcholine, or ACH. One big thing, it increases digestion, right? And the sympathetic system is activated by stress. That's not to say that your sympathetic system doesn't activate every single day. When you wake up, getting your body moving, getting your heart rate back up, getting you to be alert, that's all sympathetic stuff. That's technically stress, right? It's just not resting, right? And the main neurotransmitter here is epinephrine and norepinephrine. Epinephrine, norepinephrine. Listen to the things. Increase heart rate, increase blood flow to muscles, increase blood glucose, decrease digestion, dilate the pupils, relax the bronchi, and increase body epinephrine. These all sound like things where you're getting ready to beat the shit out of someone. Because that's what it is. It's fight or flight, right? You're gonna fight, you're gonna book it, right? Epinephrine also increases glucose metabolism, which we're gonna talk about later when we get biochem. There's so many things that happen in the sympathetic response within metabolism, right? Talk about reflex arcs a little bit and blah, blah, blah. And I don't think reflex arcs are that important, but we kind of talk about it, how like hits the spinal cord, comes back, and then signals the rest of the brain. And if we're not gonna talk And I don't think reflex arcs are that important, but
right there.
Your sympathetic system does one big thing. It increases digestion, right? And the sympathetic system is activated by stress. That's not to say that your sympathetic system doesn't activate every single day. When you wake up, getting your body moving, getting your heart rate back up, getting you to be alert, that's all sympathetic stuff. That's technically stress, right? It's just not resting, right? And the main nerve getting you to be alert, that's all sympathetic. To say that your sympathetic system doesn't activate every single day. When you wake up, getting your body moving, getting your heart rate back up, getting you to be alert, that's all sympathetic stuff. That's technically stress, right? It's just not resting, right? And the main neurotransmitter here is epinephrine and norepinephrine. Epinephrine, norepinephrine. Listen to the things. Increase heart rate, increase blood flow to muscles, increase blood glucose, decrease digestion, dilate the pupils, relax the bronchi, and increase body epinephrine. These all sound like things where you're getting ready to beat the shit out of someone. Because that's what it is. It's fight or flight, right? You're gonna fight, you're gonna book it, right? Epinephrine also increases glucose metabolism, which we're gonna talk about later when we get biochem. There's so many things that happen in the sympathetic response within metabolism, right? Talk about reflex arcs a little bit and blah, blah, blah. And I don't think reflex arcs are that important, but we kind of talk about it, how like hits the spinal cord, comes back, and then signals the rest of the brain. And if we're not gonna talk about reflex arcs, that's it. That's the nervous system. It's a lot more tame than you guys would thought, right? But there's one more thing I want to talk about before you guys leave. We're going to talk about biochemical warfare. Yeah, guys, like your MCAT tutor is just going to go on a complete random tangent and teach you about something that's completely unrelated to the test. Sarin gas is a biochemical agent. It's a biochemical agent. And it is something known as an AKI. Like that, AKI. And I'm going to explain what that means in a second. What has to happen for you to breathe? What set of muscular and nervous pathways need to be activated and deactivated for you to breathe? Well, the phrenic nerve needs to send a signal to the diaphragm, right? And the diaphragm receives that signal through the parasympathetic system normally, right? And it receives that signal via the neurotransmitter acetylcholine. Okay? So acetylcholine reaches the diaphragm and it causes contraction. Contraction of the diaphragm. Where's the diaphragm? It's right here. It's this little like globe looking thing on top of your liver and your stomach and everything. And when the diaphragm contracts, it pushes down, right? What does that make room for? The lungs. So the lungs can open up and inflate. So it takes up less space in the thorax so the lungs can inflate. So when the diaphragm contracts, the lungs fill. Okay. And then when the acetylcholine is broken down, right? ACH broken down, the diaphragm relaxes. And when the diaphragm relaxes, it pushes back up, the lungs empty. And that causes you to exhale. ACH breakdown is monitored by an enzyme known as acetylcholine esterase, or ACE, ACE, right? ACHE, ACE. If you inhibit this enzyme, you are known as an acetylcholine esterase inhibitor. And that's what sarin gas is. It's an acetylcholine esterase inhibitor, an AKI. So when people use this gas against other people and they breathe it in, they are breathing in a molecule that inhibits acetylcholine. And if you inhibits acetylcholine esterase, right? And if you inhibit acetylcholine esterase, you don't inhibit acetylcholine esterase, right? And if you inhibit inhibit acetylcholine esterase, right? And if you inhibit acetylcholine esterase,
you don't break it down, so your lungs are just filled with air.
You don't get this ACH breakdown, right? Remember, this is number one of breaking down neurotransmitters or stopping their signaling. So if you don't break down acetylcholine, it will constantly fire on the diaphragm. And if it constantly fires on the diaphragm will stay constricted and your lungs will never relax. You will die.
Yeah, sounds right. You can't relax the lungs.
Because you can't exhale. Your blood will become acidotic. Your entire body will get no oxygen, and you will choke to death on your own breath.
Oh That's wild.
That's how serum nerve gas works. So that was like we've already spoke about number 2, right? The reuptake. This is number 1, the breakdown. So what happens is that acetylcholine esterase breaks down the acetylcholine so that it can no longer fire on the postsynaptic neuron. And that causes the relaxation. And if that didn't happen, you would die. Thank you guys so much for listening. I will have this uploaded very soon, along with the little sheets and like any corrections that I find next time. So we're done with 4 chapters. We're moving quick. Next time, I'm going to introduce you guys to some study methods. I'm going to teach you guys how to use Anki, like what it is, how to approach it, how to sort of like organize all this information in your head. And other than that, I would like for you guys to be proud of yourselves because you're keeping up and you're doing great. So thank you for coming, and I'll see you on Thursday, same time. No problem. We are live. Hello, everyone. Welcome back. This is going to be day 4, chapter 5, right? So we're pretty much we're getting through information pretty well. The bio book has 12 chapters, and we should be done quicker than you guys expect. So congratulations once again. You're keeping up. You're doing well. In terms of where we are in terms of uploading, like the people back home, I have not yet gotten to uploading the chapter 3 and chapter 4 videos. YouTube's been giving me a bit of trouble with processing, but I'm trying to get that to you guys as quickly as possible, just so that you guys have that. Also, if you want to reference back and rewatch those and everything, I still do need to re-upload and record the last part of chapter 2, which shouldn't take me too long. I'm just working on getting my whiteboard situated in my room, and then that should be done very soon. In terms of just program stuff, we will be talking, someone mentioned in the crowd, we'll be talking about medical school admissions soon and like how the admissions process works and et cetera, et cetera. I'll probably make that a separate video, just like not one lecture. And I'll just be talking about how to apply to take the MCAT, what you have to do to take the MCAT, you have to bring what you need to know, how many classes you need just in order to like, actually perform well. You guys have my study schedules in the drive if you're on the email list. If you're not on the email list, come see me at any point when you're about to leave or like after the lecture, and I can get you on the email list. It's also, the sign up form is also in the email every year. Okay. So let's begin by doing a little bit of vocab. So number 1, we have glands, right? So gland.
There you go. He's starting now. Yep.
Are basically specialized structures in the body that secrete hormones. Right? Glands may synthesize and secrete the hormone, or they might just secrete it, right? But then in order to effectively talk about endocrinology, we need to know what a hormone is. And that's very simple. It's a signaling molecule. It's a signaling molecule that is released into the bloodstream to act on tissues. I'm trying to write a little bigger. Is this better? Can you guys read this a little better than normally? All right, cool. And then we have receptors, right? What are receptors? They're just the things the hormones bind to, right? So they're the sites of action. Sites of hormone action / attachment. Right? Those are receptors. Now, there are different types of hormones that bind to different types of receptors, right? So before we talk about the types of hormones specifically, we need to talk a little bit about chemical structure in general, right? Okay. So, who here, just by show of hands, knows the words or is familiar with the words polar and nonpolar? All of you guys, right? So all the hands in the room went up. Okay. So someone tell me, what is a polar compound? And what is a nonpolar compound?
Polar compound, readily dissolved in blood or water. Non-polar, it's not.
Anyone? Yeah, so there is an uneven electron distribution within the molecule. And when we talk about it in chemistry, right, we can say that certain parts of a molecule are more polar than others, and then certain parts of a molecule are more non-polar than others, right? So we say that, but normally when we're talking about hormones, we want to talk about hormones as a whole, right? We want to say, as a whole, is this hormone polar or non-polar, right? So that's how we classify them. And then obviously the non-polar compounds would be the opposite of this, a relatively even electron distribution across the whole molecule, relatively even, right? Okay, cool. So that was just to go over the ideas of polar and non-polar. All right, so since we've now introduced that idea into our heads, let's talk a little bit about what are the classes of hormones and why did we talk about polarity in the first place, right? So let's talk about our classes of hormones. So number one, we have peptide hormones.
Yep.
We have peptide hormones, right? Who remembers what a peptide is? Yeah, so it's a chain of amino acids, right? So it's a precursor to a protein, or it's a protein itself. So these hormones are made up of amino acids. Two lowercase a's for amino acids, right? And then what's the backbone? Like what's the general structure of an amino acid? It looks like this. And then sometimes that R group is polar, and sometimes that R group is non-polar, right? You can see charges over here, right? You can see nitrogens and oxygens, right? And then there's R groups, sometimes the charges are like, not sometimes charges and then sometimes just polarity, right? So this overall is quite a polar compound, right? Now, if you strung a bunch of these together and made a hormone out of it, that would be quite a polar hormone, correct? So the peptide hormones are very polar. They're very polar, right? Given that they are very polar, what is this hormone incapable of doing? So the hormone, right? Let's say insulin is a really good example of a peptide hormone, right?
Yep.
And it comes up to a cell. What is it incapable of doing?
Passing through that cell, getting inside.
Sorry? Yeah, so it can't pass through the cell membrane, right? So it's not going to get past the membrane. So that means it cannot pass through the cell membrane. So if it can't pass through the cell membrane, it can't function on an intracellular receptor, right? It has to function by a extracellular receptor, or what is known as a membrane-bound receptor, right? So peptide hormones normally function on membrane-bound receptors. Why? Because they can't slip through the cell membrane, right?
Yeah.
And what are some examples of these receptors, right? Like you're going to hear these words a lot. G-coupled protein receptors, and then receptor tyrosine kinases, right? So those are examples of extracellular or membrane-bound receptors, right? What does extracellular mean? It means outside the cell, and then intracellular means inside the cell, right?
Yep.
And so intra means inside, and then inter means between, right? Between. Does that make sense?
Yep.
I wrote this wrong. GPCR, G protein coupled receptor.
Yeah, GPC.
So normally, the action of this hormone goes, hormone binds receptor, which makes a conformational change, a change in the conformation of the receptor, right? Which activates enzymes, binds receptor.
I gotta make a new page. I don't like making any page, but there's not enough space.
Which makes a conformational change, a change in the conformation of the receptor, right? Which activates enzymes.
I can't spell today.
Which creates secondary messengers which eventually lead to a change in gene transcription. Because that's what hormones do. The end goal is a change in gene transcription, because how are you going to change things at a cellular and tissue level without changing the genes, right? And in the case of insulin, insulin will upregulate the formation of glucose transporters, right, to get more glucose into the cell. Does that make sense?
Yep.
So your secondary messengers, these are really important molecules that carry those signals into the cell from the outside. So what are some examples of secondary messengers?
Not epinephrine. Camp, inositol phosphate, calcium.
That you might come across? Anyone? Oh. A really, really famous one.
Camp.
Cyclic AMP. Camp, right? What else? Inositol triphosphate, or IP3, and calcium.
Oh, that's IP3. I did not know that was IP3. Inositol phosphate 3. That makes sense. Calcium 2+.
So an example in my notes right here, a hormone activates a protein called a receptor. It activates or inactivates adenylyl cyclase, which raises or lowers the levels of cyclic AMP. Cyclic AMP binds to an intracellular target, which is protein kinase A. It phosphorylates whatever target, and then that causes a change in the gene expression. Does that make sense? Do you guys need to know the very, very granular information about how a secondary messenger does its work? Maybe. It sometimes comes up on the MCAT, and it's good knowledge, right? However, this is the general information you need to know in order to understand what's on the paper in front of you. Once you have a full comprehension of this, then you can worry about knowing, well, what exactly does adenylyl cyclase do? And what exactly is phosphorylation of like protein kinase A? And like, what is a protein kinase in the first place? You guys remember what a kinase is? I told you a couple weeks ago, or like a couple days ago?
Yeah.
Phosphorylates what? Phosphorylates using ATP. Whenever you hear the word kinase, it phosphorylates something using ATP, right? And why do I keep making that distinction? Why am I trying to drill this into your heads? Well, it's because the MCAT always asks you, in the passage, they'll say, blah, 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 phosphorylates blah, 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 and creates ADP as a product, right? So if it creates ADP as a product, it used ATP, right? And then in the question, they're going to ask you, well, what kind of enzyme did that step of the reaction? You know it's a kinase immediately. They'll say a phosphorylase and a phosphodiesterase, and then they'll say a phosphatase, and then they'll say a kinase. And they all sound the same, right? But you know for sure it's a kinase because it said that one of the products was ADP. That's how you know it's a kinase. What's an enzyme that phosphorylates without using ATP? It uses inorganic phosphates. That's called a phosphorylase. And then an enzyme that breaks a phosphate group off a molecule, that's called a phosphatase. Right?
Why would the product be
One more
ATP if
thing.
you used it up, though?
About peptide hormones is that they are what? They're soluble in polar things. So like water, right? They're soluble in water. And if they're soluble in water, then they're soluble in blood. They're soluble in blood. That's a very important fact to remember. That they can just be secreted into the blood directly and carried throughout the body in the bloodstream. It's a very important fact to remember.
I can't see that.
So nonpolar hormones need some sort of protection or what we call a carrier molecule as they move through the bloodstream. And we're going to talk about those when we get to nonpolar hormones, which is actually next. All right. So are we good? You guys good? You got everything? Cool. So we talked about peptide hormones, right, which are very polar hormones. And now we're going to talk about very nonpolar hormones.
All right, guys, I think we're going to have to stop here, unfortunately. At least just with the video. I am going to do Funky next. I have to make sure I upkeep that. Yeah. Yup. Because I'm not going to let that go through whatever happened the last 2 days. That was way too much. So let's finish this up. And then we might call it after this, to be honest. Also, for tomorrow, I won't be able to stream. So I have I have work things, so yeah. And then maybe South Street, maybe a little bit. It depends if there's time. If not, then that's fine. And then maybe sometime on Sunday. But anyways, let's go and start. I'll be back though. I need to pee. questions, and then I'll feel happy because the thing is that I'm almost, because I don't think PS is gonna take me that much time. Like I think I'm doing pretty well in psychology. Anyways, let's go do it. See how fast we can do it. Start the clock. Go away, dude. Go away. No. Griffin's maybe? Arson is heavy. No, Arson is safe. I shouldn't say. TRH? Yeah. TRH could be down as well. What's a little bit? For sure. SVT plus, IP3, instead phosphate 3, and thrombocytes, erythrocytes. Oh man, I said thrombocytes. What are you talking about? Ability to transfer lactose, transport lactose. Elongation. Yeah, elongation. Degenerate and ambiguous. Downs. Okay. And it's granular. Heck yeah, man. Good stuff. Good stuff. Erythrocytes. Yep. Oncogenes of monocycline. Mutations of oncogenes. Non-competitive. Left. So left. Bicuspid. Or mitral. Cell division. Cell motility. Intermediate filaments, keratin and desmin. Intermediate filaments do not participate in cell motility. Hair and claws? Claws? What the heck? Large portion of nucleic acids, smaller silver proteins. Left atrium to the mitral valve. Growth releasing hormone stimulates the pituitary to release growth hormone produced by that. Viral application, which is the field of viral application and dispersion. Field of viral replication. Viral replication. Yeah. Oh, interfere. Okay. Virus infection, viral syndrome. Oh, that's pretty cool. I did not know that. That's actually pretty neat. Okay. Oh man, 13. Lions. Use compression forces. AFTB line. So the switch between small two pairs. Oxygen. Chromosome. Lysine list. K-lysine list contains carbon group, basic, hydrophilic. If blood pH is too low, respiration. No. If blood pH is too low, respiration will. Respiration will. If blood pH is too low. That's a good question. Well, let me see. I'm gonna say it's gonna increase, right? Yeah. Okay, that makes sense. Interferon. Gene proteins. All amino acids except cysteine have the S. So they all have S except cysteine, which is R. Generally develops into the ectoderm. Oh, whoops. That's a whoops on that one. Malaria is on the west front. Fair. Trachea. Trachea. Seriously. V max goes down, camo change, document emoji. This doesn't analysis as well. Type 2, diabetes mellitus. Type 2, diabetes mellitus. Methionine, methionine. AG, methionine. mRNA and proteins. mRNA and proteins. mRNA and proteins. That's a good question. mRNA? Wait, okay, let me see. So since it's mRNA, I'm thinking G1. Okay, yeah. So I'm thinking G1 then. G1 then S, and then G2 and then A. Size examination by basic session. Wait, basic session including maybe site. Yeah. Cortisol. It's gonna be beta memory, memory B cells, right? Yeah, memory. Oh, okay. Memory T cells. Oh, antigen experience T cell. Oh, okay. Should be increased temp, high temp. Mobilizing two membrane groups. Endocrine system, endocrine. And this has concentration of water across the membrane. Green matter should be memorized. Dendritic cell bodies. No, dendritic. The cell bodies of the neuron that are unmyelinated. Avery McLeodal, Avery McCarthy. Oh no, that's the difference. Oops. Plus 40 requires ATP. ATP more ATP. Memory T cells. Memory B cells. Redox structures and alcohol dehydrogenase. Yep. That releasing hormone. Erythrocytes. What is this? Oh. Wait. Adrenocorticotropic hormone. Adrenocorticotropic release hormone. Corticotropic release hormone. 30, 40, 50, 60. Wait. 30, 40, no, 30, 40, 50, 60. That's pretty cool. That's a pretty neat trick. Anyways, I'm gonna go periodic translation, and for methionine at the P site, mutations of oncogenes, cycling, size between tricuspid, between the right atrium, right atrium and right ventricle. We'll go again. Floppy baby syndrome. No idea. That's a good one. Oh, hypotonia. Interesting. Alright, granulocytes and granulocytes. Okay. Natural pacemaker. Genes of probability. Blues. Increases blood pressure and increases blood volume. Protein alters proteins on the surface of a pathogen. Oh, it's a pathogen like toxin. Also found in immunoglobulins. Okay, hypotonia. Cortisol. Stimulates the production and release of cortisol. Let's get this one. Let's stop it. Oh, cortisol. Glucocorticoids. Oh, it's ACTH. How come I always forget that one, eh? Adrenal cortex. To secrete glucocorticoids. Then glucocorticoids are where are you? Cortisol and cortisone. EG, wag, wait. Wag, uga, wa. Yeah. Wag, uga, wa. Background cap and ponytail. Please. Traditional CO2. It should fly through unicornotropic release hormone. Yeah. Oh, actually, okay, just ACTH, unicornotropic hormone. So just the other response. Yeah. Yeah. Cute. All right, guys. All right, I'm gonna call it here, but I'll finish it. I'll finish it for now, but I have to go. All right. Thank you so much, everyone. Take care. Peace.